बोल दो मैं पीपीटी चेक करने के लिए। अरे पहले हाय हेलो करेंगे सबसे। अरे वो इससे पहले जो थे ना। हम्म। फिर ये ला रहे हो।
Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. To the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions, and innovation leads to better health for all. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. Welcome to the Translational Health Science and Technology Institute, THSTI. place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all.
Check. This program still the time. We expect the arrival of all the guests. Thank you for your patience.
At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustainable. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. To the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics ready for clinical development. At TRF, we empower academia industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. The heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell. Welcome to Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here
overview of what you're doing in basic.
We have more time actually to actually learn.
You can probably follow it, but I can see whether it's industry academia collaboration or into labs, so I think good type collaboration might be better. And I simply my capital, but I expect to that's what's Cluster is not like this. All the others are also I mentioned that so that's great. So but I think also whether what's in your
Yes. Thank you.
Yeah, right. So
the question is now what can we do? So after generally is that right? Things that we
कर रहा है शायद तभी मैं कह रहा हूँ जल्दी आना भाई सारे नहीं ला रही लाई करीब
Yeah, broader, but if it's a particular
Weather conditions. There is a slight delay in the commencement of the program. Kindly be seated. We'll be initiating in another 2 to 3 minutes. Thank you. Good morning, everyone. We'll be starting in a few minutes. Could I request everyone to please settle down? A wonderful morning to all the distinguished guests. We request everyone to settle down and either to switch off their mobile phones or keep them on silent mode. We, Dr. welcome all of you 2025, the second industry business meet at Translational Health Science and Technology Institute, THSTI, an Institute of Biotechnology Research and Innovation Council BRIC under the Department of Biotechnology, DBT, Government of India. May I request Dr. Nithya Wadhwa, Senior Professor, to give a preface to Synchn 2025.
Good morning and a very warm welcome to the second edition of Synchn. Your presence today reflects a shared commitment to advanced biotechnology, forging strong partnerships, and accelerating the journey from scientific discovery to medical impact. Synchn 2024 marked our first formal step in engaging. It brought together over 150 delegates, including representatives from 67 industries, policymakers, catalyzed 14 formal agreements and several promising. Synchn 2025 builds on that momentum. This year, we are proud to share that over 100 industry representatives have registered, a clear signal of growing interest and trust in the translational capabilities of our ecosystem. With continued support from the Department of Biotechnology, showcases translational, also opens doors to collaborative engagement with THSTI and across the IBRIC institutions. This event also reinforces the importance of sustained academia-industry collaboration to achieve the national vision of Atmanirbhar Bharat and Viksit Bharat and policy stakeholders. Today is an opportunity for open dialogue to opportunities. We hope new conversations and inspires bold collaborations. Thank you for being here.
Thank you so much, ma'am, for setting the tone for Synchn 2025. May I please request our Executive Director, Dr. Please deliver the oral address. Thank you.
Yeah. Good morning, everyone, and welcome. Despite the weather being a dampener, it's very good to see this hall is full. So, I'm a diehard academic, so it's very difficult for me not to show slides. So I just put together a few slides because after last year is when we started this whole endeavor of reaching out to industry. I think it is very critical that we reach out to industry as collaborative partners so that we can take this whole endeavor of translation forward. So can I have the slides, please? Is this the one? Oh, okay. All right. So welcome again. And I just wanted to make a few general comments about the way I see. Technically, I can't call myself an outsider. I've been here for 1 year now, but still, I look at it from a different lens when it comes to how we can translate effectively. So this is something which I try to look at from time to time. So if you look at the drugs discovered in India, this is one metric of translation. We can consider that. So there are a few names, and for the practitioners, clinical practitioners, you can see that most of them are not used. Maybe there's just one which is of any use and which is prescribed recently, but most of the drugs are not used. So when you compare this with what happens in the United States, for example, they do 50 drugs a year. So there is a very big gap that we need to bridge here. And when we look at our scientists, are we somewhere far behind? Actually, this is a fairly good metric if you look at the number of publications, because you don't have to look at the number of publications per capita population. That's not a very good metric because you just need critical mass of publications. So if there are sufficient number of publications, we can make progress. We are not too bad on that metric either. So what is it that is really holding us back? So this is something which all of you are familiar with, the value of death concept or any technology, any drug, anything. But I think when you look at, say, drug discovery, or you look at uh, innovations in bioscience, there is more than one value of death. So, to my mind, when I look at this whole concept, there are three things that come to mind. So I think the first value of death is this. This is something which I encountered. P. Ram actually introduced this to me in one of his talks. So this is Donald Stokes was a bureaucrat in the old senior Bush administration, and they were considering how to stay ahead of the game when Japan was becoming a big power. They were innovating very rapidly, and they wanted to stay ahead of the game. And they decided they were going to make policy as to how to invest in this. So this is something which he came up, and this is a color version of what he presented in his book. So he thought that there are different ways in which we can do research. One is pure research just for the sake of knowledge, that is to exemplify Niels Bohr, for example. And then there is pure innovation, that is Edison, where he was not bothered about science, but he was bothered about tools can be developed, what products can be developed. And possibly the best research is like Pasteur. He identified problems in the real world and tried to identify solutions, get solutions for that. So if you look at it, most of us, most of Indian research, that is one of the things that actually is happening clinical research in India, most of it is in the innominate quadrant. It doesn't take anything anywhere forward. So we need to place ourselves firmly in this quadrant for us to make any rapid innovations. Of course, we need to invest in basic science, pure basic science research also, but then most of our focus should be in developing tools to address our problems. So I think that is the first value of that. We right in the beginning, and this is something which we in the academic community need to address. That is number one. Second thing is, all of you here, people from industry here, just look at the amount of money that is available for research. Just there is only biomedical research. We just have a fraction of what is available in the United States. But if you look at it closely, much of the funding, research funding, comes from industry in the West. Whereas in India, already the pie is very small, and most of it comes from government. And that is not sustainable. That's not a sustainable model. That is one of the fundamental reasons I think we need to have this kind of collaboration where we need to do less of, say, process-based, need to research. They need to really invest in R&D, real R&D that will deliver new products. So that is the second value of debt that you need to overcome. And the third is, of course, in the government. The regulation is a problem in this country, and we need to address it together. And we are trying. Harshit is here, many others are here. We are trying to do this to the best of our abilities. So these are three, I think, focus points that we can all work on. There must be other values of depth too, but these are, I think, three critical areas which, if we work on, we can really make good progress. So this is just for the first point is for academia. We do not want to do need-to research. We need to do research which is really critical for this country, not just avoid things which everybody else in the world is doing that is necessary for us. And we need to, this is for funders and industry and everybody else, we need to really be multidisciplinary. We need to be collaborative. We need to do research which yields something at the end of the day, and not in the very long term, but in the medium term, or maybe even in the short term. And funders, DBT representatives here, I think we need to really focus on funding large projects, a few of them, very well focused, multidisciplinary, rather than small, tiny projects which really don't yield much. So an example would be, you focus on one area and say that, look, this is what we want to do in the next 7 or 10 years. So that is what we need to do. And one of the ways in which we can do this is creating collaborative ecosystems such as this. And this is, I think, what I want to pitch for. So we have academia here, and we have partners in industry from industry. Like, I mean, many of you very kindly agreed to partner with us, and you are having presence here. And many of you will again come back here, and you'll be with us next year when we have Synchn 2026. We can say that, okay, there are many more people here then, and it'll be a magnet for other people as well. So that's all I want to say here today. And again, once again, welcome all of you here to this academia industry partnership. This is one of its kind, I think, in this country. And I think we can make this a benchmark for collaboration and developing a collaborative ecosystem. Thank you very much.
Thank you, sir, for your enlightening words. I now invite Dr. Jayanta Bhattacharya, Dean, BRIT THSTI, to introduce our institute to all the esteemed delegates gathered here at Synchn 2025. Please, sir.
Good morning. As we begin today's proceedings, what I would like to do is to briefly walk you through the journey that we have had, this institute had in last over 16 years. And also to give you a background about our position in doing translational research. So for a decade, if you see this, for about a decade and a half, Translational Health Science and Technology Institute has been driving force in India's biotech revolution, turning scientific breakthrough into the, it has been a forefront in driving biotech innovations in the country. With global collaborations, pioneering scientists, and a deep commitment to what Dr. Katrin said, to the public health, we have spent the last 16 years pushing the boundaries in this endeavor. If I speak about our strength, we have over 8 centers for excellence, as you can see on this screen. And the overall goal, our goal is to integrate in each of these centers, to integrate the fields of medicine, science, engineering, and technology into translational knowledge and making the resulting biomedical innovations in improving public health locally and as well globally. And to do that, we have created what we call supporting enabler or enabling pillars. And as you can see here, that these pillars plays very significant role in addressing all the activities, all the translational science that are being carried out in these institutions. And I just want to focus on two pillars which are getting added to this center, in this cluster. One is the Medical Research Center. My colleagues will be speaking more about what we plan to do in each of these pillars, in each of the centers. But very briefly, we have the Medical Research Center, which is going to be inaugurated tomorrow by the Minister, which has already initiated work in the area of cell and gene therapy, in working with our industry partner, Miltenyi Biotech. Ambitious goal to conduct the very first in the country, the controlled human infection study. And also this center will be also supporting what we call as early phase clinical trials. On the other hand, we are going to have this translational research facility that you see on your right, which is rapidly coming up. This is essentially this center where we tend to house small SMEs, the startups, the industry partners. We will be working hand to hand with the academic researchers in carrying out, taking innovations to the product development. And in this area, we have progressed significantly in the area of vaccines, biologics, small molecular drugs, and live biotherapeutics. And again, my colleagues in the subsequent sessions will walk you through all the activities that we have planned in these two different centers. To just give you a brief example of what we have initiated, this is an example which was essentially the DBT's Bio3 program. That are coming up in the translational research facility. One is monoclonal antibody biofoundry, as well as the live biotherapeutic biofoundry. Again, the details you will be hearing from my colleagues in subsequent session. So as Dr. Karthikeyan mentioned, last year Synchn brought significant success. THSTI at this NCR Biotech cluster provides a very important ecosystem for translating technological innovations into public health solutions by fostering collaboration and knowledge exchange between industries. We have examples with diverse stakeholders, developers, healthcare providers, and policymakers. This ecosystem will facilitate the development and implementation of new technologies and ultimately improving healthcare access, quality, and efficiency. Last year, Synchn, as I said, brought significant success in creating important partnerships in this endeavor. This year, we are looking to create few more in our journey to translate key innovations that will provide what we call real-world public health impact. I'll stop here. Thank you very much, and we'd like to finally acknowledge funders and the industry partners who are working with us hand in hand in this endeavor. Thank you very much.
Thank you very much, sir. Opportunity is everywhere. The key is to develop a vision to see it and an insight to seize it. Spread across 200 acres, the NCR BioCluster is equipped with cutting-edge infrastructure and expertise in innovation, biomanufacturing, bioprocessing, and pre-product development. In the spirit of Synchn 2025, let us hear from our industry partners on their ongoing collaborations and insights into translational research. Please join me in welcoming Dr. Prabuddha Kundu, co-founder and managing director from Preface Biotech. Please, sir.
To the distinguished guests and the organizers for having us here, and the wonderful audience. And thank you so much. I'll talk a little bit about our journey from Biotech. Many of you may know us. And then I'll give you a little bit of details that we're doing with THSTI here over the last couple of years. Some of you may research about for the last 21 years. We started with Panacea in Gurgaon. As many of you have pointed out, Molecule has never been our strength, but I'll tell you where some of got our inspiration. I was a graduate student at IIT Bombay when a visiting Japanese professor had come. He used that when he's sawing metal or wood, he uses different saws. Why do we highly trained biologists or engineers use the same vector host cell line for easy, difficult, or absolutely crazy proteins to express? And as you know, Japanese do not, by and large, have a very expressive face. So he gave me a small smile near that Maggie corner, and he said, because there is none. That became the sole call for some of us to invent or innovate a difficult to express protein platform. We chose yeast, Saccharomyces cerevisiae. Version 1 used to make GPCRs NAV 1.7 for the first time, never been done before, and hundreds of such difficult molecules. And then as COVID struck, we made the world's first triple antigen SARS-CoV-2 vaccine in the oral form, and we had to go to South Africa to do a phase 1. It was very successful, but by the time we understood how translation took place, the race was over. However, that gave us a huge impetus to now make version 2, which was for triple antigen vaccines and 4 antigen vaccines. I'll talk about them a little later. Subsequently, RNA companies started coming to us and saying, can you make RNA? So now what the version 3 has been, as we will talk about it slightly later, is that we managed to make the circular RNA in the cell, get it encapsulated in our virus-like particle in the cell, and therefore eliminating all sorts of RNA manufacture externally, no capping, no special buses, and therefore making a circular RNA VLP vaccine. Well, this does not happen overnight. It took about 20 years of learning how to take a molecule into clinics. And therefore, as early as 2008, we started partnering with companies who knew and were willing to teach us. Some of our successes has been taking, and perhaps many of you do not know, we took oral insulin into from phase 1 to phase 3 in the US. Yes, we failed the phase 3 trial, but we are restarting again, along with Orchid Pharmaceuticals through a joint venture. Similarly, with NRC Canada, we took in the recombinant CRIM molecule and have made a joint venture with them called Espra Therapeutics. So this is us. We try to take difficult problems and try to give it wings based on some of the capabilities that we've learned over the last 2 decades and capacities that we've built in Manesar. So with that, I would love to invite anyone and all of you to our new facilities in Manesar. Some of the proprietary technologies we made is the decrypt system, is the engineered yeast that I talked about. Perhaps one of the first novel genome-derived libraries out of the Indian genome. It's a French display library. 1 minute. And then the AXTEX40. Coming to some of the work that we've done with THSTI, we've worked on therapeutics, vaccines, and at present, tomorrow, we will be executing the an LOI to take the evaluating of safety and influenza vaccine candidates for animal challenge studies, and subsequently, possibly, a human phase 1 challenge studies. Of this flu vaccine, it's a triple antigen, it's a NA bias vaccine. It will be perhaps one of the first of the world with a fully length NA, HA, and M1. Some of the work that we are truly grateful for THSTI was enabling us on our SARS-CoV-2 vaccine, and at present will be the H1N1. This gives you a sort of a pipeline where THSTI has been supporting us actively on the 3 different programs, and we will be continuing with that. So with that, we are truly grateful to the partnership and the collaborative mindset of THSTI. And I will skip that. I think I've been told they're less than 30 seconds, but some of the data has gone into publications. And this is some of the work that we'll be subsequently working on at THSTI. So our vision, we're very happy to partner with THSTI and the Government of India to build a future where India leads in vaccine innovation, self-reliant, globally accessible, and prepared for every health challenge ahead. And with that, thank you very much.
Thank you very much, sir. Our next speaker is Dr. Shivakumar Natarajan, Director, Commercial and Business Development from Miltenyi Biotech. Please, sir.
Good morning, everybody. An absolute honor and privilege to stand here on behalf of Miltenyi. I've been given 5 minutes, and I guess it's what I can't say in 5 minutes, I can't say in 15 minutes. So I'll try to keep it short. As the previous speaker, I'll take a moment to talk about who we are and what we do in the world of cell and gene space, and what is it that we're going to do in India with THSTI. So this slide is filled with numbers. I just want your attention to just 2 numbers on this one. One is the one that's popping up now. We started off in 1989. And why this number is relevant is because we are possibly the first ones to enter into the cell and gene space in the world. When most of the companies were just galloping for the last 10 years, we've been in this space for almost 35 years. The second thing that we are very proud of is the second number, which says that every fourth person that you'll see in our organization is a scientist, works in R&D. So we're proud that we have presence globally all over the world, fantastic factories that you see on the screen over here, including a recent office that we opened in Hyderabad. You're most welcome to visit this facility over here. So 35 years, we've done some cutting edge research. What brings us to the conversation that we want to have with THST is particularly the one instrument that we'll talk about a little in detail, and that's known as the Clinimax Prodigy over here. For those who want to actually get a glimpse of what this is all about, you just have to walk to the next building. We've just installed this a couple of days ago at THSTI, one of the cutting edge therapy when it comes to CAR-T manufacturing. And for those who are not initiated on CAR-T, the uninitiated, I know I'm speaking to the converted over here, but for the uninitiated, CAR-T is nothing but the rocket science of cell and gene therapy, and we are experts in that proudly. To give you a perspective, to make CAR-T, they say that there are almost around 20 transfers and manual handling steps, which compromises the QC to a large extent. And this is something that can be easily solved with smart automation. And that is what we offer with our Clinimax Prodigy here. Now, Prodigy helps in smart automation when it comes to CAR-T manufacturing. And QC, which is an important parameter in CAR-T manufacturing, is supplemented by one of our flow cytometers here called as MaxQuant. You will find both the instruments being placed at THSTI very soon. And the purpose of placing them over here is what I'll be highlighting in the next couple of slides. So this is what Prodigy offers when it comes to CAR-T manufacturing. And then this is what MaxQuant offers when it comes to the QC part. And you can see how fully automated it is. So individually they are powerful, but together they are absolutely unbeatable. So what is it that these two instruments are going to do in THSTI? So Miltenyi Biotech India, based out of Hyderabad now, will now be accelerating a training curriculum at THSTI. And when I say training curriculum, you should meet some of my colleagues over here, fun-loving people. We love teaching. And we also have what is known as a Miltenyi University in our corporate head office. So we do have a pedigree when it comes to teaching. So we are offering training courses over here to certify you as experts in cell and gene space. Now you see here, this is going to be possibly the first hands-on training in cell and gene therapy that we'll offer in India, clearly to build on capacity. I was enamored particularly when I saw the pasture quadrant over here. So if you're inspired to do research, look no further than Miltenyi Biotech over here. And as I mentioned, we do have a strong pedigree when it comes to curriculum and teaching when it comes to cell and gene therapy, and we democratize knowledge to the fullest in this space. So what we offer over here, as you see over here, is we being global pioneers in cell and gene space for the last 35 years, in association with THSTI, under the aegis of the BIRAC DBT, you'll hear about it a little more later on in the day. We have come up with a training curriculum still in the works, just for the scientists who are working on CAR-T therapy over here. Talk about this to your colleagues and anyone who's interested in getting on board with the CAR-T manufacturing and understanding on what happens. And the list that you see over here are the ones who will benefit immensely from this training program. So this is a training curriculum that we'll be launching very soon. Now, how does it impact the CGT space in India? As I mentioned, this is the first of its kind when it comes to capacity building in India. We will have clinical trial readiness. You saw in Dr. Karthikeyan's slide the lead time that it takes in terms of completing trials. This is going to possibly reduce that number quite a bit. And again, you of course don't need a visa to come to Delhi, right? So you don't have to look forward for international trainings anymore. It's available right here in THSTI. So it is going to not just help you, but it is going to enhance hospitals and research centers be ready for CGT manufacturing in future. And more than anything else, it creates a workforce, right? So if you are at any point of time looking at skill building when it comes to CGT manufacturing or CAR-T manufacturing using Prodigy and MaxQuant, look no further, watch for this space. And as I mentioned, we are an absolutely fun-loving organization. Talk to us whenever you're here, and I'm sure it will be a memorable one for all of you, right? Thank you so much for your attention.
Thank you very much, sir. Now, may we invite Dr. Syed Khalid Ali, Chief Scientific Officer from Panacea Biotech, on the stage. Please, sir.
Slides. Yeah. Okay. Good morning. It's an absolute honor to be here and to talk about the collaborative projects between THSTI and Panacea Biotech. For some reason, the slides are not showing up. Okay. Yes. So I am Khalid, Chief Scientific Officer. This slide depicts the overall philosophy of Panacea Biotech. So I'm going to talk about Panacea just a couple of slides. We are pioneers, so we like to be the first in developing innovative products, and I'll show a few examples of this. We collaborate, we innovate to create value, and we do all of this so that we can enable people live better, enable better health, and help people live well and live longer. We are a fully integrated biopharmaceutical company which is in business for more than 30 years, and this slide depicts the impact that we have created over our existence. So we are one of the two companies globally that has provided more than 10 billion doses of polio vaccine. We have played a major role in eradication of polio from India and globally. We are world's first company to have developed fully liquid pentavalent vaccine, which is EZ5, DPT hepatitis B. And then again, world's first company to have developed whole cell proteosis based hexavalent vaccine. So examples of our pioneering efforts. We have several products over different therapeutic areas. One in particular, Synchn. So this actually originated from Punjab University. We took it, developed it, commercialized it, and it is in the market for several years. So this is the example of collaboration that I was talking about in the earlier slides. Okay, coming back to the major topic, which is Panacea THSTI collaboration. We collaborate a lot with THSTI at several levels. We enjoy a really good relationship with the management and with our friends and colleagues. So I'm going to, instead of everything, I'm going to talk about 3 vaccine products, which are listed here. And these are at advanced stage, about to enter clinical phase. And all of these are based on a novel plug-and-play platform, which was developed by Dr. Sweetie, Dr. Avasthi here. And this platform is actually based, it includes a viral non-structural protein, NSP. And this viral non-structural protein actually has a tendency to self-assemble and form a globular nanocage. And this is expressed in E. coli. Now, expressing in E. coli has several advantages, especially from global access perspective. So we can make this vaccine available for the entire world, developed as well as lower and middle income country. The way this platform is exploited for vaccine development is we stitch antigenic epitopes of our choice on C and N terminal, and we create translational fusion. And what we get in the end is globular nanocage of NSP with antigens displayed on its surface. And this platform is actually truly made in India. It addresses the Atmanirbhar Bharat motto, maybe even making in India for the world. It includes all the components here are indigenous. The platform from THSTI as well as formulation expertise and adjuvants from Panacea. The first candidate based on this platform is COVID VOC vaccine we call KeyVac. The antigen was designed and expressed here, and then preclinical studies were done here. You can see the efficacy results on the side. We took it to Panacea, we did process development, we did formulation development, we made tox batches, we applied and received permission from RCGM for toxicology study. Material is shipped, so tox study is ongoing. We have also received approval for GMP manufacture of this molecule, and hopefully by the end of this year or early next year, we will do a phase 1-2 study at the brand new MRC. So life has come full circle for this molecule. The next molecule based on this platform is a broadly protective coronavirus vaccine. Again, this is a novel molecule, single protein designed to protect against three pathogens, SARS-CoV-1, 2, and MERS. So briefly, we designed 20 antigens, tested them, and out of 20 yielded the desired results, and we are about to take them into the clinical phase after the stage gate review. Here again, process development and everything is complete. Finally, the third candidate that I want to talk about is the Mpox vaccine. If you remember, not too long ago, there was a outbreak of Mpox. So the team here at THSTI and then at Panacea quickly worked together to design an antigen which shows efficacy and neutralization against orthologous vaccinia virus as well as circulating wild type viruses. And the process developed, and then this candidate was transferred to Panacea. Process development is completed, toxicology is completed. We are waiting for
Today, I'm here to talk on our licensing and collaboration journey with THSTI. Just to share about my company, just one slide, Fileliner. We are a 23-year young organization headquartered in Ahmedabad. We started our journey as a pharmaceutical company way back in the year 2003, and then having a dedicated focus on probiotic and microbiome sciences, we established and dedicated subsidiary in 2011 as a Probioticals. Today, our company is supported by three important verticals, that is neurosciences, research in formulation, and biosciences. We already have an 18 global collaboration for a licensing of innovative sciences in the field of microbiome as well as nutraceutical and phytoceuticals. We are already having, catering over 125 companies across the globe, and we are a group of more than 550 people. Talking about my company, so our R&D is already a DSIR approved, and our facilities are FDA registered, UGMP compliant, FSSAI exemplar, etc., etc. So you can see that we are a commercial company, started understanding the need of a market, and then slowly moved into the research and innovation. If I talk about my collaboration, so we are pretty young with the THSTI. Our collaboration is, I believe I can say it's just a 9-month young collaboration. But yes, I must appreciate that, the way it has taken shape. Like, for example, we started our CDA, we signed CDA and we reached to a technology transfer just within 4 months. I think that is fantastic. Being a probiotic focused company, or microbiome focused company, we have been focusing on collaborating for indigenous microbiome strain for over 6 years. But unfortunately, we couldn't get anyone. We have been talking to global microbiome research center, but finally we could find Dr. Babatos and his team that we find a really good strain and which we are going to commercialize in coming days. The technology that we have licensed, that is a consortium of Lactobacillus crispus. Talking about at what stage it is, so we already licensed the strain and we have already reached to a pilot scale. So you can say we are expecting a commercial launch probably by Q3 or Q4 of 2025. So on one side, we say that it's a deep tech, may take 2 years, 3 years, 4 year period of time. But yes, if you plan really judicially, then I think you can also commercialize this product as soon as possible. Must acknowledge the efforts by Dr. Babatos Das, not only because of his scientific expertise, but the way he has taken entire lead. Sometime I can share that for the last 6-7 years we have been discussing with all the Indian scientists, global scientists, but the kind of a passion that he shares, kind of I think, unimaginable patient that is caring for microbiome science, and the way he has taken all the technology transfer process as well as the post-transfer, hand holding and, scientific development, I think that is commendable. And that's why I must acknowledge the team of Dr. Babatos Das and entire THSTI team. Obviously, I mean, it's not possible for one person to do that, but yes, I think the patient that he is caring, I think that is fantastic. So with this, I wish all the very best for this beautiful event of Synchn 2025. Thank you so much.
Thank you to all our industry partners for sharing their success stories and igniting optimism in the field of research. We will take a series of presentations that showcase potential areas for industry collaboration. Our investigators working across various domains and high-impact fields will now present research that holds strong translational promise. I would like to first invite Dr. Neeraj Kumar, Associate Professor, whose work focuses on innovations in biodesign and medical technologies. Welcome, sir.
Hi, good morning to all. And my name is Dr. Neeraj Kumar, and my domains for potential collaboration is rapid bacterial pathogen culture, and second domain is cell line development and process optimization. So, starting with the bacterial pathogen culture. So we all know that when we visit to the clinic and with some common clinical symptoms like cold, fever, and cough, and doctors suspect that you may have some bacterial infection or non-bacterial infections. But the issue is that there is no rapid diagnostic for pathogen identification and their AST profiling. So, and if we see that pathogen identification and AST profiling itself is a bundle of three independent domains. Like first, we need to culture the pathogen. Then this will grow the pathogen to the identification level, and then these pathogen will be used for the AST profiling as well. So if we see closely into this three processes, actually the pathogen culture itself takes majority of the time. So it takes 3 to 5 days to culture the pathogen so that thereafter pathogen identification and AST can be done. So based to our understanding, the biggest gap in this process is the longer time that is taken for the pathogens to grow up to the certain level. So if we see that the unmet clinical need in this situation is we need a diagnostic that is rapid, low resource requiring, as well as it is cost effective. And if you see the market numbers, so numbers for the market is also very interesting. The market of approx $8.8 billion. So working in the lab, we realize, or a majority of us know that when we culture the bacteria, they grow very fast in the lab. Okay, like if you are culturing E. coli or something like that, it grows too much fast in the lab. But when it comes to the blood culture, it grows very slowly. The situation is not same as we observe in the culture in the laboratory. So what we observed here that is if the pathogen is being cultured, if there is even a small amount of plasma or its derivative, it restricts the bacterial growth, the 90%. So that slows down its growth. And we tested this with the gram-negative and gram-positive pathogens. And so based on this, we hypothesized that if we can reduce the plasma or its component from the culture media, that will help the pathogen to grow faster. And second point, we hypothesized that if we can concentrate the pathogen number into a small volume and then take it for the culture, that can also help the pathogen to grow a little fast. Considering that, we observed that if we are concentrating 10 times pathogens and reducing the blood plasma component, that really helps the pathogen to grow faster. And based on these observations, we have already initiated a clinical evaluation of these protocols that can help to rapidly reduce or remove the plasma or its derivative from the blood culture. And we are getting some good results. So now we are open to identify or work with industry partners so that we can have some support for the productization of the observations. Second domain I'm working is like the cell line development and the process optimization. As my other colleagues said, that the CHO is one of the most commonly used cell line for industrial protein production. So need, as per our understanding, is that every company needs a high producer clone expressing well characterized, bioactive, robust protein therapeutics and with optimized bioprocess parameters. And the market is again here huge. So we have more than 10 years of experience in working with the CHO. So if any industry partner, industry is looking for some R&D component in the CHO, so we are here to work with you. And the domain is the stable cell line development, process optimization, again lab scale. And we are good at cell engineering, and we are also good at glycan optimization for improving bioactivity as well as the stability of the protein. So that's all from my side. Thank you. Feel free to talk to me whenever you get time or if you have interest in this. Thanks.
Thank you so much, sir. Let's hear from Dr. Santosh Bhattapati, Associate Professor who is advancing research in biomaterials and stem cell technology. Welcome, sir.
Thank you. No. Yeah. So we are working on two areas. One is biomaterials and also humanized liver models development. So first I'll talk about the biomaterials. So biomaterials, as you know, or it could be also called as a medical implants or medical device categories it comes under. So the recent problems are like chronic wounds, like diabetic, irregular wounds, having issues. And there are very current treatments like standard dressing materials are available, skin graft or standard medical therapies. But still, these medical therapies have not been able to resolve the issues of chronic wounds and irregular wounds. So, address these issues, placental tissue, how it could be used as a making a medical devices or making a surgical patches or hydrogel based materials. So the current market is also huge. Like in India itself, around more than $500 million around 2030, it's forecasting. So as I was talking about the placenta is considered as a medical waste. So it has the three portions. One is a placenta, amniotic membrane, and also umbilical cord. So here there are three products we have developed, and those are the placenta extracellular matrix based hydrogel. Next is the amniotic hydrogels, and third is amniotic membrane based patches. These developed materials are on the principles of like making a tissue or cellular, taking out the cells, and it remains only extracellular matrix. So extracellular matrix, as you know, this is a non-immunogenic, and also further increasing its mechanical strength by cross-linking strategies. So all these products are cellular, and some of the products as commercial, commercial available products are there. Those are cellular drugs, and in India, there are companies who is trying to bring it. But this is one of the properties, as I discussed. So second, the self-conducting advanced material. These are also for the similar applications. These are based on the natural as well as synthetic materials. Natural is like collagen, and synthetic is a MXN-based material. And these are developed on the casting method as a surgical patch material. So what are the applications of these products? The applications of the product like amniotic patch could be used as a general surgical patch material. And other hydrogel-based materials could be used for the diabetic wound, any kind of wounds where we can use the surgical patch material. So all these products, in vitro testing has been done, even preclinical studies have been performed, and we saw majorly hydrogels have given the better efficacy for diabetic wound healing, and also the amniotic patch could be used as a surgical patch material for any kind of wrapping as a wrapper for the any damaged tissue as a supporting material. So what we seek in this like commercial scale of production as well as the GLP compliant safety studies and enable clinical translation and commercialization of these products. And the second area we are working on the humanized model. As we know, some of the standard in vitro models have been used in the pharmaceutical industries are the major cell line based model like HG2 cell lines, and also the standard one of the gold standard is primary human hepatocytes cells. So these cell lines have issues, as we know some of the disadvantages of this. And to address this research gap, we are focused on the developing small molecule protocol where we can use a stem cell into differentiation into pluripotent stem cell. We have developed a protocol and is also patented. And within this protocol, we are able to make hepatocytes. And also, we are successfully tested that whether this liver models could be used as a for the making any disease model. So one of the disease models we have tested is a metabolic dysfunction associated steatotic liver disease. And so these models could be used for the disease model study as well as the hepatotoxicity studies, as well as for high throughput drug screening applications also. Thank you. So please feel to contact me. I will be here around in these meetings. Thank you.
Thank you very much, sir. Let's welcome Dr. Sweetie Sanu, Associate Professor, who brings expertise in virology, particularly in development of novel vaccines and therapeutics. Over to you, ma'am.
A very good morning, everyone. So I will briefly speak about the R&D that we are doing in the field of virology. And so for any translational work, so we are the scientists and faculty, and about 50 research personnel where our strong penchant is to understand the virus biology. So these are the viruses emerging and intermediate viruses that we are working with. And this understanding of the biology gives for the translational approach. Working for these viruses, not only understand its biology, its replication, or the structural aspect, but we also develop the virological assays, immunological assays, and preclinical model, which make it a complete package. And so that helps, for we believe that as well, this generation, the generation X, generation Alpha, smart vaccine. So for every vaccine platform, every virus may not work. So that fundamental understanding so to creating platform. So you have already self-assembled analytics platform, where structure guided that is like soluble recombinant protein. Some difficult viruses we for the vaccines. It took us more than 20 years to develop the vaccine. Still we don't have this vaccine. So there are many viruses. Some of the pathways may not work. So we hypothesize that for this number of viruses, the protein-like structural protein will be the solution to develop which can be of highly potent and can be one of us is working started so I won't go through the details of this but I would say that we years of development. So believe that this platform can helpful for developing viral vaccines for bacteria vaccines and other viral vaccines for poultry and also for parasites, fungi, and also for the aquaculture vaccines. So I would like to platform and also Dr. Professor THSTI. So we just heard about vaccines in the bacterial cell culture system because the vaccine, analysis, they understand very well, and based on the immune system, they can tweak very well. So this is the first time we demonstrated, hopefully we can demonstrate the phase 1 clinical trial of T-viral vaccine features giving protective immune response expressed in E. coli. And in E. coli, still we are getting correction about that. So then you can imagine that in the industry level, how easy it can be scaled. Slide that size, it can be from a structure. That means if you have a one battery, you can express 12 epitopes in N terminal and 12 epitopes in C terminal. So making highly antigenic vaccine. So these are already collaboration which we have done and then also develop the help. This is an collaboration with biotechnology and with the help of the pharmaceutical. This vaccine for phase 1 clinical trial. So way forward, based on this MSM vector platform, we have already initiated work for some of the viruses like common for Zika virus, and we look forward to develop this, expect this platform for development of vaccines against poultry and aquaculture viral vaccines. As I told earlier, that this platform, this bacterial expression platform may not be suitable for every virus. For example, influenza, which is heavily glycosylated. So for that, we also have this mammalian expression system, stable choline system, which shooted the work of the pandemic influenza H5N1. And we heard about from other faculty that we have the ferret facility. So from antigen design, preclinical development, we have an end-to-end solution for influenza. So we'll be happy to collaborate and demonstrate that the challenge team, which is from Thailand, it is becoming virus, which can be went in the BSL2. And another we are looking at the structural protein. It's a structure specific viral vaccines, subunit recombinant protein, and this has been supported in recently by National One Health Mission. And we are interested to vaccines, we can which is a seamless targeted vaccine for this rabies. And I don't have to tell a lot about why this vaccine and what are the defaults, but I will be very happy to discuss further. So this is one of our ambitious high-risk project that we will put forward is coming with us. With this, I would like to thank the startups and industry who come forward who believe in us as a collaborator, as have taken our supporting capacity, and these are, and I would like to thank all this funding, our Government of India and also extramural funding, and have helped in establishing different assets, preclinical animals, and in some of the vaccine development program. So I'm happy to take any questions or interactions. Thank you.
Thank you so much, ma'am. Let's hear from Dr. Shalin Vidasthana, Principal Scientist who specializes in computational biology and AI-driven drug discovery, bridging data science and life sciences.
Okay. Good morning. Good morning, everyone. So you are listening lots of about the biological part. Now I'm here because one of the revolutionary science
nowadays because of artificial intelligence and machine learning, which is integrated with biophysics. I am Shailendra Asthana, heading the team of structural bioinformatics, computational biophysics, and computer-assisted drug discovery part in THSTI. And this is our area of interest, which would like to highlight that we are not only working to understand the protein dynamic, protein-protein interaction, but we are mainly focusing on the current trend, which is protein and monoclonal antibody identification to establish the interface of identification from monoclonal antibody itself towards the small molecule discovery. The major SOP of our strength is to focus only on a specific area, because the major bottleneck of the industry is they are highly dependent on the resolved structure or the target proteins of the pockets. We are here moving at one stage advanced to understand how protein actually working, like giving the topic on allostery to unstructured to structure transition itself, which is very challenging. But here we have some success story where we reached up to up to animal model and established POCs. So if we talk about the methodologies, so one thing is very clear that we need the speed, but parallelly we need the accuracy. So if you count about the speed, none of the method better than at present is using artificial intelligence and in computational chemistry for development of the molecule. But the bottleneck is we can achieve the speed where lots of industries are working, but the problem is the accuracy. If you compensate this accuracy, then there is a compromise. So with this aim, we have a huge expertise. We developed conventional and accelerated methods to utilize along with the biophysical approach, how to merge with the existing AI ML based approaches. And with this aim, we established how to achieve. One side, if you see the left panel, the accuracy is compromised if you are using computational chemistry and artificial intelligence. In right hand side, if you get compromise the speed. So how to establish a common platform where you can achieve speed and accuracy both. And this is our SOP. And here I would like you must appreciate we are using these methods. We reached up to this area. This is a portfolio of our group. Lots of students, postdocs, they work together to achieve in this area. We are in the stage of lead optimizations. We are working very hard, means many hot topics out nowadays, which like checkpoint inhibitors, autophagy inducers, which is having a lots of scope in as a broad therapeutic applications. And now, other than anti-fibrotic using FXR, and USP7 inhibitor as an anti-cancer target, and we reached in the stage where we crossed, and now the stage which I'm showing, I have confidence that they are showing some phenotypic outcome. The mechanistic understanding, we at the structure level we are resolving to understand. However, the field cases we already resolved, which are on the top towards the main directions. So this is our portfolio we established here in THSTI. And one of the case study where we reached up to primate study. I'm showing one of the case example here. And this structure is a kind of hardcore dedication towards the small molecule discovery. Why? Because we are integrating amino acids with the small molecule. You can see this amino acid moiety, which is responsible for enhancing bioavailability and reducing the toxicity. And second, in the right side, bioactive benzimidazole scaffold, which is a well-established scaffold. We, by using the integration methods, we established how to integrate these two scaffolds to not only suffice the drug discovery properties, but to achieve the potency as well. This molecule, the best part is this molecule is host targeting the host proteins, not only single target protein, but more than one host target protein to achieve the selectivity and enhance the potency. Research which we are talking about, we can reach at the advanced level of the product outcome, but this translational outcome cannot be reached without the basic science. And this work started from the beginning, how we can identify the cryptic pockets at the base at the initial level, because the target is sirtuins. Sirtuins is having a longer homologs. So getting the exact homolog is one of the challenge. Second big challenge in the drug discovery is how to achieve the selectivity. In the second phase, these are the publications where we achieved and we demonstrated how to achieve the selectivity. Third case is now when you know how to do the designing, how to achieve the selectivity, the bottleneck is whether those translational information you can get from the protein dynamics and the structure can you translate into the small molecule? Yes, we did it, and we come up with the two small family in the different chemical subgroup, and that we have established in animal model, which we communicated in molecular medicine. So since it was already patented, we are free to establish this protocol to understand the mechanistic understanding at atomic and molecular level. While this exploration we came across that since it was discovered from the sequence, but the innovation process and the best part, which is the endogenous innate immunity, which is achieved through CXCR3 inhibition. So how, whether it is a possibility that this molecule can active by as a polypharmacological agent. So with this aim, we explore and try to establish the feature. Whosoever is working in the drug discovery area, they well know that developing a polypharmacological agent is a big demand, is an unmet need. The problem is we don't have the features. If we have the features to categorize the way, if you finalize the target, you can start from the beginning as a polypharmacological agent. So with this, we established this platform for polypharmacological features in this part. And that's it. This is the team which I would like to thank my students and the funding. Thank you so much.
Thank you so much, sir. And finally, let's welcome Dr. Supriya Deshpande, principal scientist who focuses on infectious disease biology and the translation of drug discoveries into scalable therapeutic strategies. Please, sir.
You. Hi. So I'm going to present and I'm going to talk about the potential industry collaboration in the area of therapeutic monoclonal antibody development. As you all know, monoclonal antibodies have tremendous application in the area of diagnostics and therapeutics. Monoclonal antibodies plays a crucial and pivotal role in the precision therapeutics provide to provide target oriented or target specific and effective treatment against variety of diseases. So if you look at this slide, these are the variety of diseases against therapeutic monoclonal antibodies are being used. And in our group, we are mainly working in the area of So we are working in the area of infectious diseases, and our goal is to isolate discovery and development of monoclonal antibodies against emerging and emerging infectious disease-causing pathogens of local and global interest. So coming to our accomplishments so far, we have isolated 23 monoclonal antibodies, out of which 5 antibodies are being discovered against HIV, and 12 novel monoclonal antibodies are being discovered against SARS-CoV-2 and its variants. And very recently, we have discovered 6 monoclonal antibodies against Nipah virus. All these antibodies are isolated from infected human donors, and these are all human antibodies. There is no need to humanize the antibodies. And out of these antibodies, some of the antibodies are already under product development in collaboration and engagement with the industries. Moving forward, under DBT's Biomaterials initiative, THSTI will going to establish first biofoundry platform in the country. So the goal of this biofoundry platform will be to develop novel monoclonal antibodies from sequence to drug substance in a pilot scale in a regulatory compliant manner, and which can be successfully taken forward for the GMP manufacturing. So our proposition here to invite the industries for partnering or joining hands with the THSTI in the area of analytical characterization, cell line development, process development, RCB preparation, and drug substance preparation. So we'll be happy to discuss further on this during the breakout session. Thank you. Thank you, and all.
Thank you to all our scientists for sharing their high-impact translational research and product leads. Now, it's a proud moment for us to share about the exciting new initiatives within the NCR Biotech Science Cluster. These facilities will further enhance and strengthen our translational ecosystem. I now invite Dr. Nitya, Senior Professor, and Dr. Sweetie Samuel, Associate Professor, to give us an overview of one of our new key initiative, MRC, the Medical Research Center. They will share how this facility is poised to strengthen the ecosystem and enhance the capacities of the NCR Biotech Science Cluster. Please.
Good morning once again. I'm delighted to introduce one of our most forward-looking initiatives, the Medical Research Center at THSTI. This center, backed by clinical-grade R&D infrastructure, is conceived to fast-track medical innovation from early discovery to bedside application. Over the next few minutes, I'll walk you through what we are building, what makes this center unique, and how we hope it will serve as a key enabler for accelerating medical innovation. As we celebrate growing innovation capacity in the country, there's a widening gap between early discovery and clinical or public health application. There's also an urgent need to accelerate the journey from bench to bedside. But this transition from the lab to the clinic remains a bottleneck. MRC was conceived to bridge this critical gap, create-oriented biomedical research. It aligns with the national vision of Atmanirbhar Bharat, strengthening indigenous innovation pipelines. Spread across 5 floors with over 26,000 square feet of space on each floor, the MRC brings together cutting-edge research platforms backed by THSTI's deep scientific expertise and clinical trial experience. MRC is not standalone. It's embedded within THSTI's ecosystem. What sets us apart is not just our infrastructure, but the way we've integrated discovery science with clinical operations. THSTI already has centers of research excellence in vaccines, biologics, diagnostics, therapeutics, and we bring together clinical cohort expertise, high-end research resource platforms, which we'll in some time, and CRO-grade services from the Clinical Development Services Agency. So the MRC is a 360-degree innovation to application ecosystem, ideal for turnkey translational partnerships. So this MRC is built as an integrated hub, offering a full spectrum of capabilities, and these include a cutting-edge CAR T-cell therapy development platform, India's first center for controlled human infection studies, a state-of-the-art early-phase clinical trials unit, expertise in managing large longitudinal and even multicentric cohorts, an AI-enabled advanced imaging center, a robust ecosystem comprising the CDSA, the biorepository, and data science for precision research. And together these create a seamless ecosystem that ensures regulatory-grade compliance, operational efficiency. Each function has been purpose-built, enabling scalable research partnerships with both academia and industry. I've already said in one of my previous slides that the MRC is spread over 5 floors with over 26,000 square feet of space per floor. So just to look at the layout of the MRC, on the ground floor, we have the biorepository and the record room. And on the landing floor, we have the clinical research unit where we will be receiving our participants. And then we have the robust support, operational support ecosystem of the CDSA data Data Management and Data Science Center. And on the floor above, then we have the Center for Controlled Human Infection Studies and the Early Phase Clinical Trial Unit, as well as the Center for Cell and Gene Therapy. The top 2 floors are vacant currently for future expansion. One of the most high impact centers at the MRC is the Center for Cell and Gene Therapy, the first of its kind in the region. CAR-T cell therapy represents a paradigm shift in cancer treatment, transforming cancer care not only in India but globally. At the MRC, we are building a bench-to-bedside pipeline that covers the full development cycle, from designing and developing novel CAR vectors to production of high-titer clinical-grade lentivirus particles, robust in vitro functional assays to screen CAR-T cells, and specialized preclinical animal models to evaluate CAR-T cell efficacy. Then what truly sets this platform apart is the GMP manufacturing facility, one of the first in the region, enabling in-house CAR-T manufacturing for clinical use. This will significantly shorten the development timelines and enhance regional accessibility. We are also expanding our applications beyond oncology into autoimmune disorders, where opportunities for innovation and collaboration are just beginning. Our clinical collaborations include AIIMS Delhi and collaborations with AIIMS Delhi and NCR hospitals. And we are also already engaging with industry partners like Miltenyi, Biotech, and other industry partners. Then the Center for Controlled Human Infection Studies, what we also call as CHIS. Among THSTI's most pioneering and ambitious initiatives is the Center for Controlled Human Infection Studies. CHIS is a scientifically and ethically regulated model where healthy volunteers are exposed to well-characterized pathogen in controlled clinical settings. By simulating natural infection in a carefully controlled ethical environment, CHIS enables unparalleled insight into disease mechanisms and allows early rapid evaluation of vaccine or therapeutic efficacy with exceptional speed and precision. Globally, CHIS has emerged as a transformative approach in early stage clinical development, and the CHIS Center at MRC will be the first of its kind for industry and innovation-led ecosystems. This means faster go, no-go decisions, smarter R&D intervention investments. It serves as a strategic tool to de-risk development and enables efficient dose optimization with fewer participants. And it also cuts the early phase development cost by over 80%. It also directly contributes to India's preparedness against infectious diseases. And this is just one schematic diagram of a design for efficacy study using the human challenge model. So with world-class infrastructure already built within the innovation to application MRC ecosystem, the Center for Controlled Human Infection Studies is now progressing towards establishing globally benchmark governance and ethical operations protocols to ensure the highest standards of safety and scientific rigor. And the center at MRC is built to global standards, not just in terms of infrastructure, but in governance, ethics, operational protocols, with the highest levels of scientific and volunteer safety. The center has 2 facilities. One facility is for the respiratory CHIS, and then there is a separate facility for enteric CHIS for diseases that are disproportionately affect the lower middle income countries, including India. Our first models focus on influenza and typhoid, diseases that are of national significance. And importantly, the center will enable Indian innovators to generate global quality data locally without depending on international trial networks. And for the industry, this is a rare and timely opportunity. Let's co-create a model where India not only develops for India, but also innovates for the world. The Medical Research Center also features a globally benchmark early phase clinical trial unit, which is purpose-built to accelerate clinical innovation. It is equipped to conduct first-in-human studies, phase 1 vaccine trials, dose escalation studies, PK/PD assessments, as well as bioavailability and bioequivalence studies. The unit will enable testing across a range of products from new vaccines to repurposed drugs to small molecules, biotherapeutics, monoclonal antibodies, and biologics, supporting faster decision-making and de-risk development. With end-to-end trial capabilities and regulatory aligned processes, we are well positioned to be the partner of choice for early-stage clinical research in India. One of our strongest differentiators is our cohort advantage, which is deep, diverse, and ready. With over 32,000 participants across pregnancy, pediatric, post-infection, post-vaccination cohorts, we have a proven track record of generating high-quality clinical and epidemiological data at scale. And THSTI has, in fact, led some of the most significant cohort studies, including Garbhini, which is the largest pregnancy cohort study in India and possibly even in the region, showcasing our strengths in participant recruitment, retention, community engagement, data science, and biobanking. By embedding these capabilities within the Medical Research Center, we offer a unique edge. The Clinical Development Services Agency is India's only publicly funded clinical research unit providing CRO-grade services. We are, in fact, also registering with in the process of registering with CDSO to become a CRO, and distinguished by its unique structural model and comprehensive service portfolio. This spans the full clinical trial life cycle. Its integration within the Medical Research Center gives our platforms a critical operational advantage. It ensures that our studies will not only be scientifically rigorous, but also compliant, timely, and audit ready. With secure physical and digital infrastructure, including validated archival systems and a regulatory grade biorepository, we bring the backbone of clinical excellence to every MRC initiative. This new Center on Advanced Imaging through AI and Data Science aligns closely with MRC's mission to rapidly translate scientific discoveries into practical healthcare solutions. Leveraging our proven expertise in medical imaging and artificial intelligence, we aim to take our work to the next level. The center's activities span the entire innovation cycle, from data acquisition and annotation to tool development and validation, clinical deployment and efficacy, and then industry collaboration and prototyping. By partnering with us, industry collaborators will gain access to our expertise, extensive research infrastructure, multidisciplinary expertise, and a platform to validate and enhance their products. We are eager to co-create and co-develop solutions that improve healthcare accessibility and outcomes globally. So the Medical Research Center is more than a facility. It's a catalyst for innovation, a launchpad for collaboration, and a bridge from science to solutions. We believe the MRC will define clinical innovation in India. Let's build it together for scale, for impact, and for India. We invite your ideas, investments, and partnerships. Thank you for your attention.
Thank you very much for the introduction. Joining us now are Dr. Amit Awasthi, Senior Professor, and Dr. Dinesh Mahajan, Associate Professor, to give us the introduction on Translational Research Facility, TRF, which is our another new initiative which will enhance our capacity for cutting-edge translational science. They will outline how TRF is poised to support the Synchn's mission and also to enhance more meaningful collaborations between the academia and industry. Please, sir.
Thank you. Thank you very much. So I will be briefly talking about the new facility which is coming up, TRF. Together with my colleague, although I requested him to speak, but he decided not to speak. He said, I will stand by you wherever you need me. I will just do the job. So thank you. So this is a translational research facility. This is MRC already completed, and I hope hopefully this will be completing in under 2 months, right? Or maybe lesser actually. So be optimistic. So when I was making these slides and discussing with my colleague, I was not really sure what this pillar in THSTI or in this National Science Biocluster would look like and what it will be actually doing it, right? So we decided and discussed this will be an accelerating journey from innovation to the biomanufacturing, and this is in line with DBT Bio E3 policy and the biomanufacturing. So this center or this TRF will be really enabling from discovery, which is happening in the THSTI or maybe in other big institution, will be coming to TRF, and TRF will be a kind of a place where we can take these things forward from bench to bedside. So as I speak, this building is actually more than this because this picture is slightly older, like a couple of months ago. And you can see that we actually discussing and kind of decided that how each floor of the TRF will look like. And you can clearly see on the thing, we have different floors decided for therapeutics, biologics, facilities, diagnostics, live biotherapeutics. And we obviously will be having some other wings like, Center for Excellence and, instrumentation, etc., etc. The good part is the facility is really large. You can see that we have 5 plus 1, total 6 floor, and the area is each floor is around 18,000 square feet. And this could be perhaps the first model in the industry academia partnership, and this can really take this model forward in the National Science Bioscience Cluster as well as other possibly biotech parks. So broadly, how it will look like, I mean, the building is okay, but what we will be having in the building, right? So we were discussing and decided that, we will be having different things like pandemic preparedness, the biomanufacturing, and the Center for Excellence. So I'll actually walk you through one by one how we are envisioning this for the future for industry and academia partnership. So in the biomanufacturing, as you know, that's like India is really poised to, take, discovery to the next level. Biomanufacturing, and you can clearly see, like, on the one hand you have the THSTI building where the research innovation is happening. On the other side we have the TRF. So how we take this innovation on the research discovery, what THSTI scientists are largely doing, and maybe we can bring this or extend it to the BRIC institution as well to the next level. So therefore we have this TRF, and the key area in the TRF which we have identified to begin with will be like in vitro diagnostic manufacturing support facility, pilot scale GMP for vaccine and biologic innovation, and biomanufacturing for biotherapeutics, particularly at this stage is the monoclonal antibodies and live biotherapeutic, which is briefly you have heard about Dr. Jayanta and Supratik. So on the line of biomanufacturing, THSTI has received one of the first biofoundry grant on the two of these, two of these, like one is the monoclonal antibody and one is the live biotherapeutics. And this facility will be built, is coming up in the TRF. The space is already identified, and this will be like in collaboration of industry. We will be taking some of the live biotherapeutics as well as monoclonal antibody, which are showing really good potency in, like, treating or neutralizing virus or maybe other diseases. The idea is to set up the platform. So it's not about, the disease agnostic. It could be like you can make antibodies for anything else, right? So it's a platform. You can have live biotherapeutic lactobacillus, or you can have bifidobacterium or many other kind of other agent which can be made in this facility. So one of the things which I think when we actually do research on the designing novel antigens and vaccine, one of the things when we try to take it to the next level, it is always kind of a hurdle that, industry, when we talk, the industry asks so many things, different kind of regulatory documents and how you have done and various all these things. I think so removing this bottleneck, we already have one small facility called BDDC. But now we are extending this in the TRF, that we will having a small GMP manufacturing facility in the TRF, so that whatever we have actually shown at the concept level can also be done in the GMP setup, and that can be done in the preclinical setting, and then industry will be more interested to take forward. And we are open to have industry to have partner with us like Panacea from the very beginning, from the time of conceptualization. So we are kind of open in both ways. Industry can come at a later point, or industry can come from the early time point doing this with us. So the key feature of this, we will be having a clean room facility with, modular pilot production zone, equipment, fermentation, and various other, formulation unit. We will be having our in-house QC, QA, identity potency, and the endotoxin testing and various regulatory, which is required for the regulatory parameters. And the GMP grade supply essential reagent, including enzymes and the polymerase, various things which require for the research from making a product from the bench. right? And obviously, everything will be done in the regulatory compliance with the CDSO, keeping them on the board from the very beginning. So everything will be like properly done in a regulatory fashion. The second part of this in vitro diagnostic manufacturing support facility, which will be plug and play facility with ISO 13485 compliant manufacturing space and regulator ready IVD batches. So this is something really very important. I think this is what we are trying to do here is the one of its kind, and maybe it can be extended to other facility across India in the government setup. So this is like act as a pandemic preparedness also, because we are living in the world where emerging and the re-emerging pathogens. Every time there's a threat from the new virus is coming, or the COVID-19 pandemic is still kind of new viruses are emerging. So this is kind of also help in the pandemic preparedness setting. The facility, this particularly the IVD facility, will be 8,000 square feet and will house various core zones. We have a core zones, the key equipment, the service offered, and the roadmap. So everything we can actually discuss in the detail. If some of you like to discuss, Dr. Gaurav Batra is here, who actually taking this forward. So anybody who is interested to work with us, Dr. Gaurav Batra is perhaps the contact person for this. So what we are doing different, right? We are different because we have our focus small batch, cost effective manufacturing for academic and SME needs. And we are like built for rapid translation from lab scale to phase 1 and phase 2 trial. Because now Dr. Nitya has indicated like we have a kind of infrastructure, the MRC, where the early phase 1 clinical trial can be done. So something what you can do here can easily go to the MRC and in CDSA, and then we can really work. It's kind of like you come to us and it's like everything you get in THSTI. And then we have like integration of translation R&D, adjuvant, and virology analysis, where we have various virology platforms. We can really have virology or viral banks. So various things are under the same roof. So this is what makes us unique actually. And we have support for live inactivated viral agents, BSL-2 compliant for vaccine and challenge study, in-house technical consulting on like TPP, SOPs, and regulatory strategies. So this is what makes us unique in the TRF and actually overall kind of environment. So pandemic preparedness is always a kind of gonna be, one of the things which I think all of us actually look forward, because we don't know, the virus keep coming and going, and it's kind of the old virus can take the new shape and our immune system is not able to recognize it. So therefore we have to be always in a kind of readiness mode. So I just want to what are the pandemic preparedness, because this is bioassay laboratory, which we started before COVID-19 pandemic in a very small area in the THSTI main building. And this facility really served nationally and internationally. I think internationally more clients are coming to work with us because we have these assay which is validated and certified, NABL accredited. And we have done a lot of work during COVID-19 pandemic, and we are still continuing doing work on other vaccines and other platform. So we decided that this small facility, which really shown so much promise to THSTI, should also be extended in a larger space in the TRF, because these assays will be utilized by any company which is coming to TRF. And this kind of, they can go to them and they say, okay, we need to do this. So this is something we are also expanding this. Similarly, immunology core lab, immunology is one of the key component of anything what we do, whether it's a drug discovery, whether it's a vaccine, you name anything, even the neurodegeneration, immunology is coming big way, right? So therefore, what we could actually standardize in the immunology core, we have various high-end assays what we standardize, and these are all cellular assays. Right. So in the bioassay, those are the serum-based assay. Here we have the cellular assay. So we have a kind of balanced both cellular assay as well as the antibody-mediated assay. So this facility, what we established in the THS in a small area, maybe 500 square feet, actually served largely in the COVID-19 pandemic, and every cellular assay or T-cell response, what we actually see in CDSO was done in THSTI for the vaccine, every vaccine what we got approval in the country. So now, since we showed a lot of promise in this facility, so therefore this facility can also be go to TRF and can serve not only for vaccine, but for the drug, because, you know, in the vaccine, you want the immunogenicity which is required, but in the drug, you want immunogenicity which is not required. So this is immunogenicity is the key component here, which this lab can serve actually. And obviously, when we did a lot of work on the COVID-19 pandemic and other, you know, epidemiological studies, our center, THSTI, got recognized as a global center of WHO Global Coronavirus Network. So some of these, you know, facilities will also be housed in the TRF as a part of the pandemic preparedness. Now, when we are actually making this large infrastructure, and obviously this infrastructure is not going to be empty wall, we have to fill them with a lot of things, right? Very high-end equipments and various things. So therefore, we also envisioning that maybe the center of excellence can be created in this. So for example, if some industry partners are partnering with us and say, okay, we're going to have some unit in the TRF with the high-end equipment, and this facility and these equipments can be utilized by SMEs, which we are envisioning to come to work with us, as well as the THSTI innovators or the faculties to utilize this facility. So it's kind of win-win situation we are creating. We are bringing industry, we are bringing center of excellence, and we are having discovery pipeline. So this will actually will be an umbrella which will be utilized in the TRF. And in addition to, you know, the center of excellence, we obviously is important to have this multi-omics facility, which is right now running a smaller scale in the THSTI, and this is important for various discoveries, particularly biomarker discoveries. So these metabolomics, genomics, and proteomics platform will also be having a kind of a larger space in the TRF, and that can be utilized, and having THSTI as expertise, because we have expertise in these facility. So these expertise can be utilized by the SME and our industry partner when they actually come to TRF. So I already mentioned about why we are unique, the USPs, and some of these USPs are actually enlisted here. I will not really like to read. I think if you have any question about TRF, I will be happy to answer. My colleague Dinesh and other faculty is able to answer, and obviously Dr. Karthikeyan, who is here, able to also answer during the lunchtime or any other time if you like comfortable. So with this, thank you very, very much, and I will be keep seeing you and keep interacting with you. Thanks.
Thank you very much, sir, for the introduction. We now welcome Dr. Amit Kumar Pandey, Associate Professor, and Dr. Pallavi Kshetrapal, Associate Professor. We now welcome Dr. Amit Kumar Pandey, Associate Professor, and Dr. Pallavi Kshetrapal, Associate Professor, to share insights into the facilities available at BRIT THSTI and how they are tailored for research and translational partnerships.
Good morning, all. My name is Amit Kumar Pandey. I'm an associate professor here. So in this presentation, me, along with my colleague, Dr. Pallavi, will take you through the facilities that we have built over the years and how this facility is augmenting the overall progress of this cluster. So with the vision given by our Honorable Prime Minister of Atmanirbhar Bharat, in partnership with DBT's Bio E3 policy, we have designed these facilities to accelerate the development and commercialization of bio-based products. So to begin with, these are the facilities that we are going through one by one, and let you know the missions and the accomplishment and where we are with respect to these facilities and how these facilities are, is moving like a backbone for augmenting the work that we are doing. So I'm not going to talk in detail about Panacea. Dr. Avasthi discussed briefly about this. So this facility was, as part of the SAPI centralized laboratory network. The mandate is to design novel bioassays, and these bioassays, they support the development of vaccine. So it played a very critical role for the development of COVID-19 vaccine. And post-pandemic, it has evolved and it has increased the repertoire of bioassays, and now it is currently supporting the development of new vaccines against emerging viral pathogens. So we have one of the largest animal facility at our at THSTI, and this facility is equipped with high-end instrumentation and trained manpower. Over the years, we've developed some animal model platforms, and these platforms are very helpful in performing and supporting preclinical studies. And we are giving support to both academia and industry partners. And hopefully, if anyone in the audience is interested to use one of these platforms, we are happy to cooperate and help. So over the years, we have added the histopathology and the germline preservation laboratory into this. Currently, we are in the process of getting it accredited by both ABAC and GLP requirements. So, coming to so I'm very happy to announce that CEPI has recognized this facility as one of their preclinical study sites under the CEPI Animal Model Network programs. We are very happy to announce that we just completed our first study on ferrets, and this facility is very good. We just, in collaboration with Preface and ISC, completed this study on subunit vaccine for influenza. And as you all know, this is a very apt and very unique model for studying pulmonary infections or pathogens that infect respiratory systems. And we are again looking forward to academic or industrial partnership. So please contact us if you are interested to use this ferret model. As you all know, we are trying to build up a one-shop solution. So we have RCGM approved animal BSL-3 facilities. As you all know, this facility is involved wherever the study is on BSL-3 agents, namely SARS-CoV-2, Mpox, tuberculosis. So again, this is a facility that is is catering and supporting industry and academia in clinical, preclinical serosurveillance studies, wherever the pathogen involved is a select agent and categorized as BSL-3. Again, the immunology core, I'm not going to talk in detail. Dr. Avasthi alluded to this. This mandate of this is to develop, optimize, and validate specialized immunological assays. They have, their expertise in facts-based deep phenotyping studies. Functional cellular assays have been actively used by both academic and industrial partners. So, so the BIRAC through National Biopharma Mission has declared this site as National Immunogenicity Testing Platform. So anyone who is interested to test the immunogenicity of the of the product can reach to us, and we will find the solution for them. Lastly, I mean, we are trying to bridge the gap between academia and industry. For that, we have THSTI through VDDC is facilitating scaling up of product at the pre-GMP levels. So we have already scaled up a couple of subunit vaccines that are currently being used for testing. And any industry partner who wants to use this facility is most welcome. And this is something that provides you a space, ISO 7 level clean room with process development unit, wherein you can scale up your lab subunit vaccine to at least 5 liter scale, which we have done, which have been doing in the VDDC. So I invite my colleague, Dr. Pallavi, to please go through the other facilities that we have at our place. Thank you.
Thank you, Amit. So there is an arsenal of facilities at THST, and that's why it takes two to get the other facilities going. So I'm going to introduce you to the unique facility, which is the genetically defined human-associated microbiota. So there are around more than 6,500 microbial isolates that are there in this facility that are from all over India. And out of them, there are more than 2,500 that have been genetically sequenced. And these sequences are available on the public database at IBDC and NCBI. The unique thing about this facility is that this can be used, these genetically defined microbes can be used for nutrapharmaceuticals. And we had just heard a success story in the morning today with Dr. Bhaptosh and Dr. Varun. Coming to the next facility, is the multi-omics facility, which Amit had briefly talked about. And this is a very unique facility which is based on high throughput platforms that are used for not only genome sequencing, but also for untargeted and targeted protein and metabolomic identification. And the unique thing about in metabolomics is that there are more than 9,000 pure compounds which are structurally derived for the metabolite identification. Coming to data management and data science center. So this is a very unique portfolio where it has 14 ongoing and 10 completed projects. And the interesting thing is more about automation in this facility. So there are automated tools that have been made for checking the quality assurance and the quality control, which have led to real-time research tools, which is one of the major things about the Garbhini Drishti, which is a dashboard, which is a national repository for clinical ultrasound and biospecimen collection. Looking into this, the data sciences and using all the tools that have been generated in this facility, there are new AI-generated imaging tools that have been developed, which are going for validation. One of them is the AI model to support triaging for high-risk pregnancies using ultrasound images. And last but not the least is the biorepository facility, which is an international recognized facility of ISO 20387. Which is one of its kind in the DBT cohorts, and it archives more than 1.6 million biospecimen, has a robust infrastructure for SOPs and management system protocols, which is accessible to all the researchers in the academia industry. And during the COVID times, which was briefly mentioned today with Premas also, there were some samples which were shared with them. And this sharing is done through a biosource governance, which is a robust governance policy we have, and which has DBT nominated access control committees also. So using the state-of-art infrastructure, we provide services for biospecimen archival and database hosting, the biospecimen processing facility, and also have workshops for the best practices that can be for biobanking in India. I would like to thank everybody here who are the coordinators and the faculty in charge of all these facilities, and also for the content guidance we have from the past and present executive directors here, and also the generous funding which Dr. Karthikeyan had just mentioned, that it is mostly the government funding that we have, and we would like to invite you all to collaborate with us for use of these facilities for national good. Thank you.
Thank you, ma'am. This presentation truly reflected our research ecosystem and the collaborative opportunities it can offer to all our industry partners. Now, it's my pleasure to invite Dr. Manish Diwan, Head Biofoundry, Biotech Science Cluster from Biotechnology Industry Research and Assistance Council, BIRAC, to introduce BioE3 Innovation Park and speak about its framework, functional objectives, and the broader significance within the biotech ecosystem. Over to you, sir.
Good afternoon, ladies and gentlemen. Great to see everybody here. I think all this we are hearing high intensity pitch from the research institution and the ecosystem that is getting developed. So we don't get to hear something similar from government institution and in the academic sector, but I think all the stakeholders are very, very excited to see and take it to the next level. The NCR cluster would eventually evolve into a first-in-class mega science cluster, which is planned as triple helix model. So this is 200-acre campus. I'll just take you through in a couple of slides what the vision for the cluster is under the leadership of Dr. Rajesh Gokhale, Secretary DBT. So typically, if you see, the academic research institutions would have
fresh talent, a research laboratory, knowledge to create new science, and they would have activity at laboratory scale level. If you look at the bottom side of this, yeah, yeah, bottom side of this, and if you compare the large industries, special economic zones, in or manufacturing units, they would be equipped with high-end infrastructure for product development, product investment funding, as well as product development expertise also. The two ecosystems, they exist, and they are largely okay if you are not doing deep tech innovations, proprietary knowledge, new knowledge generation, and new product development. But I think as we move forward, we need to step in to create proprietary innovation from India so that we see new bio-based products getting innovated and products technologies reaching to the market. So the Bio E3 brick innovation parks would actually bridge this gap and empower the academia with pilot level, validation level infrastructure so that they can produce industry ready output, and that can talk same language and assets can be moved smoothly from one end to the other side. So we made a humble beginning with Synchn 2024 and now this edition of 2025 to expose what deep tech science which is happening in the cluster in the THSTI. So we have showcased you some R&D projects where some of the collaborations are already running and existing. We're also creating an awareness in the community that this is what is brewing in the ecosystem. And as we move forward, some of these initial engagements start showing successful outcomes. So we'll see that the trust building and confidence building of the ecosystem. Some additions in the infrastructure and capacity announcements as we just saw, technology, translational research facility, medical research center. So these are infusions to create pilot level and validation level facilities. Now we step into expansion and building this 200-acre campus as we move along. So this is a spread of the 200-acre campus. On the bottom side where we are looking at, this is the R&D core that is likely to power up the entire 200-acre cluster. So some part of it is already built up, about 50 acres or so. Some of these facilities like TRF, MRC, they are also in this yellow circle. There is a provision for startups and SMEs to come seed with their independent R&D units as well as access those pilot level and validation level advanced infrastructure. On the far end, you can see that is the large industry core where they can come in to set up R&D innovation hubs and pre-product development centers. We also have provisions for other stakeholders like GCCs, service providers, national and international stakeholders to come and reside, co-localize into the ecosystem. There is a provision for societal connect with science so that people can realize what bio-based innovation, biomanufacturing is all about and how it can impact our future. So in the cluster, it is envisaged that we will be looking at 5,000 to 7,000 plus people working simultaneously in this 200-acre campus, and that would have many of you representing here. There would be new job creation, but that would happen with a lot of extramural and intramural funding in the public-private partnership mode. One of the elements I think we saw, some industries are already participating with us, but by and large, these suites will be built up with in PPP mode where more than one company can come forward and join hands, bring in top of the line expertise or equipments and utilize such facilities, and many such companies can coexist, and all the services, all the scale-up work, which is like pilot level and validation level, which is churning out from the R&D institutions, can be offered as professional services to whosoever comes. And the outcome can obviously be shared and reinfused into the ecosystem. As we progress over a period of time, I think today we are looking at more of in-house R&D pipeline that is getting exposed. This place would also get powered up by the pan-India BRIC institutions R&D pipeline, plus the startups, SMEs, and industry which will come in. And slowly we'll move towards new product bioinnovation and biomanufacturing, which is of globally relevant and scalable over a period of time. I think I also acknowledge the presence of investors in the forum. So this would also be a place where you could spot the deep tech innovations to support and come up with Series A and above funding. So I'll rest here. Thank you.
Thank you so much for presenting the promising initiatives that will enhance our understanding and capabilities in translational research. We now transition to a panel discussion featuring eminent leaders from industry and science who bring unique insights into the evolving landscape of translational research. I would like to introduce our panelists, Mr. Akhil Ravi, Chief Executive Officer from Origin Pharmaceutical Services Limited, Dr. Anil Kaul, Adjunct Professor, London School of Hygiene and Tropical Medicine, Mr. Anshul Jain, Senior General Manager from Mel Diagnostics, Dr. Jaydeep Kotte, Group Chief Medical Officer from Cipla, Dr. Mahesh Bhalgat, CEO and Managing Director from Vida Life Sciences, Dr. Priya Hingorani, Managing Director and Vice President of India and Southeast Asia Unit of Miltenyi Biotech, and Dr. Sachin Kale, Head, Manufacturing Science and Technology from Serum Institute of India. To moderate this session, let's welcome Professor Pramod Garg from AIIMS, Delhi. Professor Garg is a renowned physician scientist, president of AIIMS, Raebareli, Bilaspur, and Avantipura, and the former executive director of our institute, THSTI. Currently serves as professor and head of gastroenterology and nutrition at AIIMS, New Delhi, a fellow of 4 major science academies, including the Indian National Science Academy and Royal College of Physicians. He is especially known for his pioneering work in the field of pancreatology. With immense pleasure, I would like to invite all our panelists and moderator to the stage. Over to you, sir.
A very good morning to all of you, and I'd like to thank Karthikeyan for inviting me here. It's always such a pleasure to be back here at THSTI, and I'm very happy to see hall full this morning and all the distinguished panelists here with me to talk about something which is very important, not only from a point of view of an academic institution, but also from the point of view of healthcare industry in general and India's economy in particular. And healthcare is something that is recession-proof, war-proof, pandemic. It flourishes in pandemic. So that's why this industry is at the core of India's development and economy as present. And with me, I have many distinguished panelists from industry and also Dr. Kaul, who is both in industry and academia. So very happy to have them here. So I'll just request each one of them to give you their background or their company's background in 30 seconds. We have about 40 minutes, 7 people, so you'll have about 6 minutes each, right? Maybe we can start from there. Just give us your background in 30 seconds and then pass on the questions.
Anjul, yeah. I'm Anjul. I'm representing Diagnostic Private Limited. We are the India's largest manufacturing when it comes to the medical devices company. We are based in Gujarat and Vapi, and serving to the global platform of around 200 countries worldwide. And we are proud to say that we are completely Make in India company under the PLA scheme of Government of India. Thank you.
Hello, everyone. My name is Sachin Kale. I head the Manufacturing Sciences and Technologies function at Serum Institute of India. Serum Institute of India is India's largest biotechnology company, and it's also largest vaccine company in the world by number of doses produced. In this role, I am responsible for supporting the existing project as well as onboarding the new project. And that's where I come across many of the academia industrial partnerships, and I get chance to interface with them.
Hello, everyone. This is Jaideep Gopte. I'm the Global Chief Medical Officer at Cipla. Cipla is among the top 3 companies in India with a turnover of about $3 billion. We are present in over 100 countries with about 47 manufacturing plants and have products across a range of therapeutic areas. Particularly, I would say in pulmonary medicine, we're probably among the top 2 or 3 manufacturers of inhaler devices in the world. And that's a big area of interest for Cipla, including more recently infectious disease. Thank you.
Thank you, Shari. A very good morning to everyone, and thank you for your patience on a full hall. I don't think I need to speak much about the company because my colleague in the morning spoke about it, but I'm Dr. Priya Hingorani. I'm the Managing Director for India and Southeast Asia for Miltenyi Biotech, which is the pioneering company for cell and gene therapy. And I'm so glad that many on this panel are either associated or are going to be future associated with us as we start working in this space. So thank you very much.
Good morning. Good morning. My name is.
Good morning. My name is Anil. Thanks, Dr. Garg, for getting us all here together. So I have been Vice President for Johnson & Johnson R&D for almost 20 years. Currently, I'm Professor of Translational Medicine at London School of Hygiene and Tropical Medicine. I have been very much associated with CSIR Imtech previously, and I was also on board of directors for CSIR from 2019 to 2021. So yeah.
Hi, morning everyone. My name is Akhil Ravi. I am the CEO of Origin Services. Origin is a CRO and a CDMO company, and it's a wholly owned subsidiary of Dr. Reddy's. We've been working with a number of global institutions to launch novel medicines and also discover novel medicines. And proud to be also in partnership with THSTI on one of the pioneering work they are doing. Thank you.
So thank you very much. THSTI already has many ongoing collaboration between academia and industry. In fact, one of the best examples is with Panacea. I saw Dr. Khalid is here and Dr. Jain was also here some time back. So we had a very big grant from CEPI and we are developing hopefully one of its kind first vaccine in the world. And that collaboration has taught us how to engage with industry. What are the steps? How do you move forward? What are the challenges that you face? And we think that a similar thing can be replicated in many other areas. This happened because of the pandemic, I tell you. THSTI was involved in vaccine development even earlier, but during the pandemic, there was a huge demand and rush, and also everybody actually chipped in. So we think that today in India, the overall ecosystem and environment is ripe for taking this kind of collaboration forward and come out with new things. So maybe I can start with Priya here with you, because CAR T-cell therapy is the one which is currently everybody is talking about and hugely successful worldwide. So you tell us about your collaboration that's starting now, and how do you see it moving forward? And what do you expect from THSTI? Because currently, as you have just heard in the previous session, we have all the facilities here, including in the coming times to start clinical trials for vaccines and for other things also. Over to you.
Thank you so much for that question. Always difficult to go first to the panel of such distinguished guests, but just to give a brief on what's happening with THSTI. First, a quick thing on what we do globally. So if you look at Miltenyi Biotech, globally, we are associated with academic institutions and universities for either research or upskilling or bringing cell and gene therapy to the world. And like our esteemed moderator just expressed, cell and gene is the new space, right? It's a new kid in the block. Everybody's interested about it. But it's also, I think, it's about the potential this brings to the world. I was just talking to 5 minutes ago in terms of what it can do to a patient, what it can do mean to change lives. And in autoimmune disorders, I know Dr. Amit is extremely interested in pursuing that in addition to oncology. We have some results. I often refer to it like a dermatological ad for before and after, in really making impact on life so well, which is between 16 to 25 years of age. So it's all about creating patient impact. It's all about making sure that there is enough that can be done. But here I'm talking to this august audience of researchers. So I think let's focus a bit on what Miltenyi can do for research. According to me, research is what everybody sees, what all of us see, but only you get to think about what we cannot see, right? So if that is the case, we, along with THSTI, are partnering in upskilling many of our young scientists towards cell and gene therapy. We started that with a fantastic LOI, and I can see Dr. Jitendra Kumar right there from Iraq. We got this encouragement to start on an LOI for working with the government and building up something in biotechnology. I think a great work there. Then with Dr. Karthikeyan and Dr. Amit to make sure that we are able to bring in the latest in the field of cell and gene therapy and train our own Indian scientists on this. This is fantastic, not only from a perspective of being in the industry and seeing a lot of researchers, but I also believe it helps us bring cutting edge therapy to the market. What it will do, besides education, is also help us develop. When we saw the MRC facility or the TRF that Dr. Amit showed us, is work towards making it possible to bring Indian developed CAR-Ts to the market. Right? We already are working with CMC alone, where we have Velcart with astounding remission rates. And it's so, I think it's a moment of pride when you see those patients who have no, but getting into something that can give them a future in life. So here we are working on education, we are working on upskilling, we are working on providing infrastructure. And for anyone who's interested, we have projects which are established here. This is a world-class machine, probably one of its kind, true to its name. I think it's called Prodigy for very good reason. And that's an end-to-end cell manufacturing unit. So you have the leukophilized cell on one side and you get the CAR-T on the other. So plus the max quant, which is for making sure that you're making the right thing. So from that perspective, I think we are very well placed to partner with THSTI. This is the beginning. We intend to bring in more. We intend to work more. We intend to make sure that if the manufacturing needs to happen and to get the vision of THSTI and Biomaterials live, we are fully committed to doing it, whether it's point of care, whether it's making sure that there's new indications that are coming in, whether it's autoimmune diseases, whether it's neurological disorders, or many, many more. I think that's where we stand with our project today.
Although the CAR-T cell market is still in the nation stages in India, but getting crowded. Any challenges from that point of view? And also regulatory aspects?
So I would like to be a little naive there and say no. Because, well, from a competitive point, yes, the more it is, the merrier. Because like you rightly said, it's in the nascent stage. The exact noise level that is required will probably help everyone get there. But I can also proudly say that our platforms are available to many, like I said, many on this panel, either today or future, are working on our platforms. So the idea is to make sure, and doing so it's the same. From a challenge, I don't think it's a challenge. I think it's about the thinking, right? It's about the policy, it is about the people, and it is about the product that is coming there. So the triple helix that got referred to, I think, just a few minutes ago, is something that is going to make it come true. And it's true as much for us as much for anybody else.
Thank you very much. So let's change gears. So tuberculosis, of course, remains one of the major challenges India and globally. And I can tell you that THSTI is at the stage where they can actually come out with a new molecule. I think the last big successful molecule has been Bedaquiline. Call has been associated with that, and a supply company which is into multiple pharmaceutical medications, including HIV and tuberculosis. So an institution like THSTI is always looking forward to industry. Say they have come up with 4 or 5 molecules. Can you guide us? How do you take this molecule to its logical conclusion of reaching the clinics, Dr. Jaydeep from Cipla? And how can you help and collaborate with us?
I think it's a great thing that THSTI is coming up with actually new molecules. There's a lot of innovation from India coming in the vaccine space, but I think the real high-end will be very able to have new molecules. We've barely had a couple of them from India. But I think the first and most important thing for any molecule to really make it is to make sure we have very strong intellectual property around it. We're in the world of IP, and strengthening our IP regulations, in addition to that, having a very strong patent attorneys that you need will really is one of the things that the first thing the industry will ask if it's coming for licensing deal or for a co-development deal is how strong is it. So I think that's the first thing I would recommend. Second thing, I think THSTI is great to see building all the facilities for taking a product right from the preclinical, in vitro, perhaps even now the phase 1 clinical stage. Great to see this whole development with a pilot GMP plant, etc. And I think that's really what is the minimum requirement. I think having expertise is very, very important in this area. And one area that I see that perhaps we need to do a lot more from India's point of view is a deeper understanding of PK/PD of products as they're being developed. Earlier, we would more focus on PK/PD related to antibiotics, but now I see even biologics, monoclonal antibodies, people talking about the PK/PD of things. And I think that's an area where perhaps as a country, we need to understand more, because when we try to speak to people, we find that the expertise is somewhat limited, and they often refer us to external experts, which is perfectly fine when we go to work with everybody. It will be great to have inbuilt expertise, and maybe THSTI is something that can be developed. So once you understand the PK/PD well, believe me, the translation from preclinical studies to human studies is, I won't say easier, but it's less challenging. And then, of course, running through the clinical development center, and then we can really chat about that more. But I think that would be really the foundation to just lay this out.
In addition to PK/PD, one of the challenges that I found when I talked to Ramandeep and Dinesh and Nishit was that a small animal toxicity they can still do, but big animal toxicity is something that remains a challenge because it's also very expensive. And they are often, searching here and there for some money here and collaboration, but difficult to come by.
So I know, we've been working with somebody outside, and doing non-human primate studies can be quite challenging here. Maybe this is something to think about as well, because at the same time, we also want to use animal use. There's a strong move also from the CDSCO and from global regulators to try and minimize. In fact, we just started a small consortium within some of the big pharma in India as to how to reduce animal use and perhaps replace it by in vitro, strong in vitro testing or assays. But I think that's something we have to really figure out. I'm great to see the controlled human infection model coming up. I saw this about 3 or 4 years ago for the first time, but great to see that it has been coming up in India as well.
Dr. Kaul, your thoughts about it, your guidance of taking a molecule from the lab to the clinic, particularly tuberculosis.
I think any molecule, it's not only TB, any molecule, whether it's in the oncology space or it's in the neuroscience space or in the cardiovascular space, any molecule which is not what Dr. Karthikeyan said in the morning, me too. I think we in India are diseased with this phenomenon of me too. We are followers, we are not leaders, let's be honest. So we need to change that mindset in terms of the innovation, that if you have something novel, something very good, anybody else in the world will come and take it. It has nothing to do with the logistics or the operations of it, but I think it's the innovation. I think what we need to drive as a country, let's take the example of cell and gene therapy. Leave aside the TB as such. The cell and gene therapy, if you look, the global market by 2030 is supposed to be $35 billion. And this year, in 2025, 1/3 of merger and acquisitions happened in China. 1/3 of global merger and acquisitions that happen in China, and half of the global cell and gene trials are done in China. So there must be something good happening in terms of the R&D ecosystem. In this very novel nascent space, where the expertise was not lying there in any of the developing countries, which was built up and which was novel, which was innovative, that rest of the world is going there and doing all these licensing and acquisition deals. So that's the thing I think I really resonate with Professor what Karthikeyan said in the morning is the idea of me too. We have to be more innovative, and rest in terms of the regulatory, in terms of the talks, in terms of the pharmacology, and in terms of the industry coming forward will always find, because there is where the innovation lies, and there is what will eventually make the difference in patients' life.
So I think you have raised two. Yeah, please go ahead. Yeah.
Anil and I were a part of that organization. So way back in China in 2013, when nobody was talking of cell and gene, they were talking of cell and gene, which was practical and done. So I just want to second that and add with that data point.
And to be honest, I'm happily surprised by what THSTI announced this morning today in terms of setting up this the center, which is basically a kind of a skill development center as a training center, which will train people in this particular lose a race in cell and gene therapy once again. So we want to be the frontliners, and this is where this institution has done phenomenally good as compared to the rest of the institutions in India in going look in building a futuristic view on a technology platform which will deliver to the patients of India. And we know how cancer is rising in this country.
Right. So with regard to the tuberculosis molecule that we're discussing, you have raised 2 points. One is the Me Too issue, and the other is the China's example. Now, in terms of new molecules I'm talking about, the problems, the practical problem that you face, you have a molecule, you have shown very good results in the lab, you are very happy about it. Next step is tox studies, and then you go to phase 1, right? Big animal. Here is the gap. The government says, no, you haven't done this, I can't give you this much. The industry says you haven't had the patent, you haven't done the PKPD, you haven't done the animal tox studies, so I can't come on board. There is a risk aversion from the industry as well, because I personally was involved in some of the discussions with a couple of industries for tuberculosis molecules. They always held back. So you are saying everybody will come in, but they want a certain stage. Certain stage is phase 1. Government says, no, you have not reached that stage, so we can't give you more money. China, what's happening in China is that they invest as a government, and I'm happy that biotechnology and other people are also here from the government side. How do you bridge that gap from the lab to the industry where more funds are required? Some push is required.
No, absolutely. I think the key here is what I see in terms of the innovation. I think the key, I always see that the Indian industry, let's be honest about it, the Indian industry, the generic industry has been very much not focused on really building up the innovative medicine pipeline. But these things are changing now. We have seen the moment of change, because the Indian generic industry also understands that the genericization is going to be very challenging in the future, because there are different players coming up and the markets will be defined by the price. So if you have to compete on the price, it's going to be always difficult. Now people are really understanding that you need to be more innovative. So there is more money coming from the Indian industry into the innovation part. That's a big change. And I feel that in the next 5 to 10 years, there will be a marketable change in the Indian R&D ecosystem, driven by not by the international industry, driven by not by the MNCs, but the Indian pharma industry, which is quite big in terms of the scale and operations. If they go into the innovative medicine aspect, we have Reddy's here, they are already in oncology, we have Sun Pharma, we have so many other industry, Cipla is also there, so many big, huge industry pharma which have muscle power, which have money, which have finances, which have operations, they can change the Indian R&D ecosystem very much.
But they need to invest. They're very risk averse.
I think that's what is changing right now. If you look at the dynamics inside these organizations, they also know that their models are very shaky.
Right.
Anybody can penetrate any Indian pharmaceutical just based on the prices and the competition that comes through.
One industry which has really done well is the vaccine industry, and let me come to the serum in that space. So you are perhaps the largest vaccine manufacturers in the world, and this is an area where THSTI is also doing very well. So what are your suggestions and guidance to the institution like THSTI? And THSTI is taking an example. There are so many other institutions in the country which are doing this kind of work. How do they partner with you to take it actually to the clinic? And we don't have a molecule or a vaccine or an antigen coming from London or from NIH. You have to take the antigen and a vaccine, the candidate from here.
Yeah, thank you very much. So we welcome the antigen coming from India. We don't have to pay high IP fees that we have to pay to in-license antigen that we were supposed to do till now from outside. We are working with THSTI. We are in discussions, or we continue to have discussion with THSTI on various antigens that we want to pursue. There was also a talk about Me Too molecule. As a Serum Institute, we are fine with Me Too molecule as long as it addresses the issue of access. Because even if it is a Me Too molecule and it improves the access in India, we are fine with it. We are fine with collaborating with THSTI on those molecules as well. Not only on the new antigens. We welcome the new antigens that give India and us the edge. On the question of how we would collaborate, the question of risk, industry being risk averse was also addressed. Nowadays, post-COVID, we are much less risk prone to take more risk as long as it justifies. We have come down our ladder of proof of concept. Now we don't take that have the candidates that have demonstrated phase 1 or higher proof of concept. We also try to invest in the candidates that have potential to show promise and that have potential to address outstanding issues in low and middle income countries, and that are also synergistic with our portfolio. So the advice to THSTI and advice to industry such as Serum is to keep talking and keep coming up with the new opportunities in this field so that we could do a co-development of that molecule, share the risk as we go along.
And what do you advise them so that your confidence actually increases? No, this is the institution I want to go to. They will give me a good molecule or a good candidate vaccine or whatever. What is it they must be doing more to increase your confidence so that you are ready to invest?
Well, initially it was phase 1 or phase 2 proof of concept. Nowadays, I think if they have demonstrated a proof of concept even in a preclinical studies that are compliant with the correlates of protection or the immunogenicity profile that have been shown in academic labs, that would give us a fair degree of confidence in proceeding with that candidate.
So what kind of facility do you expect them to have more, other than what they have already shown you?
Well, I'm glad that they have lots of facilities in bioanalytical space. We as a GMP manufacturing institute do not have flexibility of high-end bioanalytics. So I think they already have the facility that industry like us would like to have. And I congratulate them on having that. And we look forward to utilizing those going
forward. So ready to partner with us?
Yes.
So he has committed right in front of you. Okay. Next vaccine candidate, Serum Institute of India. Panacea is listening, right? Good. So let me turn to the issue of antibodies. You have been already collaborating with THSTI. This is an area which is like CAR T-cell therapy, monoclonal antibodies is all over the place, right? Including now people think that even bacterial infections, there could be use of antibodies because all the antibiotics that you have the bugs are resistant to them. So you tell us in terms of both diagnostics and therapeutics, because antibody development has been going on at THSTI and other institutions for a while. I mean, during COVID also it happened for both for diagnostic and therapeutics. Didn't turn out to be very, very effective. For HIV, Jayanta and his group are working on that. And now you are working on Nipah with THSTI. What do you see is the future of therapeutic antibodies outside of, say, autoimmune disease and cancers, and where you think that institution like THSTI could also help develop newer antibodies?
Thank you. So I think when I first got to know that we were having an institution in India which has novel antibody, it came as a pleasant surprise. Let me start with that, right? It was not no, and I think it's a moment to celebrate it because it is not obvious to me that there were many institutions who are doing fundamental research. So I think when we got to know and then we started having conversation with the institute and the leaders, okay, what is the next step? Who is going to sell the product? How are you going to give this product to the patient? Do you know what is the price point you're looking at? They said, no, we'll look at it later. I think a lot of the conversation and rhetoric here today is around infrastructure. And I think we have infrastructure. It's the same Cytiva equipment. It's the same Miltenyi equipment. It's not going to change between Belgium and India and China. Right? It's the same. I think it's about mindset of how are we planning for it. So my request is to the leaders, especially in the institution, think patient backwards.
That's right? Very important.
Because if we are thinking infrastructure forward. I think that's and both are important. I'm not saying
Dr. Karthikeyan actually is a clinician.
Yeah, I'm aware.
Clinician first.
Yeah.
So he thinks from patient's perspective first.
Yes.
So it is bed to bench and then back to bed.
So that's one loop, right? The second loop is the economic loop, right? Different countries have different economic models. Some are insurance. In India, it's not so much on the insurance scale. So how much is an average patient willing to pay for this, right? Typically, Nipah virus, you'll have to think from, like in all pharma companies, you'll think business case forward. Okay, Nipah virus starts in Kerala, moves over here, so many patients, so much price point, and so on and so forth, right? So I think we'll have to start thinking at that lens as well in terms of how we want to take it to market, because the proof of the pudding for the industry is, okay, what is the track record of this institution in commercializing a molecule either within the country, or I was also glad to hear also in some of the other countries where Nipah virus is also prevalent, like in Southeast Asia and so on. So I think what it takes to take it forward is we have a foundation of the infrastructure and the talent here to discover and get it to, let's say, clinic phase 1. I think if we can show the full picture, then we have different experts who come in different parts of the value chain. For example, you have subject matter experts who are in contract manufacturing and development. We have experts like Cipla who will take the product to the patient, right? We might have experts in the government institutions in distribution. The challenge in monoclonal antibodies is distribution, right? You need cold storage most of the time. So you have different experts all coming together with getting the product finally to the patient, right? And I think the infrastructure which is available here and the scientific talent and the technical leadership is a great start.
But in terms of commercialization, I mean, institutions are not supposed to actually, think about that, do something about, because if they start doing that, I think then they will move away from your primary research focus, right? And in terms of the cost that you mentioned, I think this is something that will always come down over a period of time. Once you have economy of scale and you have infliximab, for example, anti-TNF antibody is something which came in when India, first time, it was 1.5 lakh or 2 lakh per injection. And now you have biosimilars for 5,000 rupees per injection. So it always comes down. The challenge is in the development of that. And I'm very happy that you have a collaboration with THSTI in relation to antibodies also. And there are many antibodies which, to my mind, will go forward and be the therapeutic modality of choice in many diseases. Dr. You want to make a point?
Maybe one important point here is that what we lack here means we have very fantastic chemistry teams. We have very good biology. We now, in last couple of years, have developed very nice facilities in terms of doing some toxicology, pharmacology. It's all evolving in a very good shape. I think what we lack is basically the clinical development plans. So I think when we start a program, we don't think about how do we do a clinical study? Why do we do with a clinical study? What will be the key clinical partners? The lack of that clinical input into the programs at an early stage. We have seen all the viruses listed here, all the potential vaccines which might come, but without any thought, probably, how do we do phase one? And not only phase one, how we do the proof of concept studies? Leave aside, phase ones are much easier to be done now these days. But how do we design a proof of concept study? Whether it's a cardiovascular study, what is the statistical power? What are the endpoints? What makes a study successful? How do we bring in the regulators at a very early stage? So for all those things, I think we really need to think about what are the missing components in the value chain.
But I tell you that this is a unique institution in that regard. Here we have all the capabilities to develop clinical trials properly. I mean, CDSA with Nitya leads, they are conducting many multi-center trials. You may be aware of that. Dr. Karthikeyan himself has many trials ongoing. He is someone who actually teaches about trial development and design and everything. So this institution is well poised for that. The only challenge is once you don't have your own molecule, which is ready to go to phase 2 or phase 3, then you design it. You are currently there designing it for others.
No, absolutely. THSTI is one of the very new institutes. I was talking mostly from the India perspective, broader Indian institution perspective.
In India also, we have the capability. So what has happening so far is that a multinational will come in. They have a clinical trial, ready. We are part of that. We conduct it, get paid for something, and then it is published. And we think the capability doesn't exist in India. It is primarily because we don't have our own molecule. Once you have your own molecule, you develop your own clinical trial. Yes.
Maybe I can actually cite an example. What we are currently involved in is we in-licensed several years ago at a phase 1 stage, inhaled antifungal drug. So there's no inhaled antifungal drug anywhere in the world. And we were actually the first country to complete a phase 2 study, a dose finding phase 2 study of an inhaled antifungal drug in a very unusual indication for which there is no approved drug. And I can say it because it's publicly known, allergic bronchopulmonary aspergillosis. Very common problem in certain northern part of India. No approved drug anywhere in the world. We've completed phase 2. We are now moving into phase 3 next year. The challenge obviously we are facing is to have 250, 300 patients to be enrolled in the study. It's not easy to have AVPA that many patients. How many sites do we need? Will all sites be able to run the study for us? What is the endpoint? There's no approved drug, so there's no clear endpoint. How do we arrive at a mutually agreed endpoint, which is of clinical relevance, but also will lead to faster approval of the drug? So these are some of the things that we are dealing with drugs, which actually we are ahead of the rest of the world.
But you involve institution like AIMS, PGI, CMC, they will give you the right guidance.
So we have in fact involved there. Totally.
Before I come to you, I tell you what, I am involved currently in a multi-center study. What I'm finding is the CRO will pick up the sites which are easy sites. They do what they are told. They don't give any input. They don't ask questions. But if you go to the right people in right institution, they are difficult because they will ask questions. They will question your trial protocol also, but you will get the right input.
So I can only say our protocol has been designed by experts rather than in-house.
So thank you. So I'll quote what's happening now. For cell and gene, CMC Vellore has developed their own process with us, which is made in India for India. It's a 9-day process compared to a 12-day process, which is running internationally. Phase 1 is done. Phase 2 application is going in. So if we are talking of that, I think that's happening. And that's also the plan with THSTI. I think that's where Jitendra Kumar and Dr. Karthikeyan got involved, saying we want to do this. Exactly like you said, PGI, SGPGI. Amit is so excited about running for auto this. So it's happening. I think that's how I would look at it.
Right, thank you. Let me come to Ms. Jain now. From diagnostic point of view, in THSTI, for example, there is a division actually on diagnostics, right? Gaurav, Sushmita, Neeraj, Baptosh, they're all into diagnostics, developing different things. So what is your take on diagnostics coming out from academic institution, and then you take up forward for some kind of trials and finally commercialization of that?
Yeah, thank you. So for us, since we come up from a surgical background and we have proven history in surgical department, so how we came up with the robotics, okay, first time in India. Same way when we look at the diagnostic field and a part of India, a lot of government programs which are running, and when they wanted the last patient and the last mile to approach, we have a competition and a threat coming from the China, as sir rightly said. Because for us, it is to provide on time, to provide on the cost, okay, effectiveness. And for us, we can do it in a scale way, but anything which is innovative, which has done their trials, when it comes to a commercial mass level, the approach, the distribution, the supply chain is the expertise there where we come from. But I was really surprised to see all of these TSTI, how they're really coming up from the academic side to really bridging that gap, which we were as an industry looking forward from the China till now. Okay, let's be very honest. But since we are making India now, so we want anything right from the source to be from India itself. So really look forward for this.
Thank you. So I'll just ask you comment with this Prime Minister's vision of Make in India. Maybe we can start from you and then move down.
Yeah, so for us, we are
30 seconds.
Yeah, we as we are 100% Make in India. We are making from right from the stands to the robotics, for any of the surgery related to cancer therapy and to the diagnostic. 100% Make in India is what we look forward. We are back integrating now. So this is what it's a really good and a proud moment for all of us.
Thank
you. Yeah.
Yeah, I'd like to say that Make in India would give us edge in terms of not only making stuff in a more accessible manner, in a more sustainable manner in India. So we wholeheartedly support Make in India initiative. We would like to see Develop in India approach more getting forward so that we could partner with the developers so that we have a edge, as I said before, to support the Making India program.
Thank you.
I think one or two areas where really India can take the lead in innovating is in the field of infectious disease. We have the highest burden of infectious disease, right from viral, bacterial, fungal. While we see a lot more developments happening in the world, I think there are many places now that I see that India is actually taking the lead in, let's say, antibiotics or antifungals. But I also believe that prevention is something that we should equally work on. And when I say prevention, I just don't mean vaccines. There are several areas of prevention through the pharmaceutical lens that you can do to actually have an impact on infections. And I think that's where we can take a lead on.
So I just spoke to that a few minutes ago, but I think Make in India is something that we as a company globally believe in, because we don't really look at Make in India for India, but Make in India for the rest of the world. Is that something that we could probably bring to fruition? That's the vision.
I think we are the world's pharmacy. We are the biggest supplier. Everybody, half of the world uses the drugs which are made in India, whether it's vaccines or whether it is small molecules. So we are already on a leader's position here. We are going slowly on an innovation scale, which is nice, because I see in the next 10 years we will have more innovative medicines coming from this particular country, and probably from all what you are doing at THSTI. So there is a huge scope in that. And in terms of the opportunities, I see opportunities in cell and gene therapy. I see because it's transforming cancer cure. That's what is the beauty of the whole thing. It cures cancer. Nobody had 10 years ago thought that you can ever cure cancer. Cancer in 20 years from now will be like any other infectious diseases, manageable. It will be preventable. So prevention, as you rightly said, is a huge thing. It's where the medicine 3.0 will land up rather than treat, prevent. Whether it's cardiovascular, whether it's oncology, whether it's neurodegeneration, prevention is the key. And when you match that with your holistic way of life, with other strategies, intervention strategies, I think that's where Atmanirbhar Bharat will land us.
Thank you.
So finally, I think first and foremost, I think the mindset is there. This conversation was not happening 10 years back. I think the mindset of Make in India is happening. I think to accelerate it, I think we need a combination of talent, which institutions like THSTI are providing. But we also need to remove the fear of international companies coming and collaborating with us. And I think one of the biggest hurdles, I think we can help them and also therefore help us, is making the policies more easier to do clinical trials, to invest. And I think that is an area where we are already seeing many companies come here. The big pharma companies are coming, setting up shop. And that is a big area where we can make their life simpler, and the Indian ecosystem will also significantly improve. And we have seen this in different industries. And I think pharma innovation is the next big thing as well. Thank you.
Thank you all very much for these wonderful comments and remarks. And thank you, audience, for very patient listening. And I'm sure that something definitely useful will come out of this institution with the help of industry. Thank you all very much. Okay.
We extend our heartfelt thanks to all the panelists for such an engaging and enriching discussion.
ये हमारे लिए अत्यंत सौभाग्य की बात है कि भारत सरकार के जैव प्रौद्योगिकी विभाग के सचिव एवं डायरेक्टर जनरल ब्रिक डॉक्टर राजेश सुधीर गोखले जी आज हमारे बीच उपस्थित हैं। डॉक्टर गोखले जी ने सैनफोर्ड विश्वविद्यालय से पोस्ट डॉक्टरल अनुसंधान के उपरांत नेशनल इंस्टीट्यूट ऑफ इम्यूनोलॉजी में संकाय के सदस्य के रूप में तथा तत्पश्चात इंस्टीट्यूट ऑफ जीनोमिक्स एंड इंटीग्रेटिव बायोलॉजी के निर्देशक के रूप में कार्य किया है। उनका अनुसंधान कार्य नवीन मेटाबोलाइट्स और उनके पाथवे की खोज के पर केंद्रित है जो फिजियोलॉजी को प्रभावित करते हैं। शोध में उनकी प्रयोगशाला ने माइक्रोबैक्टीरियम ट्यूबरकुलोसिस से नए मेटाबोलाइट्स की पहचान की है जो संक्रमण प्रक्रिया भूमिका निभाते हैं। साथ ही उनकी टीम ने जैसे ऑटो इम्यून रोग की जटिलता को समझने में भी योगदान दिया है। इस विशेष अवसर पर उनका हार्दिक स्वागत करते हैं। आपका कृपया अपने विचारों से आलोकित करें।
Namaskar and a very good afternoon to all of you. First of all, it's a really wonderful occasion participating at THSTI, one of our really which are always called brand new because it's always doing new things at a speed that is really amazing to really follow up. It's probably a kind of a small presentation. So is the presentation here? Actually, you want me to put a presentation or?
Or should we just talk like that, Achilles? So I was just trying to put together a department of biotech, looking at for the future next as in next about 5 to 10 years of what kind of milestones that we're looking at. And, without all of you participating in this activity, we'll not be able to make this really an impactful stuff. So you want to set it up or? Or email, it always takes some. No, I thought you had already sent. Maybe. Since I just planned it yesterday, my office didn't suit it out. -1 less than percent. Fine, so you can send it to see. Just download it from WhatsApp. No, wait.
Just follow my call for I'm saying. Okay, so sorry for this small break, but I think it may be refreshing for all of you to just come back to the live. I think what I really wanted to talk to you about today was how Department of Biotechnology envisages the future of our country's progress. And largely the medicine sector is going to undergo, in my opinion, so much transformation in a way that can we design it, can we orchestrate it in a way such that it actually does make an impactful for India and take leadership, as Dr. Pramod was constantly insisting on each one of you to be able to take industry partnership along with academia to make these exciting things happen. And that's why I put this first slide, which is really to do with the fact that no one can whistle a symphony. It takes a whole orchestra to play it, actually. And clearly, institutions like this are still like a whistle only. It cannot become a big picture till it encompasses with academia, with industry, and everybody coming together to make this real story happen, actually. And I think this whole event of Synchn that so very nicely Director THSTI has initiated since last year actually just has an amazing impact on how one can really stitch together all these activities. I think you need to download it because if you don't download it, all these slides will go bad actually. And it's not downloaded actually. Is there a PowerPoint on here? Yes, sir. But otherwise, I'll just speak. I mean, it doesn't matter. Look like this. It's okay. Thanks. Thank you. Industry academia partnerships are a cornerstone. Digital world, anyway, it's always fun. So let me just give you a perspective of how Department of Biotechnology is growing leaps and bounds and it's constantly evolving mechanism that is empowering this country for the future for the future readiness of the world in different ways. It started in 1986, and since then it has kind of focused on capacity building, primarily cutting edge research and innovation. And when it took up 2020, set up an entrepreneurship as a model where BIRAC was set up actually, and it really started to set out completely new ecosystem of innovation in this country. And now together with academic research organizations, startup industry, we're hoping that industry partners with us to really develop bioeconomy for the country actually. And when you talk about bioeconomy, it does require all the three pieces together of the research and academics ecosystem, the startup ecosystem, and the industrial partnership. Why this is so important actually, and I sometimes really can think of what we were and where we are actually. And this is as a country, not as an institution only actually. If you really look at not too long ago, 80 years back, many of you may recollect this 1943 Bengal famine in our country actually, which completely impacted the entire ecosystem. The food was in scarcity. And this is not very long ago, if you really think of in our country's perspective. And however, if you really now look at the recently, we now at the stage that we have an Indian obesity challenge actually. So you can imagine
from one end of the spectrum, we can reach other end of the spectrum where one the scarcity of food and the scarcity of food. And now there is an enormous amount of lack of food and agriculture that is available, which is actually a kind of tremendously interesting. And therefore, what we do is to create health and wellness, which is very different from both the spectrums of the systems that we are talking about actually. And as these systems, we've kind of been a country where things were in scarcity and many of these were kind of uploaded onto us to define. Now we've reached a stage where we can start defining our contours of what India means for tomorrow. And that's where I think the health and wellness becomes a very, very important point of view actually. So to begin with, of course, if you the health and wellness, the most important, the critical component of food systems actually. The food systems of our country are actually has to balance supply, sustainability, nutrition, and security. And this is very important because at any given point of time, the health cannot be only looked at from a pathological point of view. It also includes the component of wellness, actually, right? And the more better healthy individuals are, it's better for the future of our country. And it's going to come from the food systems that provides the balanced nutrition. And our recipe for feeding completely right by producing quickly and putting sustainable strain on the planet is actually one of the biggest challenges that we now as a country faces actually. However, if you really imagine, there are 800 million people still don't have enough food to eat, and about 2.8 millions cannot afford right kind of food. So this imbalances in the world obviously impacts health and impacts variety of activities as well. And then therefore the decisions need a very different way actually. Can we develop our sustainable, secure solutions? Can we unpack best practices and policies? Can we untangle the agro-industrial complexities that we are struggling with our country? On one side, we have small farms and large sectors to look at. And then the pressure test approaches that we need to do actually to be able to define how inclusive economic outcomes can come from it. That needs to be tested in every industrial sector as well. And these are very, very important things because it talks about the resilience of the country's perspective actually. Coming to the other spectrum that has actually an industry that has actually again revolutionized everything is really the part which comes from an immunization programs that was developed in 1974 actually. Again, if you look at immunization of activities was worldwide, was decided for 6 diseases, tetanus, polio, measles, and tuberculosis. And in last 50 years, if some of you would like to go back and look at a very nice review article in Lancet, actually, that talks about how additional 7 other infections like Haemophilus influenza, HPV, hepatitis, rubella, pneumococcus, rotavirus, and more recently HPV and COVID, in fact, are adding really benefit to the human life and values actually. Now, all of these are vaccinations, mind you, and what we are talking about today is solving the problems of therapeutics and how we can make health and wellness as a together perspective. But if you really look at this paper, it talks about how much dramatically the innovation has been one of the most universal innovation that was for the humankind, because brought about the deaths dramatically low. It improved the quality of life of people across millions and billions of people actually, and how impactful this whole ecosystem has been. That it has kind of provided a completely in a completely different mindset to the entire ecosystem, and therefore, with these results in mind, one is the food system, one is a vaccination system. One needs to start looking at the other component as well, really well, which is a therapeutic world actually. And presently, the kind of looks at symptoms, looks at diagnostics, and finds drugs for each of the conditions actually to look at. And this is being classically practiced to the pharma industry's perspective. And for example, if I take an example, if you take a type 2 diabetes, classical 46-year-old male, which you may find it very commonly in India with type 2 diabetes, NAFLD, dyslipidemia, and despite pharmacotherapy, the person is undergoing early sarcopenia markers, there are cardiac conditions, flattening cortisol rhythms, and visceral fat on DEXA. And so many of these challenges, actually, you don't know which trajectories to take, you don't know what kind of medicines to give, you don't understand what kind of, mechanisms that you treat these patients with, and you keep identifying symptoms and keep qualifying them for doing these activities, right? So how do we get this such that we actually get into the more integrated systems, actually? And I think that's where the biology is the future of the world, actually. And I think that's where all of us are developing our abilities and setting up this. And the whole of the systems will focus from the symptoms to the systems, actually. And so how can we really design a mechanism by which variety of tools and technologies can empower us to really start looking at a vision that is actually very futuristic, yet within the range of this, actually. And you can imagine, multi-omics of clinical pathological symptoms, all the multi-omics data, which is continuum of variety of collection of data sets can be collected out. And with the immense computing power and AI technologies, actually, we can really convert this into what you would like to call a graph, which then guides into pathway simulations and gives you a perspective of response predictions, actually. And eventually, I don't think it's too far away. You can have personalized health plan created systems on your cell phones that provides you looks at various aspects of your life, not just talks about diseases and symptoms, but looks at the pathophysiological consequences of various activities to do it. And can we do this? Can we work on this together? Can we bring out groups of industry, academia, and startups to really design this ecosystem is what is very, very important and futuristic. And I think it will provide an incentive to each one of us to actually build it in the most possible way. So the back end of this requires a lot of work. And this is where the digital twins are going to be the most critical component. If you look at what the details of this activity, which the phenotype that I talked about, the back end will be very, very important. For example, if you say that hypothalamus pituitary adrenal gland axis is perturbed actually in this kind of a patient, actually. And because of this, there are lots of metabolite changes which are leading to the oxidative cells. There's also mitochondrial functions are actually quite compromised. And due to this, the gut immune axis is also perturbed, actually. And therefore, you are leading to the muscle breakdown and various parameters. And I think these are very powerful symptoms that one can start predictions to be able to look at. And therefore, probably prescriptions would be more like targeted strategy and tailored actions where you not only talk about what kind of molecules to take, but also what kind of substitution and food systems you need to do, what kind of other activities like pranayama and others can be added into it so that you can actually alter the axis and bring it back. But mind you, this requires for us to integrate and really build the activities to be able to, come to the best of this level. I mean, let's not deny the fact that we are actually a global hub for production of affordable vaccines and generic pharmaceuticals, actually. And we should celebrate these successes as well, actually, because it's no mean achievement that India is one of the global player, particularly for low and middle income countries, actually. And so therefore, we should be really what we have been able to deliver both in the clinical level and all the activities that we do. The challenge really is, despite producing 65% of the world's vaccine and supplying high quality, affordable medicines to countries, the innovation ecosystem in our country still suffers from a variety of challenges, actually. And I really think nobody can deny that next generation therapeutics leadership India can provide. And I am one of those who really believe that this is indeed possible in the next 5 years if we really start playing each other's role in an integrated way, converge in a way such that it actually tells you very clearly what one wants to define. Now, this is where the precision biotherapeutics has been listed out as part of the BioE3 policy as one of the key cornerstone of our country's perspective, actually. And this requires 3 different aspects to be done. One is obviously the individualized treatments, how one can one size fit all treatment, which is the kind of example that I showed. Almost all the people with same kind of symptoms will have identical treatment as of today. Can we really have targeted therapies such as gene therapy and monoclonal antibodies and variety of other RNA therapeutics? And then our systems medicine culture of Ayurveda and also understanding various things such that we can really use this to the best of the practices. And obviously, there's undeniable, that 2047, if economically developed nation, it has to be integrated with healthy and productive population, actually, because without that, that cannot be thought to be a very economically healthy perspective, actually. So clearly, health is integrated in all the ecosystems, actually, in a way that actually becomes very, very important for this activity. I think I should really point out that one of the key programs, that was one of the program for vaccine industry was really transformative in our country, the Mission COVID Suraksha, actually. Of course, you can say everybody was focused on only one aspect of it, and therefore this became such a very successful model in about 2 years' time. But what it shows that if you put in all together efforts as an integrated way, we can really achieve something that is outstanding. I mean, for example, you look at DNA vaccines were set up, RNA vaccines were set up, nasal vaccine was set up. Every platform that you can think of that vaccine platform is required was enabled in this country, actually. And I think that's where the real key challenge is that we know we can do it. The challenge is why are we not doing it is why we should continue to ask this question. And if you address why in these meetings like Synchn, you probably will be make solutions for it, and therefore start finding wonderful problem statements and solutions at the same time. I also think health and sustainability are also two very deeply intertwined models, actually. You need to have a healthier planet that leads to healthier people. And unless you do that, for example, every now and then in winters, we talk about the air pollution in Delhi, actually. And, this has been repetitively talked about it, but there are no solutions found. And there are solutions. It's not that there are no solutions. The countries have got rid of it. There are various ways of doing it. And I really believe that that's where the country is looking at, where Honorable Prime Minister heading the cabinet actually approved this policy called Biotechnology for Economy, Environment and Employment, actually. And it's almost about 6 to 8 months that government has given us specific mandate and a framework for how to put future scientific, industrial and societal advancements. And this is really the future of how biotechnology is likely to really be the key player in industrial revolution of the fourth industrial revolution that we all talk about right now, actually. But to do this, as I said, the strategic roadmap has been identified, and biomanufacturing and biofoundries and setting up a variety of things. But as you see, precision biotherapeutics is one of the key perspective. That is one of the key component of it, actually. However, this has to get integrated with various other important activities such as bio-based chemicals and enzymes, functional foods, climate resilient agriculture, carbon capture and utilization, and marine and space research, actually. Now, just last couple of thoughts about what academics really need to do and how our systems needs to get modified. Because if you want to achieve this bio 3 goals in next 25 years during the Viksit Bharat, what is really important to do, actually? And I put together these 5 thought processes, which I really think is doable. And therefore, I'm 100% sure that we can achieve these targets if we really coordinated our way, actually. And some of these are obviously elevating our research excellence, leveraging and mobilizing finance systems. Actually, our finances has to get integrated into an innovation ecosystem such that it brings value, and the value keeps rotating back into the system so that the system stays robust and innovative. And the monetization of diverse portfolios and coordinated regulatory systems, actually. However, if you really try and think of it, the kind of sectors that we talk about really cannot only be done by the government, actually, sector. And what is the key challenge is that the government budget allocations really means that we mobilize private investment through a structured facilitation mechanism that allows us to integrate our resources rather than leave it into the hands of somebody else to dictate, including investors and including private players, which are kind of have their own ways of looking at it, actually. I think, as I said, BioMaterials together and institutions like these kind of have been putting together the best models to be able to look at. And I wanted to talk to you about this alternate investment fund model that BioMaterials and DBT has set it up actually, which is one of the, I think, one of the best ways to really take forward and accelerate the funding mechanism. And the reason I'm talking about it is because through the new 1 lakh crore fund, which is now being announced by the government and kind of will be mentored by the NRF actually body, the body and the DST. I think there is a tremendous opportunity for us to aggregate our resources and aggregate our ideas and innovative challenges to put together it. This is an example what BBT BioMaterials has already done actually. You should look at, there's about 150 crores of about investment what DBT has done and has put together about 16 ACE fund partners with about 349 crores of direct investment by other partners. And in fact, this has resulted in total daughter funds investment of 1,200 crores. And if you really add two of these, the total in all these companies and raised on other investments, and so total investment that has been leveraged in the ecosystem is about 5,500 crores. So you can see how 150 crores of investment of Government of India can leverage very large amount of funding in the systems actually. So just imagine what one can do with 1 lakh crore in this country. And I'm again saying this because we need to set up programs and projects of 500 crores each, about hundreds of them in this country in next 5 years, such that you can really amplify biotechnological innovative solutions in various parts of the country. Actually. And I think this is where what we can achieve by 2035. And I really think that it's the most critical time in our next 10 years of this country is very, very critical and important of how we define this and how we carry forward this. And that's why this RDI scheme of 1 lakh corpus fund is actually so timely that it can be completely transformative for the fourth industrial revolution that we're talking about in terms of various other sectors that will put together and convert into this model. I leave this part, but just to end, actually, what we also need to do is to change our ecosystem and Director THSTI and various other institutions are planning to do actually. And I think Manish has already talked about it today. Have you? You've kind of mentioned it briefly. Is to really change our academic ecosystem to embrace other parts of innovation ecosystem as well actually. And I think what we are planning to do is to trying to set up what we call BRIC Bio3 innovation parks in about 5 cities in the country right now actually. And this being the first nodal place to be able to look at it. And you can see this is where you are now actually. If you really look at this picture, about 200 acres of land in Faridabad, Gurgaon, the most ideal location for having an inflow of skilled human resource, scientific research, having an IBDC, the data center over here, having an academic setup over here, medical hospitals, business schools, and industrial zones. And I think if you really put together this as an ecosystem about the next 2 to 3 years, if you can mend it, you can amazingly see the impact in the country that you cannot even imagine. Of course, THSTI, to its credit, has done a lot of fantastic things, like have been an important player in COVID vaccination programs, have been the IBDC, as I said, is there. There are BSL-3s, there are a lot of animal models available, all sorts of technology platforms for vaccines and drug and variety of activities. And this is an opportunity to be part of the Viksit Bharat goals of our country, actually. And if we miss the bus now, we probably can never get it back. And every industrial revolution is an opportunity for the prepared minds to really embark upon the journey in a collective way, which I'm sure the Department of Biotechnology, along with other institutions, can really make this happen. I thank you all for coming over here, and my really goal is to really motivate you to say, please come and innovate in India and institutions like these. That is our best ways to really make it successful. Thank you, all of you.
जीव विज्ञान पर आधारित स्वास्थ्य सेवाओं के विकास पर अपने विचार साझा करने के लिए आपका हार्दिक धन्यवाद। मैं एग्जीक्यूटिव डायरेक्टर महोदय से अनुरोध करूंगा कि वह मंच पर आए और डॉक्टर राजेश गोखले जी को स्मृति चिन्ह भेंट कर उनको सम्मानित करें। Now we move on to our next session that takes a closer look on how strategic investments can drive deep tech innovations and build sustainable translational ecosystems. Joining us for the fireside conversation are two exceptional leaders. Dr. Siv Kumar Kalyan Raman, CEO, Anusandhan National Research Foundation, ANRF. And with him, we have Dr. Manish Diwan, Head from Biofoundry, NCR Biocluster, BioRack. Dr. Kalyan Raman, a celebrated engineer and thought leader, heads ANRF, a newly established body aimed at fostering R&D across India's academic and research institutions. A fellow of multiple prestigious academies, his work spans cutting-edge tech development and public research leadership. Welcome, sir.
Thank you, Dr. Shiv Kumar, for agreeing to be here and in this August gathering, first of its kind, where we are bringing industry much closer to the academia. And since morning, we've been hearing this particular region, what it wants to do, both at the institutional level as well as the regional level, and making an impact at the national and global level. In the audience, of course, the front rows and the middle are occupied by the industry representatives. We have investors. I can see some shining faces. And there are scholars, PhD students, scientists who are partly here, as well as whatever we talk, that is also going to be heard in the corridor outside. So everybody is all ears and eyes to you, essentially. So thank you and welcome you here. I think Dr. Shiv Kumar comes from an industry background, and with Microsoft, IBM, GE, GE also. Yeah. So, and he has taken up a position, the highest position in the statutory body ANRF, Anusandhan Research Foundation for India. And now he has the deepest pockets in the country from the public side. So maybe kindly help us understand to begin with the ANRF vision and what you intend to do with this large responsibility.
Thank you, Manish. And it's wonderful to be here. I want to thank DBT Secretary. I want to also thank Secretary Gokhale, who is on my board, as well as Dr. Karthikeyan for inviting me here. Karthikeyan ji just came to my office and spent a lot of time and said I must come here. So it is really wonderful to be here and be educated by all your talks. And since I'm not from your community, it is really helpful for me to understand this further. So thanks once again. So first, I want to mention that ANRF has two parts. One is the core set of things that we do. ANRF does a bunch of horizontal investments.
Sir, I'll just confirm. Can you hear sir at the back end? All okay, sir.
Okay,
Sorry.
wonderful. No problem. So ANRF has a set of core investments that we do through our grant programs, the erstwhile SERB has been integrated into ANRF. So, for example, tomorrow there is a deadline for an advanced research grant program. That single program we will invest more than 1,000 crores of hundreds of grants. That's just one example. In addition, there are early career grant programs, many fellowship programs, and so on. But in addition to our horizontal investments, we also are doing mission mode investments. You will see us do a lot more mission mode as we get approved. And we are also, in addition to doing mission mode, which are more outcome oriented, in these mission mode programs, we will be having more stakeholders, not just academia and research labs, but collaborating with them. We will see increasingly other entities like startups and others partnering. So, we'll be shortly announcing a couple more mission mode programs, of interest to you. I don't want to spill all the beans here. I keep joking to some of our colleagues that we will be launching mission mode programs like Rameshwaram Cafe launches dosas. So, going forward, there'll be many more. So, all these mission mode programs will span TRLs, more than 3. So the base programs will be 1 to 3 or 4, and then the vertical mission mode programs go up to 6 or so. And then the RDI program you refer to is additive to on top of all of that, right? So, that will be more for the scale up and so on, right? So there'll be a bit of overlap between the 4 to 6 area and between the RDI and what we do. But fundamentally, the 7 to 9 scale up will be supported by RDI, which Professor Gokhale also mentioned.
I think we talk about generic research, Me Too research, and I think there is a recognition that we need to move towards deep tech innovation, have our own in-house proprietary knowledge. Do you think that these mission mode programs and there's larger focus to nudge the ecosystem towards that side?
Look, I think it's not just the mission mode programs. In fact, if you read our call for proposals for ARG, which is our base program for, we are encouraging a more bolder proposals, even though the grant size might be much smaller. So it is not a matter of size. It is just that our ambition to doing more important things, whether it's in the Pasteur quadrant or even if you're doing basic science in the Bohr quadrant, aim bigger, right? I think that's the general message we are sending in all our programs, number one. Number two is we are focused a lot on enabling collaborations of various kinds, collaborations between industry and academia, and we'll be investing more in different types of vehicles for that. We are also going to be doing interdisciplinary collaborations. Again, that's something we encourage across all our programs. Now, mission programs are completely outcome based. It's not a center investment and so on. We assume centers exist, but our goal is to drive towards outcomes. Now, those outcomes could be, sort of TRL6 outcomes. It could be different types of outcomes, but something that is well on the path to scaling, right? So that would be the nature. So there again, we would be focused on breakthrough ideas. We will be prioritizing our investments in things that are not Me Too and things that push the boundaries, right? So that would be the nature of how we do things across the spectrum. And as part of that, we are inviting various sorts of collaborations, and our money is also going to more and more types of stakeholders. I think this is the general pathway without being specific on any specific mission, how we have done it in the past versus in the future.
So as we saw that biomedical innovations and bioprocessing innovations take a lot of time, and also different set of expertise. Some from core subject matter knowledge. Some, I think, for the translational activity need to see industry stepping in. Some to do, let's say, clinical evaluation. You need professors from, let's say, clinical side, hospital, clinical research. So do you think that the kind of work that got reflected today in the morning, proposals which are multi-institutional, multi-PI centric, could have better take rate And if, for example, industry wants to participate, can it have first of all, can it have a window of opportunity to receive some amount of risk funding so that they can also contribute? Maybe I'll leave it up to you. What are the expectations both for both of these?
Yeah, look, I think the way we are looking at our core programs and our mission mode grant programs, these are all grant in aid programs, right? So whereas the RDI fund is completely targeted at the private sector. So the idea is that the RDI fund will invest only in the private sector, whereas the other programs which we are doing will have a mix, right? So the horizontal programs and the fundamental foundation programs, we are funding academia, we are funding research labs of various kinds, and we are inviting collaborations. We have a mechanism called an honorary PI where the collaborations can happen where international travel is standard. So we are enabling many other forms of flexibility to drive collaboration, even though the capital is not going directly to industry in those programs, number one. Number two is in our vertical mission mode programs, we are starting to liberalize and having more capital go towards a combination of startups, combination, not in isolation, right? It's not just I'm giving money just to startups there directly like Bharat does in some of their programs or your programs. So we'll be looking at a combination of academia labs and so on to really drive those collaborations. See, if you look at it, there's no point hand-wringing that industry academia collaboration is not happening, right? So I want to put my money where my mouth is. If I want collaborations, my funding is available if you collaborate, right? So that would be the nature of our thinking in these grant programs. And of course, specific mission mode programs will have different characteristics, and you will see some shortly coming up in the next month, which will give you more of an idea of how we are thinking. But this is not an end state. We are progressively liberalizing it further as well. Now, we do have ways for larger industry to participate in these mission mode programs, either through 10 or 20% contribution and so on, versus me sending money directly to the larger companies at this point, right? So whereas the RDI program is where we are directly investing through a multiple set of vehicles in a mix of private sector entities. So that's the thinking of, different parts we are supporting, but the middle tier of our mission programs are places where pieces of this puzzle can come together, and then there is a liftoff in a much more focused set of sectors in RDI. So that's the theory of the case. Right?
I think, and do you think that there is enough appetite into the ecosystem for the later phase adoption from academia and the industry coming up to taking it forward from TRL 4 to all the way upwards?
We've got tremendous response for the RDI program. I think DST has been putting together all the mechanisms, and the cabinet approval has happened, and we've gotten lots of inquiries post that. But of course, as was clarified by DST Secretary and PSA in various interviews, the RDI ANRF will be essentially a custodian of the capital, which will then be sent to second level funds, which will then do the onward investment. And those can be of different AIF forms, such as what DBT Secretary mentioned, or other forms, right? So there will be different approaches that will be experimented with significant amount of capital. And we expect different outcomes from different vehicles. Right, so that would be the nature of how we plan to make those, and we have seen a lot of interest coming in. But I think clearly this is just the start, right? Yes. We, at least I would love to see this to catalyze various things. The Honorable Prime Minister has been clear to me that our goal is to be a catalyst, not a crutch, right? But if that goal is to be true, this should catalyze more collaboration. This should catalyze more translation. This should catalyze more research labs showing up in India, more fully private sector owned research and risk taking happening, right? So, and risk taking is not just for the sake of risk taking, but the risk taking is ultimately with the aim of reward. And if that reward or return on capital is only going to happen through better market linkages, I think the more we are able to identify all those gaps And the government, as a catalyst, sort of removes those gaps and makes it easier, then the entrepreneur's journey is eased a bit, right? So I think that's again, the theory of the case of how we're thinking about it.
I think, thank you for elaborating. So there is something for all the stages from TRL 1 to TRL 3 to create the base level POCs, and then for the further translation where industry participation can happen to take it from TRL 4 and upwards. I think we, through Synchn, we try to kind of figure out what industry would want and how we can engage better with the industry. What is industry ready output? What is the starting point where eventually somebody might be interested down the line? So you have come from a large stint and experience from different industries, of course, different areas, mobility, renewable energy, computing, providing multiple solutions. And you've seen not only India as a geography, but many different geography, cross-cultural. So could you, from that hat, could you also tell us what are the important points that I think as an ecosystem when we are trying to build, how the two worlds, industry and academia, can come closer to take it?
I think we're at a wonderful point in that journey. Excuse me. But I think that's the best way to say that. When I worked in the US or in Australia and so on, right? So the quality of collaboration is much, much higher, right? So I think we really need to focus more on quality in addition to just quantity, right? And also the aiming for much bigger things should become second nature to us, right? So that's why I'm so pleased to see what's going on here with THSTI, both in terms of your people, the folks you have assembled here, the quality of conversations that are going on, and also the various facilities, and not just what you already have, but what's yet to come, and also the depth of collaboration that is, and also the ambition attached to that, right? All of these gives me great hope. Okay, wonderful. We need to be able to replicate all of this more and more.
I hope people are listening. Dr. Shiv actually spent the entire morning here. He was the first one to come here at 8:30 and actually took a tour of the facility, interacted with our scientists. So after maybe he finishes his remark, I'll probably open up for one or two questions from the floor, from guests as well as from in-house. If somebody wants to ask a question, kindly raise your hand and somebody will reach out with the mic. Yeah, sorry, please.
Yeah, I mean, I think this is a great model, and I look forward to working together with Secretary DBT and your director here, and with Barak and other entities. So our goal at ANRF is to drive collaboration in all its forms, and therefore drive better outcomes. So the capital is just one part of it.
Yeah, of course.
To be more visible.
Maybe capital is the catalyst, right? We are missing in the entire engine. So I think this is an excellent opportunity. Doesn't come very often. Dr. Shiv Kumar is right here with us. Anybody who wants to give it a shot? I see one hand here. Can somebody reach with a microphone? And of course, I think the investors are up for the grab. So I think one from Amit, both from Amit. Yeah, somebody can reach out with a mic.
Yeah, otherwise you can speak up, and
I can
we'll be happy to respond.
We can reach out.
Repeat your question.
Namaskar. I am Rakesh Bhatia from the Association of Indian Medical Device Industry, IMED. So on behalf of our industry, I mean, you must be aware we are under the Department of Pharma. Medical device industries, our nodal ministry is Ministry of Chemicals and Fertilizers. So there is already a scheme going on which is called PRIP scheme, PRIP scheme, to promote research and innovation in medical device as well as pharma sectors. But there are certain, companies which don't fall under that eligibility. So is it possible to approach ANRF for such requirements or any special requirements?
So right now, the answer is probably no at this point. See, right now, I would split our ANRF goals into two parts, right? One is where we are providing a lot of grants and grant making. The second is the RDI program that will be put together. And there, ANRF is not probably the entity to directly approach. So we will be working with DBT, will be working with, whether it's THSTI or Department of Pharma, as we put up this second level of funds. There is an empowered group of secretaries that will decide what are the areas where the investment is going to happen. And that will lead to a whole bunch of second level fund managers that will be put together. And then there'll be governance approaches, investment committees and things like that, right? So my suggestion is, right now, I'm in conversation with Secretary Pharma as well on how we complement each other. I don't have anything to say at this point immediately, but please stay tuned. Of course, think of the investment that is coming up will complement, and our goal is to again be catalyst and a multiplier effect. That would be the general points you're making.
But medtech is a definite focus area into
medical technologies is definitely a focus area in addition to pharma. So again, I don't want to spill all my beans, but there are mission mode programs we are considering in various areas.
We have one more question here. Amit, if you can stand up for that.
Hi, very good morning, sir. Hi, Amit from IVCA, the Indian Venture and Alternate Capital Association. So we are a network of India's venture capital and private equity investors across the country, both domestic and global funds. And obviously, we've been, keeping our eyes towards the announcements, and we've actually gone back and met everyone from DST to, obviously, we work very closely with Biotech, etc. So just wanted to understand one thing. As of the initial announcement, the funding is supposed to be interest-free loans, 50 years, etc. So, I mean, how is it going to be channelized to equity is the question that I wanted to ask. A lot of investors keep on asking me, so I just wanted to put it forward to you. Thank you.
Yeah, so there is a little bit of misunderstanding overall in the community, right? So clearly, one is that it is meant to be patient capital and capital that also helps crowd in and drives the long-term outcomes that we want. That is the broad area of thinking. So, but that doesn't mean, so the 0% 50-year loan is a mechanism which Parliament gives to the custodian, which happens to be ANRF, which we will be onward giving to on a small interest rate to the other, second level funds. Okay. Now the specific interest rate and the specific period of some capital coming back to us, those are obviously important parameters in terms of determining the incentives for onward investments. So those are again going to be defined in a way that drives the patience in the capital. Okay, so we've had several industry consultations to determine that, but the detailed sort of procedures and guidelines are being written by DST. I think it's important to also realize that ANRF is not a policy entity. We are a funding entity and a vehicle for execution. We are not a policy entity. So all the policy guidelines are being done by the nodal ministry, in this case, DST. The second point is that when it comes to each AIF will be given a clear mandate. It will have a nature of investments and the types of investments and areas of investment. And then it will be given an appropriate horizon and, broad return expectations and so on. So around that, there is a lot of flexibility in how those things will be structured. Now, whether that's a deep tech fund of funds, or if it's a separate types of vehicle, and then we'll be looking at it overall as a portfolio, I think that's the broad thing I'd like to say at this point. And of course, there have been several interviews that DST Secretary has given as well, right? So beyond that, I think I would request you to await some further details. We do have a governing board meeting to be chaired by the Honorable Prime Minister to get a final approval on some other matters, and some more details are being worked on, right? So to operationalize.
So thank you so much.
Yes, Sabr Kafal Mithai. So I think something was brewed. I think it will come out very, very quickly. Sir, I think I'm getting signal. I think
can take one question.
Would be here for the lunchtime also, and maybe
Yeah,
spend little time.
meet me over lunch and so on. The other is I would also welcome conversations. I don't mean to say, please don't come and meet me and so on. So look, I think it is important for investors and also partners to be engaged at all stages of the pipeline, right? So I think if you look at again the global benchmarks and different countries and so on, it's not like somebody is suddenly showing up at TRL6 and then say, okay, pick up some technology and go. So they are engaged at earlier stages of the pipeline in different ways. Now, of course, every individual company or individual entity has to decide how much earlier you want to drop in. So ANRF is open to partnerships at various types of things. So we have done some foundation level partnerships, with the Wadhwani Foundation who's here, as well as the Gates Foundation. So there are pure capital type partnerships we do. There are other partnerships where we have partnerships with industry and so on at the earlier stages. And then the later stages are pure investment where our capital goes out to the private sector as well, right? So I would urge you to think of it holistically. And, as you engage with us, please come to us with a holistic point of view, right? I am talking to different stakeholders, including venture capitalists, including associations and so on. And we'd be keen to see what is a special thing or, unique thing you can bring to the table. And, the past is not going to be an indicator of the future. That's something I can clearly say.
I think I see Amit representing IVCA and next to him Vikram, who is PAN IIT alumni endowment fund. So I think there would be quite a bit of follow through. Kindly as well, I think without private participation, I think public can't move too far, and without public, private people can't move too far. So I think it's mutual. So PPP is the fundamental philosophy to take our nation, build the deep tech innovation ecosystem, so that not only for India, from India, for the world. I think, thank you, sir. Thank you for coming over and spending little time with us.
It is a pleasure. Thank you so much.
Dr. Shivakumar Kalyan to stay on the stage. I request our Executive Director, sir, to kindly felicitate our guests. Thank you, sir.
Thank
you. We kindly request a little patience from our esteemed guests for this one last pre-lunch session, after which we will break for the lunch. So now we delve into a conversation that offers a broader outlook, exploring both the national and the global perspectives in the significance of deep tech ecosystem and the expectations from the NCR Biotech Science Cluster. Joining us for this session are Dr. Jitendra Kumar, Managing Director, BIRAC. Dr. Kumar has championed entrepreneurial models for commercialization, mentoring startups, and catalyzing innovation through private-public partnerships. Welcome, sir. Next, we have Mr. Vikram Gupta, founder and managing partner at IVCAP Ventures, one of India's largest homegrown VC firms. Under his leadership, IVCAP has created India's first endowment fund at IIT Delhi and helped numerous startups scale up with strategic support. Welcome, sir. And the third speaker, Dr. Amit Pandey from Indian Venture and Alternate Capital Association, IVCA, brings insights from the venture capital ecosystem and to accelerate the biotech ventures. Welcome, sir. To moderate this important dialogue, I would like to invite Dr. Gaurav Batra, Associate Professor with THSTI. Over to you, sir.
Good afternoon, everyone. First, apologies for the delays. And I know we are and the lunch will be very crisp. So as India's bioeconomy is growing very rapidly, annual growth is around 18%, and we are about to touch 300 billion USD market by 2030. And that is primarily because of the rapid innovation as well as biomanufacturing push. So in this or we will discuss about how we can keep that momentum and how deep tech and cluster like this, deep tech investment and cluster like this can support this momentum. So I will welcome our guest. If you can just briefly tell something about your organization. Most of the crowd here knows about him, but if you can say something about
So, Vikram Gupta, I run a venture capital fund, IVK Ventures. We manage close to now 6,000 crores, all domestic capital. We invest from seed to Series B. And part of our profits also go back as endowments to the IoTs and IIMs and other education institutions. In fact, we helped conceptualize and set up India's first endowment fund at IIT Delhi. So that's what we do.
Hi everyone, my name is Amit. I'm with IVCA, the Indian Venture and Alternate Capital Association. We are an industry body for the private capital ecosystem. All of the VC investors that operate in the country, whether domestic or global, are members of IVCA, and we are a bridge between the investors and the government of India. Hence, we work very closely with organizations like DBT, Biotek, and across from Ministry of Finance to SEBI, all the regulators, etc., to make sure that we have we are fostering the private capital investments into India's startup and growth stage companies. And I manage the VC ecosystem within IVCA. Okay. Thank you.
I am Jitendra Kumar, Managing Director of Biotech. So as you all might know, Biotech is the central enabler for startup in India, biotech startups. It nurtures startups, funds startups, mentors, and creates networks and creates programs also for the country. So one is the grant program, which starts for ideation, Biotech Ignition Grant. Then we have mapped the whole value chain of the biotech ecosystem in each sector. And wherever there is a gap, we have funded. For example, next level of funding from idea to POC would be scale up. So that we have some funding, and that is not a grant. So grant is only 50 lakhs that we give. After that, we have co-funding mechanism, revenue sharing. And then we also have an equity fund where we fund these startup companies through incubation centers, and we take some equity. Incubation centers also take some equity. And then we have also heard from Secretary Sir about the ACE fund. So fund of funds, where alternate investment for, many venture capitalists are impaneled, and they, fund on Biotech's behalf. So right from ideation to commercialization, we have, funding mechanisms and also any resource that is required. For example, we have incubation centers. We have funded incubation centers where these companies can do proof of concept studies and even scale up studies to some extent. Then we have EUA centers. And so it's the whole ecosystem that we have built over the last 13 years.
Thank you, sir. So I'll start with you. Biotech support in terms of grant as well as in terms of equity investment has transformed many tech startups and venture. What are the key bottlenecks you still see in translating those innovation into final products? And how can the cluster like ours and institute like THSTI can bridge that gap?
Very important question, and is the crux of everything that we discussed so far. First of all, the deep tech innovation requires institutional mechanisms from institutions that are funded, created, so that startups and the spin-outs come out from academic institutions. Because most of the funding goes to these public research institutions, at least in our country, We see the R&D happening mostly into these public funded research institutions. And we talk about deep tech R&D. That means public research institutions have to actually come out with a mechanism so that these startups and spinouts come into the picture, and they grow and they are funded by different other stakeholders that come together. However, we have seen some good R&D institutions, I mean, the industries also taking this R&D. For example, Glenmark, you must have heard yesterday, they have signed a deal with AbbVie for this, anti-cancer molecule. Then we have Wockhardt, which has announced that they are full-time into R&D company, which is very, very good for the country. But all I'm trying to say is that since most of the funding goes to public research institutions, the mechanisms for commercialization has to be robust here in public research institutions. We have in place the technology transfer, the classical mechanism of some molecule, something coming up, and then you license out. However, this is a very weak system, to be honest. It's not a very efficient, system where monetization can happen, in a good way. It's not a system. It's not even, quantification wise. We see a lot of IP sitting on shelf and not being transferred, etc. So we see a lot of problems. We foresee that through BIRAC, at least from the point of view of BIRAC, we think that technology transfer definitely can happen from these institutions to the big industries. However, we have to create mechanisms through which these intellectual property sitting in the public research labs in the form of IPs or technical know-hows have to be spun out as a company or as a spin-out ventures, and that mechanism has to be in place. You must be knowing we have started entrepreneurs in residence. Actually, officially we have not started, but we have actually about to start. We have already thought about all the action plans. So this entrepreneurs in residence would be the mechanism through which these startups could actually be formed from this industry, R&D institutions. And this will be paid. Venture capitalists are involved right from beginning. We have already roped in 4 venture capitalists. So this is a very, very good institutional mechanism that can play a very good role. So to answer your first question, these are some of the things that we have to actually strengthen. For instance, spin out. If spin outs come, then incubation happens, and how much time an inventor can put, etc. And what are the equity mechanism, distribution, etc. of the equity between the institutions and the investor, etc. I think inventor, etc. has been already worked out. All we need is the implementation of those. We need examples of shining examples of faculty entrepreneurship or student entrepreneurship, which can set an example, which can inspire others also to take up such activity. That's all we need actually right now. Now, coming to your second question as to what this cluster needs to do, I see that this cluster is one of the foremost clusters in India. One is because the presence of a very good academic institution, research institution, not one but 2. The Translational Medical Research Center that we saw that presentation about the cluster that is coming up. So all the components have been mapped, look like, and there's a huge space, the area. We have presence of multinational companies like Miltenyi, which has set up their facilities here. It's not a small thing actually for a multinational company, for cell and gene therapy to set up something in academic institution. And then we also have support from the department, DBT. So I see all components coming together here. All we need is the thrust on this area. I mean, the momentum. The momentum is there. I think we'll set up some examples. So, for example, there are research. We talk in the first panel discussion. We saw a panel where inventors were sitting and the industry was, Serum Institute and other industry was sitting, and we already saw one deal coming up. Molecule Serum will definitely would like to take up. So if there is a lack of early stage capital in our country. There are no capital investments, venture capitalists who will come and say, okay, you take this money and you take this molecule to, I mean, there are very less. I wouldn't say no, but it's not as compared to say, I was in the US. It was like so many Chinese companies vying for venture capitalists. Where were the Indian companies there? Yes, no, actually, I didn't see any Indian company, competing for those funds available. So early stage venture capital, of course, it's a market play. Time will come, the country will also have that. It's just a matter of time. But I'm bigger, creating a partnership between the bigger industries and the startups and the academic institutions that we saw in the first panel. That's a very good sign. Let's create a platform where bigger companies can actually have a strategic partnership with the startups, with the academic institutions where they are new leads. This Glenmark molecule, the anti-cancer molecule, actually the pilots, I mean the preclinical was done here, and then clinical trials would be done by industry partner or international multinational company, FBI, etc. So that kind of model has to come up in our country also, where a lot of startups, a lot of inventors from institutions like this coming up with new molecules, especially physicals, and then attracting venture capitalists, attracting lot of collaborations, multiple collaborations, etc. So those momentum has to be built. We have to be more seen at the global level also. I see when I go to global bio US, this time it was in Boston, I could hardly see our startups industry compared to other countries who are leading. So definitely we should pay more emphasis on that.
Okay. Thanks a lot, sir, for very insightful answer. So now the second question is for Mr. Vikram. What makes deep tech startups and venture investable, and where India stack up globally?
Yeah, so first of all, I totally agree with what Dr. Jitendra said. And I also want to, first of all, say that it feels great to be sitting here. A bio cluster like this is an amazing thing to happen. So I think this is a great initiative, and especially for people like us who are actually investing in early stage. Something like this actually provides the entire ecosystem for startups, and I think we just need to market it properly. I think probably that one piece has to be done properly. Now, to answer your question, so I'll take a step back. My background is mostly healthcare life sciences. So I spent most of my time working with pharma companies like Dr. Reddy's, Lupin Laboratories, Pyramid Healthcare. In fact, I launched India's first healthcare and life sciences fund way back in 2007-8.
Oh, wow.
Which was sponsored by the Pyramid Group. And even in the US, I was running the clinical genomics practice for IBM way back in 2000 to 2005. So I have seen this industry actually, from different lenses over time. And I actually believe that a lot needs to be done. In fact, at IVCAP, we made a few very interesting investments. We invested in a company called LUC Data, which is drug discovery AI company.
Okay.
Young IITians who are actually sitting in the Silicon Valley, catering to global pharma companies out of there, biotech companies, academic medical centers, having a back office in India. And I think what Dr. Jitendra was saying earlier is actually very true. I think, see, what's happening, if you look at from the lens, look at how the support system has evolved in the US versus in India. US is the largest market cap of biotech company, Eli Lilly, $800 billion of market cap. In India, you have which is $5.5 billion, which is 45% of revenue is actually from biosimilars, and 25 to 30% revenue coming from generics. So innovation in biotech companies, even at scale, you don't actually see that. I mean, most of their investments is not going into a lot of new innovations, but still the support that you need at early stages. I think the challenge that we have from a venture capital investment at that stage perspective, risk is so high that even VCs are not able to support at this stage. I'll tell you why. Last year, US had $75 billion of venture capital investment only in biotech. Only in biotech, right? So we're talking about the last 11 years in India, overall venture capital investment was about $150 billion, right? The largest in India was $30 billion in 2021. And biotech is just about $2.5 billion, right? So and the challenge is not just about understanding the sector by the VCs, but also about the fact that you eventually are answerable to your LPs. You have to return your capital, and VC is a new phenomenon. And I think and in our case, it's all domestic capital. We have LPs like State Bank of India, Government of India, LIC. These are our LPs. We have to return money back to them, and then given just 5 to 7 years as a holding period of investment, it becomes very difficult to actually just go at very early stage hoping that something will come out.
True.
So I think the model that we have actually conceptualized is we said that the profit that we are going to make, in fact, in a fund, 5% of our profit we are giving back as endowments. We conceptualized the model in 2011, started conceptualizing endowments, where we said that the endowment capital should go back into funding research and development across the education institutions of India, where it should go as grants. So that model we already given back 20 crores. Across 3 funds, we're giving back 200 crores. And that multiplying with 5 year actually raising capital to bring more corporate capital into that to make it 1,000 crore capital. Then give that capital to early stage startups, which has got a venture capital color, but with a grant as the objective. So I think if you follow that kind of a model, there's a much higher likelihood of really supporting those kind of startups, which is what we're trying to focus on right now.
Oh, thanks. So basically, these if we follow as a campus, as a cluster, an institute, that is what encourages you and other funds also to invest in the cluster in some capacity.
I would say the cluster model is amazing, and this is I'll tell you what is exciting with this model is that typically in a biotech firm or in a early stage innovation of this sort, you need an ecosystem to play together. You need scientists on one side, you need commercial thinkers on the other side, you need funding on the other side, and you need infrastructure support. So, and then the talent has to come together, right? So if you have to bring all this ecosystem together, it's very hard to find at multiple places. You go, one place to the other place, and you see even our IOs don't talk to each other, right? So it's very difficult to bring, people on the same platform. So if you have a cluster like this, there's a very high likelihood, and I think we should very actively engage with the investment ecosystem here. We'd love to partner here, bring the investments here, and we're very active there in entrepreneurs here and actually investing in them and leveraging the entire support system here.
Thanks a lot for your detailed answer. So now, Mr. Amit, what would you see, because you are representing one association of investors, what do you want to see in this cluster which will bring them invest in either this cluster or other regional cluster? What is the expectations from VC side?
So I think, first of all, thank you for having me. Secondly, I think, very well covered by Dr. Jiten and Vikram. Actually, I mean, you'll have to give it to, BIRAC for being so ahead of time, because, I mean, when all of this started, probably, BIRAC was one of the first movers in this space. And that is very clearly visible. See, I over the weekend, I was in the Northeast area. There was an event in the Northeast at IIM Shillong organized by the Indian Institute of Corporate Affairs. And I was not very surprised when I met, several startups that were backed by BIRAC. I mean, it was quite amazing because, in the Northeast area, people were like, struggling, sort of in different sectors. I mean, so biotech of all, I imagined would not have reached the Northeast. True. But through BIRAC, it had. I think, a huge round of applause to BIRAC for that, because it is only possible because, BIRAC has actually spread its wings across the country beyond the metros, which is really amazing. So those are some of the things that we have been observing that, Biotech has already done a job, but some of the gaps that, and again, thanks to Dr. Devan and the team here, I was here last week along with my colleague. We were, giving a small session on VC, upskilling for some of the scientists and PhD scholars here. And not to my surprise, of all the people who were in the room, only one person knew that, like Vikram mentioned, 5 to 7 years period to give an exit. So not a lot of scientists knew that, right? So see, we have to understand that, if you want commercial ready companies that are pitching to VCs, they have to have an understanding of VCs first, and they have to have a complete team. So I mean, the understanding of VCs is a bit skewed at this point, whereas Biotech has done an amazing job, but probably some of the talent building, capacity building, some of those things, and again, we are sitting in the right place in the hub where all of this will be solved in due course of time, because this place brings everyone together. Like in the second half, we're having those sort of investor pitching sessions. Again, IVCA is proud to have partnered with that, where we have invited a lot of investors to come and be a part of those pitching sessions. So we need more of these boot camps, right? Okay. And if we continue in this direction, then definitely from Delhi to Sikkim, we'll have startups in biotech that are going to be raising money from VCs.
Okay, thank you. So what I would do, just ask one single question from all the three panelists, and only I need answer in 30 seconds, sorry. So what would you like to see or emerge out from this campus and THSTI when we will meet in the next Synchn next year? Started with you, sir.
I think the core focus has to be IP creation as well as the some commercial aspects. I would say that if we can create a unicorn out of this campus, which is actually starting from scratch and it's progressing towards that, and if there are a handful of those companies moving in that direction, that'd be really great.
No, I think, I mean, we should be having 12 or 15 boot camp, a year from now. So I think, the more we have, the better the chances for raising more money for the startups here. And yeah, definitely, like Vikram said, definitely some big companies coming out of this place.
I think next year I want to see the biotech park coming up with a lot of industries here. At least I would say 5 to 10 big companies. The startups that are incubated here actually growing and graduating to the biotech park and scaling up. I would also like to see some of the venture capitalists who have funded these startups and your good connection with the venture capital world. And I would also like to see some bigger industries creating or collaborating with these startups and your faculty members to have new molecules, taking forward to the clinical trials and commercialization.
Thanks a lot for this insight. I really appreciate the panelists' insight and also the audience for the patience. We will close the session here, and let's thank our panelists.
Thank you to all the panel members. I kindly request all the panel members to stay on the stage. I request our Executive Director, sir, to felicitate the panel members.
Thank you so much for your patience. so much for your patience. We can proceed with lunch. Please note that THSTI faculty, principal scientists, and our industry partners and business guests, the lunch is arranged on the third floor of THSTI. For participants, the lunch is arranged on the so we can take our groveling place there. See you in some 40 minutes. Just are requested to please assemble. Thank you so much.
THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to health. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council with the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council with the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council with the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council with the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council with the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal-child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions. Innovation leads to better health for all. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step forward. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world.
कौन सा नंबर है 3 नंबर ग्यारह नंबर।
Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution, with leading scientists,
ये 8 नंबर 9 नंबर 10 नंबर चप्पल में।
cutting-edge research, and strong collaborations. THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all.
ये 9 नंबर पे।
At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research areas that impact lives every day. THSTI has built strong global partnerships and earned both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics, ready for clinical development. At TRF, we empower academia-industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. Welcome to the Translational Health Science and Technology Institute, THSTI, place where science meets care and innovation touches lives. Located in the NCR Biotech Science Cluster, Faridabad, THSTI is an institute of the Biotechnology Research and Innovation Council at the Department of Biotechnology, Government of India. Since its inception, the institute has been at the heart of India's biotech revolution. With leading scientists, cutting-edge research, and strong collaborations, THSTI is delivering health solutions that are faster, better, and more accessible. Its work spans diagnostics, vaccines, therapeutics, maternal child health, and clinical research areas, both national and international recognition for its impactful research. The institute believes in teamwork, dedication, and the power of science to bring meaningful change. Every breakthrough here is a step toward a healthier, stronger India. It also plays a vital role in nurturing future scientists through training and education. With a commitment to excellence and inclusivity, THSTI continues to strengthen India's biotechnology and public health systems. THSTI, where science turns into solutions and innovation leads to better health for all. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. Welcome to the Translational Research Facility at THSTI, India's hub for transforming cutting-edge research into life-saving healthcare solutions. Our mission is to create a sustained pipeline of preclinical leads from vaccines and monoclonal antibodies to diagnostics and live biotherapeutics ready for clinical development. At TRF, we empower academia industry collaboration through state-of-the-art infrastructure, from pilot-scale GMP facilities to an ISO 13485 compliant IVD manufacturing suite, immunology core lab, bioassay lab, and advanced multiomics platforms. Our Center for Excellence in Innovation and Development of Monoclonal Antibodies produces custom monoclonal and polyclonal antibodies and offers cell line development for targeted therapies and diagnostics. We serve startups, SMEs, and global health initiatives, driving affordable, accessible healthcare innovation for India, LMICs, and pandemic preparedness worldwide. Join us at TRF, where science meets solutions. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions and industry, MRC transforms lab breakthrough into life-saving treatments. The Medical Research Center at THSTI, where science will be transforming into healing, innovation will be driving impact, and every breakthrough will be taking us closer to a healthier and better world. At the heart of medical innovation in India lies the Medical Research Center at THSTI, spanning across a 26,000 square feet area. The mission is clear: to accelerate indigenous innovations and translate them into real-world clinical solutions. With cutting-edge research facilities and a commitment to pioneering therapies, MRC bridges the gap between discovery and deployment. From early-phase clinical trials for vaccines, biologics, and therapeutics to controlled human infection studies, MRC ensures safety, efficacy, and impact every step of the way. MRC is pioneering CAR T-cell therapy, translating preclinical models into patient care, with support from a GMP facility supplying CAR T-cell products for regional cancer treatment. Collaborating with top institutions.
Welcome back. We'll be starting in another 5 minutes. Welcome back. We hope everyone had a refreshing break. It's time for now Ask Me on Anything, an open dialogue between students, research scholars, and the industry leaders. This is a session which has the unique opportunity to understand the real-world challenges in translational research, product development, and career trajectories directly from the experts. To monitor this interactive session, I'm happy to invite Dr. Sushmita Chaudhary, Associate Professor, BRIC, THSTI. We invite everyone to participate enthusiastically since your questions will make this session truly meaningful. Over to you, ma'am. Thank you, Kamala Priya. So, hello. I hope that the students are back after lunch. I can see most of them. So, I would request our esteemed panelists, if Dr. Manish. So, I'd request our panelists to please come on the stage and take your seats.
Hello.
Anywhere. So since morning, scientists and faculty and leadership and people related with industry who are at the top level, they have talked, and I hope as a speaking of the event, our young workforce are actually going to drive this entire movement in the next decade. They already feel quite inspired and excited. So this particular session is for them, for all the students and postdocs and young researchers, researchers. So you have complete freedom right now. You can ask anything with the industry experts here with us. I would request all of them to just introduce in 15 seconds, starting with yeah, please.
So Dr. Priyabrata Patnaik, Deputy Managing Director of Indian Immunologicals. We are a 100% subsidiary of National Dairy Development Board and based out of Hyderabad.
Dr. Jaideep Gokte, I am Global Chief Medical Officer of Cipla. I handle clinical development and trials for India, drug safety, and all medical and scientific affairs for the company globally.
Good afternoon. My name is Anil. I've been with Johnson & Johnson for almost 20 years now. And currently I am at London School. And previously I was at CSR Impact.
Hi, my name is Pravudha Kundu. I'm one of the co-founders and managing director of Preface Biotech. We are a protein research and vaccine development company, which also offers services for development and clinical development. Glad to be here. Thank you.
Good afternoon and namaskar. I am Shirchandra Mukherjee. Just a correction to what Sushmita said, I am not an industry expert.
Yeah.
So I have been working in funding agencies for the last almost 2.5 decades. But by training, I am a medical microbiologist, worked with pharma companies to start with, and last 2.5 decades working with funding agencies like the Wellcome Trust, BioTech, Bill and Melinda Gates Foundation's Grand Challenges India program. Right now, I am heading an innovation program called the Wadhwani Foundation's Innovation Network as its managing director. Thank you.
Good afternoon. My name is Khalid. I'm Chief Scientific Officer at Panacea Biotech, responsible for vaccines and biologicals development. By training, I'm a physician scientist, studied medicine PhD. And prior to Panacea, I was at Hillman Laboratories and International Vaccine Institute. So that's about me.
I'm Suri from Idea Spring Capital. We invest in deep tech, deep science startups, early stage, B2B legacy B2B startups for the world. We have invested in 33 startups across two funds. About 5 of them are in the life sciences biotech space. Before that, I did co-found 3 startups in 15 years with 2 exits. And before that, I used to work in Wipro building compilers and networking software. So it's an engineer turned entrepreneur turned enabler.
Great. So to the students and our researchers, so you can see it's a very balanced mix of academic scientists, industry scientists, philanthropic funders, as well as venture investors. So come up with your questions. Hans, who wants to ask the first question? Yeah, please. Mic to Divya.
Come here.
Or just shout.
Hello. I am a Divya, a second year PhD scholar. So I want to know what are the biggest gaps when a student graduate as a PhD from industry setup and want to join an industry setup. So what are the limitations, or you can say lack of skill set that they want to learn before joining an industry, or if not, what are the skills that they have learned from PhD in academic setup that they can use in industries? Thank you.
So anybody can take up. Probably Dr. Kaul is the best person.
Yeah, I can take this. So I don't think there are, to be honest, in my view, because I have hired personally a lot of PhDs and postdocs at the industrial J&J as well as London School, which was the offshoot of J&J. So I don't think there is per se any gap or anything specific what we are looking when people come out of the academic institutions. Of course, the key with the industrial postdocs or industrial scientific role is that you need to be working as a team because the projects are mostly in a big setting with a bigger pool of scientists all coming together trying to address the problem. So I think that teamwork is the key thing. And that's one thing. And also, I think what is sometimes which is very critical is the data rigor. So having that rigor in your experimentation and that training in data integrity, data rigor, and data accountability is very important learnings which an academic institution can give to any of its students. I keep on telling people failed experiments are always very good. Every failed experiment teaches us something which is very important for future, for the next experiment. So to showcase those failed experiments with as much ease as showing any successful experiment is also key. So I think these are the basic learnings. And of course, the industry people think that we don't publish. If the programs are at the early stage, the publication is also something which is very critical.
Yeah, good. This is Salil Chawla. I am actually a co-founder to MyEasy Pharma. It is in the healthcare space. My son is Yajur Chawla, who is founder of it. He is RCB student. I have come to present him here because he had exams today, so he could not come. And being a co-founder, I have the legitimacy to present. I am trying to make a dichotomy statement here. On the one hand, we see surviving and thriving startup ecosystem in India, touted to be third largest globally. And but given the situation today, we see the fund winter. I know that given the geopolitical tensions, stuff like that, money has become a tough thing. Even if there is a dry time, it is not chasing the startups or that hot ideas what it used to in early days. So the question I have to ask, a pinpointed question, that unlike China, which has opened up between 2000 to 2010, they made it a religious that attempt. as long as your idea has gravity, we are here to give you legs to really see that we are in and uh, both of us become institute experience and from that's Pilani now he's doing from RCB again tier 1 institute. So can you give some encouraging statement here, you know? Thank you. Anyone can answer my question.
See, like you said, there is no money, right? Like actually, I would say there is money. They're going after the few ones which they think is good, right? That's how VCs work. So I'm assuming that, okay, by pitching to people, they might have at least given you some feedback on what actually is lacking or what are the reasons why they have not been able to make decisions to invest in your startup. So basically, I would take that and actually correct those things and keep working at it. That's what is entrepreneurship, right? So I have done 3 startups. So just to give an example, the first startup I got $1 million before we started. The second one actually like we took like 6 months. Then MVP on that road. The third one actually we decided not to go to VCs, still we got some customers. So we have to decide and so keep thinking on it and then see how to actually go about it.
So Shushanda, you have something to say?
See, if I can make a statement, which I think in the first session also, CEO, DBT Secretary, CEO, and CEO also was there. Is not a basic problem right now in India. You know, as a funder, I can tell you, we are evaluated how many good projects we bring to the table. As was said by my predecessor, you know, when we support a project, if we are not successful, we have to give a very good reasons why you are not successful. And 60% of the time, I have data with me in my organization that 60% of the time, if they agree what we have told to them that these are the gaps, they are successful in the next round. I think we have to be when you don't get fund, don't get dejected. Take the criticism positively, put that back into your next thing. Clear it might be a lack of scientific data. Many of us might be representative of the projects, and you know that. And I think the success rate increases as you move around in the next line. You know, if any funding agency I talk about, there is a 10% success rate in any of the funding calls we bring out, 10% success rate. So I think that 10% success rate, you are there, that is what I think. But the major thing is you learn from each round you make. You have to learn, improvise it, and come back. Success has to touch you one way or the other.
So is there any specific question on this particular topic about startup funding or entrepreneurship specifically? Yeah, please.
Hello, I'm Subhanshi. I'm a PhD graduate here. So similarly, like students or researchers, we do a lot of research here and may develop some products as well. But in order to uplift it to an industry scale as startups or something, so what you guys actually looking for such kind of products to invest in? Like what are the parameters that you guys looking to invest into a new startup or something?
You're asking about the product or the startup?
The startup or the product line is. So what are the investment criteria that you guys look for? Yes, sir.
Yeah.
What funder looks at?
Yeah, maybe I'll take a look.
Okay, you can.
So, see, the first thing we look at is the team. Okay, let me because team is the one which actually has to take the startup forward, right? At least, okay, in software startups, we also pivot. We change things, right? As we hear from in deep research, that doesn't happen often, but still things can change, right? Maybe we have one molecule, then maybe like you will figure out, okay, that can be applied to some other problem also. You may pivot things like that, right? So team is the first thing. So the two, when it comes to deep science-based startups like yours, what have you done? Like how much research you have done? Where is the what is the current stage of the research, right? And what is the problem you're trying to solve? So all that becomes very important for VCs. And also, in what time frame can you actually go from one stage to another stage, right? Because we come in very early, and then whatever we give will not be enough for you to take it to the end, right? So because of that, we need to understand with the money the investor is giving, what stage can you reach so that other investors come and invest in you. So that's the only path, right? Those things we look at. Yeah.
So yeah, so you were talking about the team. So like as in scientists, we know just to develop something. So as in a company, the team should have different kind of domains.
Yeah.
How can we build that?
Yeah. So it will be, and you also need those people at different stages of the journey, right? So first research is very important. And actually, let's assume like after the research is done, you have to do a clinical trial. You have to get a lot of people right around you. So how to do that? Finally, you need to get somebody who can go and actually do business development, right? You can go to the molecule and go and talk to pharma companies and all that, right? So you have to build a team as you go along. So initially, research is very important, and as you go along, all the other things come in picture.
I will give you a very simple answer. As a funder, and I'll give you an analogical answer. Have you heard a fairy tale? Fairy tales, it always starts with a once upon a time, the first line, and the last line is always happily lived ever after. So when we used to, our parents would tell us when we used to read, we were pushed to read the next line of the story. So when you write a grant application, when you go to a funder, how compelling story you tell. Story in the sense your science has to be strong. Without science being strong, 60 to 70% is your science. It has to be strong. 30-35% is your planning. As sir said, your team. If there is a regulatory thing involved, your regulatory thinking, your IP thinking, your exit strategy, what will you do after 3 years of funding? That doesn't mean that at the end of 3 years you have a product in the shelf. No. But your strategy, your thinking, you have to have 360-degree thinking about your program. And that is what I'm telling you about the story. You have to build the story within yourself. And that is what, I can tell you a number of examples from my funding pot that these stories literally help you. Science strong unmet need of that science. It has to be a need. The science needs converts into a need. As said, it is, either you start from the bedside to the bench or bench to the bedside. That has to be a need. So I think about those lines. And if you have a compelling story with good science, I think you are there. Am I right?
Yeah.
So the next question. Yeah, please.
Yeah, so hello everyone. Myself, Ashwin, student. So I wanted to ask that we as early career researchers or as a PhD student, how can we best place ourselves in any industry? And do industries have any direct recruitment process or placement drives for PhD students? And we know sizing what to actually in the interviews or CVs. Thank you.
So, hello, am I audible? Okay. So, at Panacea, we do take what we call, at Panacea, we take what we call scientific officers. So, essentially, we take for our production sites where we do our manufacturing, people with master's level qualification minimum. We hire 30, 60 every year. We rotate them through different functions. And then after 6 months or a year, they have an option to be absorbed or they have industrial training. So their marketability already has gone up and they can go find job elsewhere because they now have industry experience. In R&D, we look for people who have done PhD. So that is preferable. And we do hire directly. But as with everything else, what matters is what is your skill level, what is your competence? Because whenever we open a position, if we have one position, we receive maybe, let's say, 3 or 4 applications. What we do is we evaluate individuals based on their personality, their scientific acumen, their
suitability to the job that we have, and we just pick individuals. So the short answer is yes, there is a direct route to entering industry after either your master's or PhD. And I gave example of my company because we are based in Delhi and Chandigarh in this area. So it's very relevant.
So Prabhita, if you can take up, or maybe Dr. Kaul.
Maybe just a quick thing, because there are a lot of students. I think I can tell you every company has a PhD or postdoc program. These days, everybody across all the pharmaceutical industry or the biotech industry recruits PhDs for their postdocs. Like I can give example of J&J campus in Belgium, which is one of the biggest R&D site of Johnson & Johnson. We had almost 150 postdoc fellows, we call them. And as my friend said earlier, out of those 150 fellows which were recruited every year across different domains, like there is neuroscience, there was infectious diseases, there is oncology, different kind of expert, and there is also chemical sciences, manufacturing sciences, formulation sciences. So different kind of PhD profiles were taken in, and those postdoc committees were self-organizing, they were self-driven, they had their own kind of a forum. And the best thing about it that out of those 150 or 200 fellows which come every year, 50% of them are absorbed eventually and given a permanent role. So I think sometimes you will see these days more and more of these opportunities coming on LinkedIn. So you will see student internships after masters, you will see postdoc internships. So keep a watch on those kind of profiles. And if you know somebody who can refer you sometimes internally, because there's a formal referral process which goes into that, that also helps a lot, because somebody who can say, okay, I know this guy, here is the CV. So it's just to go on the recruitment board and put the name of the person out there. So it's not only J&J, it's every big pharma company these days relies heavily on the talent which comes out of the universities. It's very commonly known, especially doing the early exploratory biology or early exploratory science is what we call.
Hi. I will take permission to be a little blunt. Science is not for the faint-hearted and it's not easy. So when you come to industry or you come anywhere, even in academics, if you think you want to contribute and there is competition, come prepared. So preparation means learn your science, know your techniques, and understand the philosophy of the industry. This is where we see the biggest disconnect. Just because you have done a gene or a protein doesn't mean that it means anything in this world at the moment, till it has a place in the universe. You have added to knowledge and which you shall be recognized for. But when you sit for an interview, please come prepared. Look at the website. I mean, I must have in the last 25 years taken thousands of interviews. I mean, now my HR doesn't want me to sit in interviews because they say select people. And I ask very basic questions. Can you define pH? You can't imagine how many people fall on the definition of pH. And you want to be a protein scientist, you want to contribute to protein sciences. If I mix pH 3 and pH 5, I'll get pH 4. Statistically, that is the 86% answer I get. How can we recruit you? The right side is the journey you run in academics is the same journey you run in the industry. The techniques are absolutely the same. So what are we really looking for? We're looking for a person, if he or she has done a PhD, ability to look at a problem, make some initial analysis on it, and come up with probability solutions. Maybe right, maybe wrong. If you can do that in an interview table, I think you have a fairly strong chance at getting in. Most of us know you will not be able to perhaps solve the problem because you're new. And if I'm not mistaken, I think all of us have come from academics. All of us have come from academics. So there's no difference. We still have 2 legs, 2 arms, and a little bit of brain left. So there's not much of a difference. The difference is in the thought process. And maybe I'll cover it a little later, which I'm really trying to study, is ideas are not enough. Operational excellence is the most important thing, but I can cover that later on.
So, in the same note, if Anu can shed some light on how to get into, other than R&D, the other domains of industry, if any student is interested in that.
Thank you for that. So, hello everyone. I'm Anupal representing USP here. I agree with you what you said. Understand the industry philosophy, right? That's an important aspect because you know, when you are doing PhD, you are more focused around your research work, your thesis. But when you go to industry, the requirements are totally different. And that's where we have seen that there is a gap. And I am representing USP, United States Pharmacopoeia. So this is a standard setting organization who sets the standards for industry to follow. Like how would you maintain consistency and quality in your product, right? So now in biologics, that's the gap that in pharmaceutical, there is a course on pharmacopoeia, so they know. But in biologics, nothing like that. So that's where the awareness about pharmacopoeia is not there. So when you come to industry, then only you will realize, oh, there is a book, there is a pharmacopoeia which needs to be complied to. So, and that's where it is important that, you know, we bring in awareness about pharmacopoeias. And
is there kind of any internship or anything who wants to, you know, change back to the regulatory domain?
Yeah, we do that. And also we are, I mean, we do hire interns from like different, but from pharmaceutical background. So that's where probably something for us also to think about that in biologics, what we can do really about it. But what we are trying to do is to collaborate with, like we have recently collaborated with DBT Biotech on how can we build awareness around. So we are planning to do like a series of webinars where we will talk about different resources which are available. And trust me, like many of these resources are complementary available. So what are the best practices, informational guidances which can be found on USP website. So that's one aspect. Other thing what I also wanted to mention is mainly for PhD students. The very obvious thing is to go into R&D. So they don't know about that there are QC departments, there are QA departments, regulatory. So their horizon is huge. So do explore, be prepared on when you are looking for going into the industry.
I think I can add something here. So Indian Immunologicals, we are only one health company in Asia Pacific having a strong 50% presence in animal health and human health. I interview almost every day one candidate, mostly from master degree, PhD to diplomas. I'm writing a book on what did I learn by interviewing millennials. So the bigger issue that I see is unable to extrapolate the knowledge into an allied area or a different area. I have hired plant molecular biologists developing clones for cellular therapies. I have hired BA in performing arts doing column packing in chromatography in purification suites. The question is when we talk to people, is the person has the knowledge that can be extrapolated. That is one thing that is very, very critical. It's not just the domain knowledge. Many times people say, explain yourself. They say, I'm a plant scientist. That means the person has already boxed themselves in a very limited, narrow scope. And that is one area. The other thing is what she mentioned, understanding of regulatory. And that is it is extremely important when you are developing biologics or working on infectious disease or immunology or on allied areas or molecular biology, understanding the regulatory landscape and importance is in industry, time and money is the essence. It cannot be just continue perennially doing exploratory research. There is a timeline to delivering certain things and a budget to it. and it has to be worked. The other thing also very, very important, many times I see people coming for interviews, they don't know themselves. So they don't know who they are, what's their personality and choices. Maybe you are doing a research where your blend of mind is you would be fantastic doing selling of something related to biologics. You are not a compliance oriented personality and you are not interested in accuracy to the 10th decimal, but you want to go into QC or an R&D, you will struggle. So very, very important to know who you are, your personal preferences, and then choose a career path, not just because it is very sexy in the market and everybody is going, so I should get into. You will enjoy that initial 1 or 2 years, then eventually you will struggle. And I have seen that many people struggle at the entry level because they don't know themselves very well.
So maybe I can add to what he said. I think regulatory is a very critical part of the entire process. And I don't think scientists at doing the PhD are really exposed to this whole concept of what is called as regulatory science. It's not biological sciences, it's not just clinical, it's actually a movie kind of film on regulatory science. So I think that's important for perhaps for THS to get an exposure to what regulatory science is all about, especially if you're looking at industry. The other thing we have seen is while scientists typically get into R&D or toxicology or preclinical teams, we've also seen them making transition to more of the clinical side of things. So finally, when the product is going into clinical trials, I think somebody who has that bent of mind of curiosity, bent of mind of designing a study can transition to actually clinical trials and build clinical protocols. There are guidances that are available that you can build. So we've seen that quite successfully. A few people actually making the transition now from basic sciences to more clinical sciences and even to medical affairs, which goes into medical education of doctors. So you can really, and somebody with a PhD background can actually make a very strong pitch. As they say, scientists love their hypothesis. So they're the best salesmen actually. So many people I have seen from PhD actually doing a very good job on the med affairs side because they're able to really sell the story of the hypothesis for a drug and the molecular mechanisms to the clinicians who really don't understand basic sciences. So I think there are different opportunities. And of course, I'm not talking in the commercial part and the other part of things right now.
Actually, just to say that I'm a PhD postdoc and moved to scientific affairs, which is equivalent to kind of medical affairs.
I think that was a very good broad spectrum insight. We captured almost everything history is about. And I think what the students, while they're doing a PhD, don't know, they kind of learn on the way. And while maybe doing their first job or the second job, and then people figure it out. But figuring out that who you are, and then based on your preferences and your personality, choosing the right domain is probably the most important thing.
And just want to add before we close, as was said by our earlier speaker, it's not that you have to do always in the bench in the science. There are Manish is sitting here, I am sitting here. There are a lot of people like who have done, I have done my PhD, then I did my LLB, then I did my management from said business school, and then did my LLM. And then we have IP, I have been an IP patent agent also. So, and then we are managing program management. So there are various options available into the system, and you have to, do your best strengths analysis and, take your chance. But science has to be always strong. You have to understand science holistically before moving into any of the directions you move in.
Other than R&D, all these other domains, deep and sound science. That's what the basis is. Next question. Yeah, please.
Good afternoon. Myself Gagandeep Singh. I am a PhD student. Actually, sir, I want to ask.
A little louder, please.
I want to ask, during PhD, most of the researchers focus upon papers. So, but how does one evaluate it for industry? If I were to say, as you mentioned, that you judge during interview, but most of the scientific students, they are good in their work, but not so does not have those intellectual skills to be able to do better in interview. So how would you judge those things?
Again, when you say intellectual skills, can you, I mean, I'm a little lost, but I was curious to answer your question. What do you mean by other intellectual skills?
Like, sir, being prompt enough to answer at that time. For example, one know how to do scientific communication skills. Yes, sir.
I can't speak for most of us. We're in the business of science, not in the science of business. Okay? We don't need editors. They are very easy to find. We are really looking for scientists who can unravel a problem. Now, when we ask you deep scientific questions, or at least unravel, and another thing is, if you see walking on the road, you never fall on a big rock. You always fall on the small chips. So one of the things we have trained during our interview process, we look at 4 things: your technical knowledge, okay, your personality as a person of perseverance. What happens if you fail? What do? You run away? So those things are asked. Then the third aspect of you as a person, which we try and look at, is you've spent 5, 8, whatever number of years pursuing a topic. Do you have certain amount of mastery in it? You really know what you did. Which means tomorrow, if you put you onto another subject, you would apply the same rules of diligence, effort, trying to go deep, finding out solutions. You know those things. And last and not the least would come your ability to communicate. So as long as you fulfill the first few process parameters of what we do in evaluation, if a person does not have a failure story in their life, I don't think we'll recruit them. We are all failing, perhaps daily. So if you can't speak honestly and with integrity, those are things we keep a very strong lookout. And the way you are representing data, are you it's your PhD. What do you do between 2 and 3? What's the accuracy of your data? Are you able to communicate that accurately? Becomes very, very important for us. So it's not difficult, but it's very basic. I hope I've been able to help you.
Yes, sir.
Thank you.
Yeah, please. I probably will take just one more question because we have to start the student pitching session. Yeah, please.
Good afternoon, everyone. I am a teacher in a pharmacy school, Jamia Hamdard, you must be knowing. Some of you must be knowing. And also representing Indian Pharmaceutical Association here. I have one basic question. Since being academician, I often face this. Being all industry people and financiers, most of I could see. And I have one small question that how in Indian academia research, which I primarily am into pharmaceutical small molecules, be better aligned with the needs of export-driven pharmaceutical manufacturing and global market access. Since we are right now into like a global thing, like we believe that India is the pharmacy of globe world. So how will you like help us as an academia to go into it? And one more thing is that the PLI scheme, how you can see as an industry expert, we academician pitching into it, and maybe we can be of any help or do anything with that respect. Thank you.
I think I can address the first part of your question. The question is, how do you, I think if I understood correctly, you were asking, being in the academia, how do we bring the industry element into the teaching process and making them translated to students? I think I myself is an adjunct faculty with 3 different universities. I'm every alternate month or same month in multiple places, going and addressing students to make them understand what industry needs. I think every academia need to have that kind of platform or a forum where they will bring in industry to address to the students that gives them a understanding of a lay of land, because it's very difficult to capture that in a book. It's very difficult to give it in a capsule. Multiple sessions of discussions, then the whole landscape opens up. There is QA, QC, as simple as somebody writing a pack insert that gets into a vaccine box or a monoclonal antibody box that has so much of technical details, and there are regulatory frameworks into it. There are a lot where science can be utilized. And even from our product management side or in business development side, there are people with science background who are doing those things. But many times it's not just the research. Research maybe is just the 5%. Of my early 15 years of my career was purely an academician, managed PhD students, postdocs, and more than 100 publications. But then next 15 years transition completely into industries, have patents and develop products and developed 18 vaccines that are into different phases of clinical development. So the question is that somebody who can make that transition, who understand both the sides, then it makes it easy to connect to somebody in the student side to help them understand the industry aspect. And having academia bringing in industry to address the audience, the more frequently it happens, that brings an exposure.
See, always academia has gone to, industry has gone to academia as adjunct faculty and all that. Actually, I want to see when actually academia also goes and spends time in the industry, like during the vacation or whatever.
Yeah. So I have in fact the other question. Can industry spend 1 month in academia?
Yeah.
Because internships on the other side are quite common, but I've had several instances where I find if somebody who's moved into our team, for whatever reasons, they're rotating, they don't understand some things, where can I send them for basic academic training? Like I was commenting on PKPD here. Yeah. Can I send someone somewhere for a basic academic training, maybe for a month or 2 months?
Actually, some European countries, there are methods.
Yeah.
Probably should start doing that, visiting professors from the industry.
For example, we as a company, we are very interested in AMR. But if I want someone to really understand clinical microbiology,
right,
I can't always hire a microbiologist in my team. Right? But if I have a pharmacist who understands the drugs part of it, but I want him to understand clinical microbiology, where can I send him to learn? I can't do it in Cipla. Can I send him to Hinduja Hospital in Mumbai to learn clinical microbiology? So if we also set up these kinds of things, can I send him to a public hospital to learn?
So maybe Synchn can be another platform.
That can be another thing for THSTI to consider, to bring industry in people, to intern them, and vice versa.
So in the interest of time, I'm really sorry, we cannot take more questions. So I'll thank all the panelists to patiently have answered your questions. And I hope we have a lot more questions. They are very widely available for, your queries. Thank you so much. Thank you so much. Yeah.
The last fact is that we in industry thinks we know it all, but we don't know it all. Yes, we know. So we granted that there is more competence in the industry, but I find incompetence also in the industry. So we're all happy. What about the other side of the table where you are in now? Do we think the same?
सरकार के प्रमुख वैज्ञानिक सलाहकार प्रोफेसर अजय कुमार सूद जी के प्रति आभार व्यक्त करते हैं कि वह आज व्यक्तिगत रूप से उपस्थित न हो सके, फिर भी उन्होंने अपना संदेश भेजकर इस आयोजन की गवाही दी है। आइए हम सब उनका भेजा हुआ वीडियो संदेश सुनते हैं।
In industry-academia partnerships are a cornerstone of modern innovation, merging the fundamental depth of academic research with the practical expertise of industry to foster progress across science, technology, and the economy. These collaborations not only accelerate journey from lab to market, but also equip students and researchers with vital exposure to real-world industrial practices. Together, universities and companies build dynamic ecosystems capable of tackling pressing challenges, creating employment, and driving sustainable development. Globally, successful models have shown how sharing ideas, talent, and resources across institutional boundaries fuels breakthrough innovations. This has redefined national competitiveness and established thought leaders in the global knowledge economy. With nearly 9,000 biotechs startups as of 2024, as compared to 50 in 2014, the country has witnessed an extraordinary growth in just a decade. These dynamic startups are leading the charge in health tech, diagnostics, agriculture biotechnology, already developing over 800 products. India's bioeconomy is projected to reach 300 billion US dollars by 2030, and various initiatives of our government, including BioE3 policy, which stands for Empower, Enable, and Encourage Bioeconomy. These efforts are helping boost innovation that is sustainable, create jobs, and bring together researchers and industries to develop bio-based solutions that can compete globally. Moreover, biomedical innovation today is no longer confined to healthcare. It intersects with sectors like agriculture, environmental science, digital technology, and aerospace. This growing convergence is fueling interdisciplinary progress, where developments in one field spark transformative advances in others. India's expanding footprint in space science exemplifies this potential. The integration of biomedical research with aerospace technology, especially in emerging fields like space medicine, bioastronautics, and space manufacturing, is opening new frontiers for discovery and commercialization. A prime example is the recent collaborative initiative between the Department of Biotechnology, DBT, and the Indian Space Research Organization, ISRO, which jointly led 7 pioneering microgravity experiments during the Axiom 4 mission piloted by our group captain Subhanshu Shukla. These studies will investigate a wide range of biological phenomena in space, including muscle regeneration, edible microalgae cultivation, cyanobacteria for nutrient recycling, seed sprouting for space nutrition, tardigrade reproduction, cerebral hemodynamics, and human interaction with electronic displays. These groundbreaking studies hold promise for both astronaut health and space-based innovations, marking a significant step towards India's vision of building a cutting-edge microgravity research ecosystem and preparing for its planned space station by 2035. I'm happy to see a strong momentum being carried forward from Synchn 2024, which resulted in 14 meaningful agreements and letters of intent with industry partners. This event brings together the scientists, entrepreneurs, educators, and policymakers, setting the stage for purposeful dialogue and impactful collaboration. Academic institutions should focus on nurturing foundational research and training talent, while industries can guide commercialization pathways, provide funding, and scale promising technologies. At the heart of this collaboration lies the shared mission to drive India's bio-innovation forward. Academic institutions play a crucial role by deepening research capabilities, nurturing the next generation of innovators, and fostering an environment of intellectual curiosity. At the same time, industry partners bring the ability to translate discoveries into market-ready infusion funding and mentorship and amplify successful technologies at scale. India now stands at an exciting crossroad where strategic strengths, government support, and global collaboration converge. By deepening industry-academia partnerships, the country can develop robust technologies that solve real problems. The time for transformative change is now, and India has both the vision and the capability to lead the way. Thank you.
प्रोफेसर अजय कुमार सूद जी के प्रेरणादायक संदेश हेतु हम उनके प्रति हार्दिक आभार व्यक्त करते हैं। धन्यवाद। Forward to the most exciting and most awaited session of this event, pitching to the investors. We request that all the students, PhDs, and postdocs to be present here to assist our guest for the breakout session, which is again a parallel session, where the respective So the arrangements have been made for the thematic discussions across designated meeting groups. All delegates and business partners are requested to meet the investigators. For clinical research, you may please go to first floor meeting. For vaccines, you may please proceed to the boardroom. For diagnostics, second floor meeting room, and for the other facilities at THSTI and the cluster, you may please proceed to the meeting room and their respective facilities. Please note, post this breakout session, the high tea which will be after pitching to the investor session. Industry representatives are requested to process for the feedback for how they can be associated with THSTI, particularly with respect to upcoming translational research facility, and see for their feedback. Thank you so much.
Good afternoon, everyone. I hope you have a wonderful day so far. I am Poonam Bishnani, Officer Equity Schemes at BIREC. Now I am here with you for the session. We are going to start the startup pitching session here. It gives me a pleasure to welcome you all for the session. This is a deep tech innovations pitching session being organized to provide the platform for the innovators. Today we have a curated PhD scholars, postdoc researchers, entrepreneurs, early stage startups who are going to present here in front of panel of investors. We also have a honor to panel of investors across biotech and venture capital domain. May I kindly request the investors to shift to the front row, please. Investors. Kindly please shift to the front row. Thank you so much. The session is being coordinated by BIREC and IVCA. IVCA is a Indian Venture and Alternate Capital Association. To open the session, I would now like to invite Mr. Amit Pandey, lead at IVCA, to deliver the opening remarks.
Oh, okay. Thank you very much, Poonam. Thank you very much to the BIRAC team, the Department of Biotechnology. Hello, everyone. Good afternoon. My name is Amit. I'm with IVCA, the Indian Venture Capital Association. We are an industry body for the venture capital and private equity investors. And I manage the VC ecosystem within IVCA. Happy to be here. So I'm glad to share that we've been working with BIRAC for quite some time now, and the intent has been always to make sure that the talented startups and the PhD scholars and scientists do have an access to funding, and this is a good kickstart. Again, thanks to the BIRAC team, thanks to Dr. Ivan for giving us this opportunity here, I'm glad that we have a decent number of investors that have come in. In fact, not just from Delhi, but from different other parts of the country. So it is very heartwarming to see the interest that we have received from the investors. And now we're looking forward to the startups to come and pitch. The only piece of advice would be, see, these are bankers, and for them, time is everything. So please stick to the 5-minute timeline and try to complete your pitch. And just look at it as an experience. And definitely the conversation can go offline as well. The front row is packed with investors. We see from IDESpring and IVCAP and Stride and a bunch of other investors also in the room. So, and all the best to all of the startups pitching today and the investors. Thank you, everyone.
Thank you, Amit. Now I would like to request each investors to introduce yourself and about the investment thesis, but very briefly, kindly.
Surya Prakash, you can call me Suri, from IDESpring Capital. We're based in Bangalore, based in early stage, deep tech, deep science-based startups. In life science and biotech too. Yeah.
Thank you, sir.
Hi, good evening, everybody. My name is Dr. Vishal. I represent an organization called BioX Venture Advisors. Our investment thesis is from early stage to late stage. But when it comes to early stage, we are very keen on evaluating opportunities in the area of digital health. Then definitely, genomics is another segment that we're very keen on. So I think these are 2-3 sectors at early stage which resonate, whereas for our late stage, pretty much everything related to pharma, biotechnology, stem cell, molecular diagnostics, anything and everything we have evaluated, and we've also been fortunate to be a part of 40 plus companies in deep tech sector. Thank you very much for the opportunity, Dr. Divan. Yeah.
Thank you, sir.
Hello, everyone. I'm Dr. Rishika Agarwal, a venture partner at Colossa Ventures. We invest in early stage, mostly deep tech, but also some sector agnostic startups. But our key point is we invest in awesome startups with awesome female entrepreneurs.
Thank you.
Good afternoon, everyone. I'm Abhishek. I'm part of the investments team at IN Group. We invest right from pre-seed to Series A opportunities, very much focused on life sciences, biotech, healthcare, pharma. We also have a categorically structured program for focusing on life science and biotech companies under Bio Angels, which was set up in partnership with Biotech 2 years ago. So we are right from pre-seed, we are going as high as Series A opportunities and supporting across multiple stages. I'm looking forward to interacting with you today. Thank you.
Thank you so much.
Hello, everyone. Good afternoon. This is Rahul Gupta. I represent IVK Venture here. We are a venture capital fund, invest from early stage till Series C. We are managing around 6,000 crore of AUM. So looking forward from you guys. Thank you so much.
Thank you, sir.
Hi guys, I'm Devansh from Local Capital, early stage investors. So $500,000 to $1 million is the first check that we put in. Biotech is definitely a space that we're very interested in. And yeah, we're investing with a fund. This is all into a recruitment fund. So we're definitely excited to see the entrepreneurs that are innovating in this space. Thank you.
Hi everyone, I'm Jap Shabat. You can call me Jap. I'm part of the investments team at THSTI Ventures. We're a sector and stage agnostic fund. We're managing approximately $7 million in AUM. We start as a pre-seed seed level. And happy to meet you all and interact more.
Hi, good afternoon, everyone. I'm Akshat again from Stride Ventures. As Jap has already mentioned, we have about 180 portfolio companies. 18 of them are currently unicorn. We invest across the segment and also developing a keen thesis in deep tech, clean tech, as well as other avenues of focus. Thank you.
Thank you. Anyone investors left? Thank you so much. Let's now begin with the presentation today. Each presentations will get 3 minutes, followed by a brief interaction with the investors. I kindly request everyone to adhere to the strict timelines. Our first presenter will be Sneha Bajpai.
Thank you. Good evening, everyone. And my name is Sneha Bajpai, and I'm a third-year PhD student under the care of Dr. Krishna Mohan Atmakuri. And as Amit mentioned, I am please don't expect that it's going to be one of the conventional pitches that you get in your startups, but I've just tried to put in effort. So when I started with my PhD, I was not aware that infections in neonates is such a big concern. You can see here that over 5 million neonates, they die per annum, and that's a very huge number when you look at it. And especially when you are talking about LMICs, over 6.9 million episodes of possibly severe infections are reported only from LMICs. So the problem, the major risk factor is actually the neonate itself. And I'm not saying that the neonatal immunity per se is very poor, but their immune system is trained in such a way, it's engineered in such a way that it's more tolerance biased. And there are reasons to it, reasons to that. So why is management of neonatal infections so difficult? So one of the points of exposure to the pathogen that the fetus experiences is while it's still in the womb, and that is through the ascending infection. So the genitourinary tract hosts pathogenic bacteria as well in some cases in the mothers, and this can ascend to the sterile embryon and thereby contaminate it and expose the fetus directly to the infection, which causes a cytokine storm and can eventually lead to premature birth as well. Following that, the third exposure takes place when the baby is born, like at the time of delivery through the vaginal route. It's again exposed to the genitourinary pathogens, and the fourth and the final exposure takes place where it is exposed to the nosocomial pathogen reservoirs. So in some cases, the neonate is brought into the neonatal care facility, which we have seen has many pathogenic hotspots. So the question is who to blame. So as I mentioned here, that most of the neonatal infections are actually from bacterial origin. And amongst these as well, in India especially, Klebsiella pneumoniae is one of the most notorious pathogens, which has been isolated from many sepsis positive cases, neonates, sorry. So the next question comes that if this is a problem in clinical settings, there must be a solution, there must be a treatment. So in the West, they had a problem of GBS, group B streptococci, which is a gram-positive. And to manage it, the clinicians, they used to give IAP, that is intrapartum antibiotic prophylaxis to the mother. That is, as soon as the mother is induced for labor, she will be given a bunch of antibiotics to reduce the burden in the genitourinary tract. But as I was telling you, that the exposure, the possible exposure could have happened when way before the postpartum stress even began. So next, the next question arises that if not antibiotics, because of the AMR as well, what is one of the most secure and assured strategy that one can take up? So as I was mentioning about GBS, there is already a vaccine which Minerva has come up with, and it's in the third phase of clinical trials in Denmark as well. That it goes by the name GBS-NN, and it's targeted towards pregnant women. So what happens is that as soon as you can see here, as soon as the first trimester is about to end, the maternal transfer of immunity through antibodies begins, and it reaches at its peak by the end of the third trimester. So if the plan is that if we prime the mother with prime or boost the mother with a vaccine that protects them against these escape pathogens, then possibly we can reduce the burden of infections in the neonates. And surprisingly, a very recent study also showed that if you boost the mother during pregnancy, the kind of antibodies which are generated against the antigen are way more efficacious because they have pregnancy-induced PTMs, post-translational modifications. So it's not that maternal immunization is new in India. We already have a bunch of recommended vaccines, preconception and during pregnancy, which are recommended to women, especially Tdap if you see here. And even during we saw that clinicians and pregnant women, they had a great vaccine confidence. So it's not that India is, like you would be surprised to know that as compared to the Western countries, India has more vaccine confidence and less hesitancy. So that creates a good market as well. So again, where are the vaccines for the escapes? As you can see here, that against Klebsiella pneumoniae, you just have one vaccine which is under clinical trial. So, and this is one of the, I don't understand economics. I'm so sorry, but I have gathered this information for you, wherein it is estimated that with a full rollout of full implementation and rollout of the GBS-NN vaccine that I was telling you about, the initial investment will be around $1.7 billion, and the returns or the benefit that you could expect can range from 1.1 to $17 billion. And this is just the cost which will be averted from healthcare management of neonatal sepsis.
Kindly conclude your slides.
Yeah, sorry. So I'll quickly walk you through this. The plan that I have in my lab or my PhD, what I'm working for is that I have 3 candidates against Klebsiella pneumoniae, which I have characterized, and we will be patenting them soon. And we have partnered with an industry to acquire few FDA-approved adjuvants, which we will be testing in our preclinical model for sepsis, for monomicrobial sepsis. And eventually, once we are done with our efficacy studies, we will try using these, we'll try testing these vaccine candidates in pregnancy model in pregnancy mice model and also in neonatal pups. So briefly to walk you through what our monomicrobial sepsis model looks like, this is standardized in females. What we have done here is we have inoculated the pathogen in the interperitoneum cavity, and we have assessed all the parameters that are assessed in a human patient in a clinical setup. All the data regarding this study, I have a poster set up in the foyer, so you can visit afterwards as well. So I can show you the data as well. And we have really convincing results which show that the monomicrobial model actually resembles the clinical events which occur in the human sepsis as well. Now, along with this, I've already tested my first candidate with an adjuvant, and again, this is the immunization scheme that I have standardized in lab for the murine model. And what we see is that we get pretty convincing results. Like when we give a lethal challenge, that is a challenge which is sufficient to kill all the mice under 40 hours, we get 100% protection with the candidate one. And when we increase this challenge dose 10x, that is 10 times more, we still get a 50% protection in the group that received our vaccine. And sham here is just adjuvant only. So I would like to mention that this is the PBS group. This is the negative control, where you can see that all the mice are dying within 40 hours of infection. So finally, to summarize, we have developed in our lab a reproducible model for monomicrobial sepsis, and the model is compatible with immunization workflows that we have standardized. And we have also standardized the downflow assays which are required to, check the correlates of protection after immunization. And finally, the work that I am currently going through is that I'm screening all the 3 candidates with 3 adjuvants. I'm making them into 9 combinations, and I'm evaluating them one by one, so as to identify which of them is the best for taking into the pregnancy model. And also, I will, as I mentioned, that I will be assessing the protection in pregnancy model of sepsis, and also in the neonatal pups, to see if there is any intergenerational tolerance developed or not. So with that, I would like to thank my lab, THSTI, BRIC, and also DBT for all the support, and this wouldn't have been possible without them. Thank you.
Thank you so much. Let's now have a quick questions, comments from investors.
Thank you so much for the presentation. We didn't get your, ask. What is your ask actually? What is it that you're seeking from here?
As of now, we are looking for, investment in terms of guidance, and a little monetary investment would also help, because a lot of resources go into development of models and assessing their immunological parameters. So per se, we have partnered with the industry for acquiring the adjuvant. So at this stage, because it's a very early preclinical stage, we would appreciate that sorts of an assistance from the investors.
Okay, so in terms of TRL, you are at 2, 3, 4. Where are you?
Yeah.
Just about 3.
I'm so sorry.
Well, I don't think you have a startup, right?
This is not a startup.
So how do you expect us to put money in a in a project? Like, does that happen? Like, I don't know. Startup? Yeah. So SPV, you want us to create an SPV?
So I think just a small correction here to set the expectations. So all the pitches that you will hear, they will be from the students who have invested 3 to 5 years of their career. Yeah. So this is more to, so these are not startups, not formally incorporated as startups. These would be the future startups in way. And also it will also give you an insight from the cluster. So the objective through this pitching session is to expose the forum about the a potential pipeline that is building up. And I think this could be a first primer to all the investor colleagues who have come. But I think as a follow through, you will see company picking up such leads, startups picking up such leads. So it's a very early exposure to all of you.
For future pipeline.
Yes. Yes.
Yeah, I've just given you a hint that there is a gap that needs to be filled. And from there on, obviously, I wouldn't want you to take up without me. But yeah, that's as Dr. Diwan said, that this was just the work that I am doing in the lab. And it's a potential research gap and which needs to be addressed. So yeah, thank you so much.
Thank you so much. Next, we have Manya Sahu, a PhD scholar. You have 4 minutes for the presentation.
Good evening, everyone. I'm Manya, and today I'm going to talk about cervical cancer. Did you all know that it takes cervical cancer almost 20 years to grow and only 20 minutes to detect? Yes, this diagnostic test has been around for almost 80 years. So why are we losing 1 woman every 8 minutes to cervical cancer in India? Because this test is inaccessible, invasive, and not built for them. Today, with FemScreen, we're changing that with the everyday use pad that turns into early detection. Cervical cancer claims over 70,000 lives each year. And moreover, these cases are diagnosed at a much later stage where the survival rate is only 16%. This leads to an estimated 25,000 crores in economic losses due to lost productivity and treatment costs. And the overall market size being around 200 million women in our target group of urban women aged 25 to 60 years old. Cervical cancer takes almost 10 to 20 years to grow, and if caught early, 90% of the cases are preventable. Despite WHO's periodic screening after the age of 25 years, in India, less than 2% of women have ever been screened for cervical cancer. So why aren't women screened? Because the current tests were never built for dignity or comfort. HPV tests and pap smears require a speculum-based exam, which is painful and often stigmatized. And you know what's surprising? It's not even available to unmarried women in most settings in India. Even newer products like Cervicheck and MolBio still require vaginal insertion and only detect transient HPV infection, which creates fear instead of clarity, as it clears in 80% of women naturally. Hence, there is a need for a user-friendly and accessible screening solution that removes all barriers to early detection. Presenting FemScreen, a painless way to screen for cervical cancer using a sanitary pad for sample collection. So the study that we did with PGIM at Chandigarh has resulted in biomarker identification in vaginal fluid, which was checked in 3 groups: precancerous, early stage cancer, and healthy women. Now, based on this data, we've developed a prototype to passively collect vaginal fluid in women. So here's how the test works. First, a woman uses a sanitary liner as part of her normal routine. While she wears it, the sponge collects the vaginal fluid over a few hours without any special steps or pain. The sponge is then transferred into a cassette, which is then dispatched to the lab. In the lab, the vaginal fluid is analyzed for precancerous biomarkers and screened for cervical cancer. Finally, the woman receives her report digitally with clear guidance on whether she needs a follow-up consultation or not. This workflow is designed to be private, simple, and empowering. So unlike other products, FemScreen is completely non-invasive and accessible to all demographics of women. It requires no speculum, no insertion, and no special training. Unlike other products that only detect HPV transient infection, FemScreen identifies precancerous biomarkers, which enable accurate identification of cervical cancer at an early stage.
Our go-to-market plan is reaching our primary target audience of young and unmarried women through website, e-commerce, social media marketing, and partnering with home healthcare companies and diagnostic centers. Our business development options include either working with an established healthcare firm or starting our own venture. Lastly, I would like to say that FemScreen isn't just a product, it's a statement that early detection should be effortless and built around a woman's life, not her fear. And it's time to end the cycle where preventable cancers go unseen before it's too late. Thank you.
Thank you so much. Any comments?
How many samples you have tested?
We've checked in 200 per group. So we checked in 3 groups, so 200 samples each group.
Is your technology ready to deploy? Because one of our portfolio companies, which is called SevaMo, they do lot of primary screening work, right? So we would love to see that if your solution could be adopted by them and they can offer this as an integrated part of their package for doing a lot of work around CSR, right? They do screening for CSR. So I would love to do a deep dive with you on the technology and of course the go-to-market. Thank you. Good presentation.
Thank you.
Thank you so much. Next, we have a startups called Easy Pharma Private Limited.
Yeah, next के लिए आप arrow press करो. Okay. Thanks for giving me the opportunity. This is Salil Chawla. I am co-founder My Easy Pharma Private Limited, and our virtual office is in Bangalore. Operational office is in Delhi, and my founder is Mr. Yajur Chawla, who is in RCB only, is pursuing his master's for clinical research, and he has done his undergrad from BITS Pilani. And I did my MBA back in 1988. I am from NMIMS, Tier 1 Institute, University of Mumbai. Before we get into the structured exercise, I like to just give a kind of a precursory information, We are in the space of healthcare, which is in the preventive healthcare space of NCDs, diabetes. Everybody is the diabetic capital of India. It is being supported by NDCCC, Dr. Rajiv Chawla ji, who is running a diabetic this and he also happens to be RSSDI ka ex-president. So we enjoy very deep network and ecosystem support from Diabetic Center, from Diabetic Institute for India. And we are in the process of curate a kind of tech app, which is the intervention for patients. And also we have got around already 25 data from our NDCC center only, which we have already trained AI model. We are in the process of creating chatbot by process of giving it to me. And app would be a physical intervention, which will be based on strong data. And the idea is that those who are pre-diabetic or predisposed, before they get into the diabetic drug, as everybody knows, diabetes is not curable; it is only manageable. With this precursor, I like to give. This is basically the web app for diabetes and pre-diabetics, as I told you. And it is basically LCD. And there are the problem statement is that before you fall into this, it gives you an alert. It gives you kind of signal that, look, you are going to get into something which is incurable, So that is the life. Once you get it is very difficult to get out of it. You can get it for lifelong, And prototype, I have process of making. Web app, I have got somebody from Bangalore, she's ex-Emphasis, and she's creating app. I have got 2 interns from IIM Nagpur also. And the target audiences, it is a huge. Time is 14 million, much more than that, because diabetes is there is a very serious study saying that every 7th person is able to get into this is imbalanced lifestyle. Our youth is mentally and side with competition is there are medieval in size 37 but since we come from the ecosystem of diabetes, we have a place under the sun and the right place, to start with. And it is B2C Bible, as I said, interference in patients, and it is subscription. As of now, early stage, we are zero. And we are not in a hurry because we thought if the product is right, value is right, valuation and everything. But at this stage, to have arrive and take it to level next to can make us really, it can help us and for the welfare of the society and welfare for the those who are going to get into the diabetes. And I have already told about myself. I have told about Yajur Chawla. I have told you about Dr. Rajiv Chawla ji, who is the first two are founders. He is our mentor and he is our backbone, supporting us. Thank you so much. Any question? I am open to answer them.
Thank you, sir. Any questions, comments?
Sir, there are many more startups which are addressing this problem, right? And some of them have been able to scale, and a lot of them have not been able to scale. So, do you know anybody that you're going to be competing with? I just want to know, have you done your research on the competition?
Sir, I'd like to in a way say that I dodge this question in the sense that our I, like Arjuna, is at delivering competition as we have to be vigilant, and cannot take our eyes off it. But since I have already 2,500 real data that to coming from an active diabetic center, I don't think many have that capability, Based on that 2,500 ka strong data are that AI is going to interpret. Once the patient is going to enter the vitals, around 23 of them, he'll get exactly a personalized kind of this, that where are you? And this AI heart, we have got 3 colors. If it is normal, it is green color. If it is yellow card, that means you are pre-diabetic or predisposed. And if it is red, that means you are entering into that territory. So our biggest, this is ex-president of RSSDI, you understand India is the diabetic capital, and he president of it, twice he was president. And he is one who is supporting us. So we feel that our biggest strength is because we have got the ground under our feet, Rest, it is, as you know, startup journey is in the God's hand. If
but when it is startup, it is like, you have to also look at the external environment. And we have tracked many such diabetic companies which are trying to solve the big problem of diabetes. Everybody knows TAM is big, but it's very difficult to scale, especially the B2C game. So one of our company called Livofy, which came on Shark Tank also, and we had to literally tell them to pivot model from B2C to B2B, because B2C, what we saw, maybe you are able to do it differently, they were burning cash. And before, they got it into B2B model, they were only burning cash. Good number of clients they were acquiring, but that didn't create value. So this is just a wisdom based on our practical experience of supporting startups in healthcare we are sharing with you. We would certainly like you to, do a little bit of deep dive about the competition, because this is absolutely not a new problem. It's an old problem being solved by not many people. But you should really think that what really didn't work work so that you have a jump start. So I just want you to understand and appreciate that point. Rest, I think it's your startup. You know how to use it best. Thank you very much.
Welcome, sir. Still, kind advice. I'm deeply obliged. Thanks for your concerns. And thank you once again for giving the opportunity. Good evening, sir.
Thank you, sir. So next we have Rohit Kachwahan, a PhD scholar. You have 4 minutes for the presentation.
All right. Thank you, ma'am. I'm gonna keep the duration of my presentation short because the outcome of the disease I'm gonna talk about are not. So, all right. Everybody is aware of that pregnant mothers, how they are taken care of because they are the most vulnerable population that we have. Reiterating from the presentation from Dr. Gokhale's talk that if the youth or the population is healthy, the nation would be healthy. Taking notes from that, if we have a healthy gestation, the mother is healthy, the future generation will be healthy, and so the nation will be healthy. Let's talk about during the pregnancy, which is a hypertensive disorder. Dates back to 7 BC. However, the clinical manifestation of the disease has remained the same as such till now. There are many industries which are exploring the diagnostic potential of this disease, onset of the disease. However, the late onset of the disease is still elusive, where the interventions are not available also, which is very detrimental for the mother as well as for the baby. So we are pointing out specifically on the late onset of this disease, which is called as preeclampsia. The paradigm of the disease is not completely understood. So with this notion, we wanted to test what are the molecular markers that are there in the clinical setup. We tested that in the preclinical in vitro model using a placenta-based cell line, where microRNA X, which was the trigger of the disease, which we found out as per the database and in our in-house study as well. When this microRNA is upregulated, which is, let me remind you, is pathogenic to the developing placenta, see levels of these markers listed on the graphs can be seen to your right, where the yellow bar, which is upregulated. These are the clear-cut manifestation of the disease. If this dysregulated, which will cause preeclampsia. Now, the thing is, we wish to explore more on this molecular marker, whether or not this can be used for the early diagnosis of the late onset preeclampsia, where this microRNA can be used as a screening marker with the vision to detect this disease early. We can diagnose it properly with confidence, as well as to provide early interventions which are more powerful. With this, we just want to project that it will ultimately reduce the pathological concern, which will ultimately lead to improved quality of life, thereby reducing the economic burden on the nation. For more information, please feel free to contact Dr. Pallavi Shetrapal, who is my PI at Perinatal Lab of Perinatal Research. Thank you very much.
Thank you.
Actually, I have seen a startup which is much advanced. Okay, the same solving the same problem. Right, that's all for me.
Preeclampsia, I've heard of it.
Yeah, we should talk to them too.
All right, we do have some evidences from published data that we do have a roster of microRNAs which we are testing. So I have taken that. Thank you, sir. Thank you.
So technologically, I think it looks like a good solution, but what about the feasibility from economic perspective? Have you seen that it's something which can actually be used as a sort of a primary tool from a diagnostic to therapeutic perspective? Any thoughts on that?
All right, sir. So in terms of diagnostics, yes. So this can be used for early screening. Some kits available which are extremely expensive. I might not mention them, but some kits are available. However, if this microRNA, if the genes which are already known that is dysregulated because of this microRNA X, we can use this microRNA X as a screening marker. We can reduce, for example, from a number of 100 mothers to at least 20. Then these mothers can tests which are somewhere a little expensive. So the vision is to develop this as a screening marker, which most of the people can afford. All right, sir.
Any other investors?
Yeah, just a quick question. I'm from Alchemy. So one of the markers for preeclampsia is, let's say looking at the albumin content that comes in the urine, right? That is actually a screening tool which is used in the late onset of preeclampsia, and that's quite cheap actually. So what are you trying to replace here? Because there is a solution that exists which is not very expensive in the clinical setting.
Absolutely, ma'am. You're absolutely right. We do have a dysregulation proteinuria in the blood. I agree with that, ma'am. But that doesn't differentiate the mothers whether they are coming from the early onset or the late onset stratification. For early onset, we do have interventions available so that the gestation period can be pushed even towards 37 weeks, which is a normal period of gestation. However, for the late onset, the markers are not available. So maybe this microRNA can work as a PE marker, pan PE screening marker, whereas studies are not that deeply, for the late onset, there's no deep connection has been found. So this can be used specifically for late onset, or if not for that, at least for the pan PE.
Okay, thank you.
All right, ma'am. All right.
Thank you.
Okay. Thank you so much.
So next we have Baljinder Kaur, a PhD scholar. You have 4 minutes for the presentation.
Good evening, everyone. I'm Dr. Baljinder. Have it ever happened to you that you enter in your room and forget where the keys are? That's quite normal. But what if one day you're holding the keys in your hand and you still can't remember what they are used for? That's not just forgetfulness, that's dementia. And by the time these symptom appears, damage has already been done. Presenting CogniTrack, a simple blood test that predict cognitive decline across all type of dementia years before symptom begin. Dementia is a global crisis with around 55 million people living with it and 10 million new cases added every year. In India, its prevalence is around 7.4%. And with increasing life expectancy rate, these numbers are expected to grow. It is one of the fastest growing contributor to disability adjusted life years, providing growing strain on families, healthcare system and economy. Now, this burden is reflected onto market as a total opportunity of around 2,000 crores and a segment of 2,000 crore in early risk prediction. Our niche market being 11 million individuals that are more than 40 years of age and are working in organized sector and under some health coverage scheme. So what are the shortcomings of current diagnostic test? The current diagnostic test focus on the later stage, that is either the second or the third stage, where the brain damage has already been started and is often irreversible. We are shifting that approach by focusing on the pre-symptomatic stage, a stage where the brain is changing silently 5 to 10 years prior to other stages. Now, why we are focusing on the pre-symptomatic stage? Because recent studies have shown that 39% of dementia risk factors are modifiable using non-pharmacological interventions like daily lifestyle changes. So this will provide us with time to delay the progression of dementia, thereby decreasing the cost and increasing productivity. Coming to the competitive landscape, if we look at the current available tests, including the blood test, they detect dementia at later stage. So not focusing on the pre-symptomatic stage. And the biomarkers which they are focusing on has very limited value to predict cognitive decline at the early stage. So the clinical gap being that there is no solution that is accessible, affordable and reliable to detect dementia at pre-symptomatic stage, a stage where action is still possible. So the unmet clinical need being a predictive test to identify cognitive decline for individuals more than 40 years of age. This is how CogniTrack will work.
First, blood sample is collected. Then, it is analyzed using LCMS, focusing on validated biomarker panels. And lastly, a risk score is generated. Once this risk score is generated, it will stratify individual into high, medium, or low risk. And after that, that person will be provided with a tailor-made non-pharmacological intervention to enable further action. So if you look at other competitor, what is the difference? The difference is first, we are predicting cognitive decline at pre-symptomatic stage. And as we are using LCMS as platform, it is easily incorporated into the existing lab infrastructure, thereby making preventive brain health screening more accessible than ever. We'll scale through B2B model using existing LCMS platform at different hospitals and labs. Our go-to-market options are either building a startup or partnering with an established healthcare companies. Our end user being a longevity clinic and in emerging India. At the end, I would like to conclude by saying with CogniTrack, we are shifting the paradigm from late diagnosis to preventive brain health screening to sustain neuroplasticity for India's aging population. Thank you.
Yeah, first of all, that's a very good presentation that's been done. Recently, I've came across by only 40 years and above age.
Yes.
Because recently I've came across a few cases where people have developed this kind of disease with the age group of around 36 to 39 years of age. So why only 40 plus? Why only 40 plus?
So for right now, we are focusing on people above 40 years because as the dementia will start after 50 or 55 years, and the biomarkers which we are focusing on, they predict dementia 10 to 15 years prior to development of symptoms. So roughly 40 years is what we are focusing right now. But what you are talking about, that will come under young onset dementia. So that is a different category, but we will focus on that after later stages. Like once we are done with this, that will be in our pipeline.
Thank you so much.
Thank you.
Hi, this is Alka here from Alchemy. Is similar work happening in other places? Have you tracked the work that's in progress?
Yes, ma'am. As in my competitive landscape, I have mentioned in India only, there is a company that is Urvo Bio that is using exosome-based studies to detect dementia at early stage. But they are also focusing on mild cognitive impairment stage, that is the second stage, just 5 years prior to development of symptoms or dementia.
Thanks.
Yeah.
Hi, this is Vishal. So just want to check, are you looking at technologies outside LCMS? Because if you look at healthcare, from a diagnostic perspective,
yes,
80% of the healthcare facilities do not have any exposure to LCMS. In research lab, yes. In diagnostic lab, it is not there. So I'm not sure about the feasibility of the solution, and that too you're proposing you're going to do at 500. I have no idea what assumptions you have made to do it at 500. So these two things I think you should validate with larger population and do some market research to really arrive at this. The problem that you're solving is real problem, absolutely real problem. And I think your presentation is pretty good, but I just wanted you to be aware about these two aspects, which I felt they're not very commercially techno-physical. That's what I wanted to convey.
So we have done research related to that also. There are clinics and hospitals that have LCMS as their in their research facilities, and also right now at tertiary hospitals also they have LCMS unit. So there is a major number that is all of LCMS unit being present around the world, majorly in metro cities like Bangalore, Hyderabad, and Delhi. So it is a large number is present. So we'll start by LCMS and later on we'll shift to point of care test.
How did you arrive at this 500 rupees test?
Sir, this number we have estimated for now. If you look at the global market, per sample test is around for $1,250. So we are estimating our test to be for 500 rupees per sample, just for now. So like that, we have included the number by the first one is the total addressable market, include the number that are individual working in organized sector more than 40 years. And the subjected one is the people who are under health coverage insurance. That is how we have calculated.
Thank you very much.
Thank you, sir. Thank you. Next, we have lovely PhD scholar. You have 4 minutes for the presentation.
Thank you. So, good afternoon, all of you. I'm lovely PhD scholar from Dr. Krishna Mohan Atmakuri's lab. So, I'll start with the question like, despite the continuous efforts and very effective drugs in our hands, still we are lacking behind, and TB remains a challenge and far from eradication. India alone accounts for more than 25% in the recent WHO reports. So why this is happening? So we see the challenges is the 6 to 24 months, the treatment duration with the lots of drugs and the high doses of the drugs, resulting in not only the side effects which the patients are suffering from, also there is economical burden, not only the common man, but also the government also through their TB Mukt Bharat program. So with this, how can we reduce this with the shortening of the treatment duration, reducing the side effects with the reducing the doses of the drugs, and for the quick elimination of the bug before the emergence of the XDR and the MDR strains? So for this, in 2014, in the transcriptomics data published in the TBDB database, so there was an interesting gene that we found that was responsible for the tolerance to the higher doses of the drugs, and found mostly in the XDR MDR strains. So what's that? And we were interested in that. So for that, we planned to explore the role under the stress conditions. So we made the knockout in the MTB. And we exposed to various stresses that the bacteria encounters inside the host that I have mentioned. So you can see that deleting this particular gene, the bacteria cannot survive more than a day under these stress conditions. So what's about the antibiotic stresses? So in the antibiotic stresses, we can see the green one, after deletion of this gene, the bacteria is not able to it was struggling to grow in the higher concentration of the drugs, INH and the rifampicin, which are the first-line anti-TB drugs. So, but also with the reduced amount of the drugs encircled. We were excited to know that can be targeted in the Mycobacterium tuberculosis. So for that, from the literature, we took a inhibitor that was specific to this protein, and we exposed the wild type to various concentration of the INH without this inhibitor. So you can clearly see the blue in the presence of the 4x INH, the bacteria still survives. But in the presence of this inhibitor, it gets clearly killed just with the exposure on the 12th day itself. So this excited us to check what happens inside the animal model. So we did this animal experiment. The guinea pigs exposed the guinea pigs with the strains, knockout and the wild type, and also the complement. And for over a period of time, we looked at the bacterial burden, and we observed that not only the lungs and spleen, but also the liver, we found the reduction in the bacterial burden on the 12th week of post-infection. Also, the interesting thing we found that in the blood, the wild type bacteria was flourishing, but after deletion of this gene, there was no growth in the blood. So this suggests that by deletion of this gene, it also helps us in reducing the systemic spread through the blood to other parts of the body. So also suggesting that it can reduce the risk of the spread, the extrapulmonary TB as well. So what happens in the histopathological analysis also, we found similar results. So the necrosis, which is helping the bacteria to spread throughout the body, causing the extrapulmonary TB, we found there is negligible necrotic granuloma formation in case of the knockout. So this excited us to let's find some molecule against this target, and for that we, in collaboration with Dr. Asthana in Computational expert team. So we screened FDA-approved molecules and some other libraries targeting this, and we found some molecules, best molecules showing the in vitro activity at just 10 micromolar. Another interesting results, we found that this knockout was also sensitive to other FDA-approved molecules at just 10 micromolar. So that is the another aspect we are looking also. And this suggests that this is a very promising target. So our main aim is to shorten the TB therapeutic regimen with the reduced amount of the drugs and doses, and with the least side effects for the TB patients. So I will thank my mentor for this opportunity for this project and the advisory committee as well. And thank you, THSTI. Thank you.
Yes. Thank you. Any comments from investors?
I just want to make a general comment that India has pledged to be TB Bharat Mukt by 2025. This was a target, but I think we will achieve in 2030. So keeping that in mind, do you think it makes sense for you to do what you're doing, like from our research to commercialization? If you're doing research, it's fine. But if you're looking to do this from research to commercialization, will there be a market in India for the drug when TB Bharat Mukt happens? And I think it'll going to happen certainly by 2030. So any thoughts?
So what I feel that unlike the other diseases, the duration of the treatment is very long with lots of drugs. So the disease is curable, but the problem is the side effects due to which the patient, like, they don't complete their treatment regimen and they stop the completion. So this emerged out the XDR and the MDR strains. So we are thinking that if we inhibit this particular target, so the current available therapeutics drugs that can be more effective, and also within a shorter period of time the patient can be free of like completely curable disease, it can be made. So I really think that this can be this can contribute importantly in the not only the TB Mukt Bharat by 2035, also the world in some ways, if it is supported. Like,
thank you.
Thank you, sir.
Any other comments from investors? Yeah, thank you.
Thank you.
Next, we have Dr. Pratibha Sharma, a project scientist. Presentation.
So a very good afternoon to all. I am Pratibha Sharma, and our group is working on sepsis. So first of all, what is sepsis? Sepsis is a dysregulated host response syndrome due to multi-organ failure and death. So globally it has affected 55 million cases, and India alone has contributed to 1/3 of this global burden. So the etiology of sepsis remains unknown in majority of the cases because the blood cultures, which are considered as the gold standard, remains negative, it has culture negative sepsis. So the prompt diagnosis is very important to initiate initial treatment so as to ensure the better patient outcome. However, since sepsis is diagnostically challenging and because of standard diagnostic tests which are not available for sepsis, all cases which are suspected of sepsis are administered with a broad spectrum antibiotics. So this uniform therapeutic regimen is given to the patients complicates the situation by to the emergence of antibiotic resistance. And on the other hand, if the patients are not responding to the therapy, it may further sepsis to sepsis shock, which is an advanced form of the advanced form, and further multi-organ failure and possibly death. Also, if we are able to segregate non-survivors, the clinician could start a targeted treatment and the lives could be saved. However, this uniform therapy also fails to segregate the early potential non-survivors for the targeted treatment, which further adds to the unnecessary cost, morbidity, and possible mortality. So the current challenges which are associated with sepsis are segregation of the septic from the non-septic patients and segregation of the potential non-survivors from survivors. So in the today's scenario, the diagnosis of sepsis is basically linked on the markers which are used in the clinics, which includes the calcitonin, which segregates septic from non-septic. However, they are non-specific. Lactate being the which is proved best in the advanced form of sepsis in septic shock. And none of the markers are available today to segregate non-survivors from survivors. And if this has to be discovered in patient group, then a huge funding is required and sampling from the multiple hospital cohorts is required. So therefore, to shorten up that, we established a new model for biomarker discovery. In our study, we have developed a bacteremia-based sepsis mice model to look for the markers that could distinguish sepsis from no sepsis and also the non-survivors from the survivors. So this is a monomicrobial sepsis model, bacterial sepsis model that has been established where the pathogen is administered in the mice through and following this, the progression of sepsis was assessed. It was assessed on the basis of the increase in the sepsis scores, decrease in the weight, temperature, and other parameters, leading to some survivor mice and some non-survivors, which are termed as septic and non-septic. The blood samples were collected at as early as 1 to 2 hours, 6 hours, and the time of death, or in survivors, it is 24 hours. So, following this, a panel of inflammatory cytokines and chemokines, which was selected from the literature, were evaluated, and along that, was done in order to assess the metabolites which are varying in sepsis. So, we found that 9 cytokines and chemokines and also 6 metabolites, they were found to exhibit a significant difference between the septic and the non-septic, and survivors from the non-survivor at an as early as 1 to 2 years post-infection. Also, there were 2 metabolites which could help monitor the progression of sepsis and the potential outcome. So this is so on whole, we have developed a bacterial-based model of sepsis, where we have identified biomarkers that could segregate septic from non-septic, and also the potential non-survivors from survivors. So on whole, the shortlisted biomarkers now needs to be taken up to the evaluation in the patient samples on a small scale, and then further in the large scale, and followed by the commercialization in the kit-based format. Thank you.
Comments from investors?
How long does it take to actually detect sepsis in your test? Once you take the blood sample, how much time does it take to identify?
So we can for diagnosis.
Yeah, because that's what skills.
Yes.
That's time.
So we can proceed for the, like, once it is commercialized, then we can go for the lateral flow assay, which is only a like 1 minute procedure, and if we can further go for ELISAs, which is I think half a day method.
I'm asking you because actually we have invested in a startup,
yes,
which actually using light, they can detect this in like 15 minutes kind of thing.
Mm-hmm.
So because the timing is very important in this. That's why I was asking that.
Yes, sir.
Yeah, couldn't agree more with you. I think it's a TAT, which is very, very important in clinical setting. So like right now in laboratory setting, how much time is it taking for you?
So for the so we identified these this ELISA that we perform, it is a like half a day procedure. Like once,
but that's a very conventional procedure, right? That's a gold standard for you right now.
So this is
right?
This is a 10-plex kit where we can identify the 10 cytokines. So that is it's half a day procedure.
No, but that's the current methodology being adopted by clinicians, right?
Yes, sir.
So what is it that you are trying to do?
We have as of now identified, because so there are no biomarkers that are available for the segregation of non-survivors from survivors. So we have currently identified those markers, and
So
that
you
can
have
take
how many biomarkers that you've
So we
identified
have
with?
9 biomarkers.
9 biomarkers. So will they work like a one after other, or will they work together for you to be able to say, yes, the chances
Seen
of...
as that we have seen all the 9 biomarkers had shown significantly increased values in as compared to the survivor versus non-survivors, and also in differentiating between the sepsis and the non-sepsis cases.
Okay.
Yes, sir.
Got it. So is there a clinical validation or something that you are
That is
planning, or you will need to do?
Yes, sir. That's
Okay,
right.
okay.
Yeah.
And this has been your research area for how many years?
Sir, actually, I have recently joined the lab. It's been 1 year. So this metabolite work was done by my previous postdoc here, and I have joined the lab. So for the other pathogen, we have given the samples, and the metabolites we are waiting. But this work is my previous postdoc, which were present in the lab. So his work was, I think, 2-3 years.
So metabolite and a biomarker. So what's the correlation here? Like how many metabolites equal to one biomarker? Is there a kind of a correlation? Have you been able to
So
establish?
6 biomarkers, 6 metabolites, which were found to be correlated with this segregation between the non-survivors and the survivors.
So which means 6 metabolites is equal to 6 biomarker. Is
Yes,
that
sir.
what you're trying to say?
Yes, sir. We have
Okay.
to see that if one biomarker is even raised in the patient samples, so what is the outcome? So we have not done the clinical studies as of now. So we
But
will
then
be
if you do 6, at least the chances of predictability is more
Yeah.
correct rather than just taking one biomarker, right?
Yes.
That's the hypothesis that you're working on.
Yes, sir.
Okay, got it. And in terms of the TRL stage, what stage are you at?
No, we are
Maybe 1 or 2 laboratory
stage
side.
only, sir. As of now, we are. at
Have
the
you
lab.
done any provisional patent filing?
No, sir.
If you've not done
Might
it,
be
please do it.
knowing that
Because this is a very, very important area. It's underaddressed area, and it's something which, if you can find a problem around, like a solution to a TAT, which could be like 1 hour, 2 hours, I think you are solving a real problem. So
Periods,
at least try to file a provisional patent with whatever claims that you think you can, and then prove it over a period of time and file the final application. But it's a very, very impactful area, and thank you so much for sharing that. Thank you.
Thank you.
Thank you. Next, we have Dr. Arpita Singh, a PhD scholar.
Good evening, everyone. My name is Arpita Singh, and I'm a PhD scholar here. So here I have a verified scientific idea that I need your help to turn into a scalable and sustainable product. So I'll start with the problem here, which is the preterm birth. So the preterm birth is any live birth before 37 weeks of gestation. And it's the leading cause of fetal morbidity and mortality here. And the actual problem is not preterm birth, but the complications associated with it to the infant in early and later stages of life. So there are tools and kits available for the prediction of preterm birth in market, but the problem with these kits or these tools is that these are too late. If you can see here, this is the scale of gestation age. Gestation age means the 9 months of pregnancy. So it's in the weeks. So we can see that most of these tests are being performed after 22 weeks of gestation. So these tests are more like triage tools instead of the early risk assessment. So the one which is available at 20 weeks is based and it's not LMIC validated. So here I want to propose an idea that is the maternal serum metabolites. So metabolites, as we know, is something an established concept already in the market. It's proven, it's regulatory accepted, and it's clinically feasible. So we can see that metabolites are being used in the diagnosis of various diseases like diabetes and kidney
dysfunction, etc. So here in our lab, we have performed a study where we profiled the metabolic pregnant women pregnancy. We had the serum samples collected from them at early time point, like 15 to 20 weeks of gestation, and we found these 8 metabolites to be dysregulated in preterm birth mother. So out of these 8 metabolites, 3 are verified and can be and have the potential to be used as the signatures of preterm birth. So talking about the market size of preterm testing globally, so right now it is 1.4 billion USD and predicted to be 6 billion in 2030. And in India, the market currently is 48 million USD and it is also predicted to be by 2030. So the path forward, like here our goal is to make a product which is more affordable because our current platform is LCMS, so which is excellent in accuracy but is not feasible in the point of care test settings like clinical settings. So here we need the support to validate and develop a product which can be translated to the clinical setting. So thank you.
Hello.
Yeah, ma'am.
So if you find out you're going to have a preterm, what are the interventions? Do you avoid the preterm or what do you do?
There are two things. First, if it's in early, if we can predict it early, yes, we will try to delay the saturation. Second, we will like to intervene it. Like we can fast forward the development of the baby, and also we try to extend that gestation period to be 37 weeks only.
Hi, Rahul here. Yeah. I have seen this is a preterm baby, gestation period 23 weeks. Up to 27, 28th of week, we don't know if it's a preterm or not.
Yes.
But suddenly about 29 to 30th week, I remember that doctor suddenly said the BP is fluctuating and we have to get it operated by the second and third of week and some water level is going down because of the BP issue. So how will this handle in that situation? It will work to avoid that situation or not?
If we predict it early, yes, that is our ultimate aim to delay that delivery and to prevent the baby from going through all these complications.
And you mentioned it's expensive in the market. So what's the price of this product, the test in the market?
So currently, as I told, this is not in the regular practice in India. So basically, it's being done after 22 weeks. So the regularly these are not being used in India. It's only in US mostly.
And what price you introduce in this Indian market?
Is I don't remember at the exact price right now. So I can tell you later when if you want to discuss. Yeah.
Just a follow-up question. So globally, these tests that are available, what are they used for? Like, is it part of the clinical protocol that after a certain week you have to start doing it every week, or how does it work?
So that is the problem, that it's more of a triage tool. So when a woman has already developed the symptoms of preterm birth, like early contraction, then only clinicians use these tools. It's not a part of early risk assessment. Not yet. So that's our aim to bring it to the regular pregnancy test. It can be a next glucose test in the pregnancy care.
So, how early are you thinking like that? If there's a viability or feasibility of this, that
Sorry, I didn't get it.
thing that we started to incorporate in as a part of the regular pregnancy test.
If it gets the part of the pregnancy,
how can it? So
your voice is not audible actually.
We
Can't hear you.
This test available is at the clinic that is also globally not in India and it's pretty expensive. This question is, Can this be brought down a little early? Can this begin at the 6th week, 8th week, or do you have any other time in mind?
Yes, actually, 6th and 8th week is very early. But we are planning here to for 14 to 20 weeks. Like it's the early second trimester. Till now it's late second trimester, but we are trying for the early second trimester.
Next, we have Dr. Puja, PhD scholar.
We have 4 minutes for the presentation.
Yes.
Hello everyone, I am Dr. Pooja. Today I am going to introduce a novel diagnostics device, that is Stone Sense, development of a urine analysis based portable colorimetric test for early detection of the kidney stone to facilitate its management through hydration and dietary changes. Kidney stone is a silent disruptor of the healthcare. In India alone, the prevalence is 12%, that makes it 168 million cases. The incidence is rising at the rate of 2 million per year. Men are 3 times more likely to suffer from the diseases. The burden is even higher. The prevalence is 20%, impacting 700 million lives. The problem doesn't stop here. Even after treatment, the disease reoccur at the rate of 75%. Symptoms often overlap with the many other conditions like GI disorder, gastritis, and many other conditions that leads to often misdiagnosis of the case and 40% delayed diagnosis. That leads to the surge in the complications like surgical intervention and the chronic kidney disease and the kidney failure that push the patients into the financial crisis of the 40,000 to 1,50,000 direct cost per patient. The disease progress mainly in the 4 stages. The first 2 stages are the asymptomatic stage, stage 1 and 2. The stone size is the less than 3 mm. That are easily manageable with the dietary changes and the water intake. But the problem is the only 20% cases diagnosed the stage 1 and the 2. And the rest 80% progress to the stage 3 and the 4. When the stone size is greater than 4 mm, that requires the only surgical intervention. Due to the sort in the available diagnostic like ultrasound, x-rays and 24-hour urine analysis that doesn't detect this stone at the smaller size, that is at the early stage. Only CT scan detect this stone at the early stage, but the CT scan are very expensive, not available at the average PHCs, and expose the patients to the radiations, and these all the current diagnostic require a high resource requiring, that is a skilled labor and skilled technicians are required and not field deployable, only can perform at the clinical infrastructure at a specialized hospitals by the specialized clinicians and technicians. So there is a critical gap, a way to develop a point of care device for the early screening of kidney stone at the less than 3 mm in size to avoid the surgical intervention and prevent complications, to prevent the condition by only minor lifestyle modifications and the hydration. The limitations in the current diagnostics with the high recurrence rate of the disease and the lack of the available diagnostic at the primary healthcare center, there is a need of early screening of the kidney stone. So here comes my proposed solution. We are developing a urine-based portable colorimetry screening tool for the early detection of the kidney stone based on the chemical analytes in the urine, combining with the microfluidics technology that will detect this stone at early stage less than 3 mm in size, which will give the result within the 5 minutes and differentiate the stone types to facilitate the treatment. And there will be a visible color change for the easy self-interpretations. This will be accessible to the all demographics. This will be non-invasive, radiation-free, equipment-free, no lab or expertise required, broadly compatible to all type of populations, and this will be self-usable and home-based. We will be detecting the different types of the stone through the colorimetry reactions by identifying their unique biomarkers through the color-changing chemical reaction. For example, mainly there are 5 types of stones. The calcium oxalate comprises 80% of the stone types. There will be a specific biomarker for calcium stone in the urinary, and by identifying the biomarker will react through the specific reagent, and there will be color change from orange to green. Similarly, the uric acid stone, strobate stone, brucide, and the cystine distinct color change will be for every stone type. There will be microfluid or multi-channel. Okay. There will be multi-channel microfluidics coated with the reagent. The biomarkers will react with those reagents and color change will occur if the biomarker exceeds the threshold. Color change indicates presence of the kidney stone. No color change, no risk of the stone. Here's my solution, the stone sense, which will detect the stone at very early stage, even in the asymptomatic stage. It will be field deployable, turnaround time is less than 5 minutes. Very low resources will be requiring as it is user friendly. Kidney stone diagnostic market are growing in 2024 from USD 2.39 billion, projected to grow in 2034 to USD 4.42 billion with the compound annual growth rate 6.3%. Total addressable market is the 8,400 crore. Serviceable addressable market is 3,100 crore, and serviceable optimal market 20 crore. We will be tapping 3 areas for our revenues: B2B healthcare providers, primary healthcare centers, urologist clinics, and diagnostic labs. Direct to the consumers, those at the high risk and the recurrent kidney stone disease and health conscious consumers, and the business to the distributor. That will be the medical device distributor, retail network supplier, and global distributor. Globally, 10 million urology ultrasound are performed per year and 500 million abdominal ultrasound are performed per year. In India alone, 14 million ultrasound are performed per year. By tapping this, by creating the first screening line for our ultrasound scan, there is a great segment for this market. Current status is we have already developed 3 of our chemical assays. We have developed our alpha prototypes, and the prototype testing is done using simulated fluid, and the microfluidic chip design has been already done. We are building a platform technology, not just a product. In phase 1, kidney stone detection, this is our flagship product. And in phase 2, we will go for UTI, CKD, gallstone, etc. And in phase 3, multiple urine-based tests in one device. This multi-disease pipeline to scale our revenue scale across the markets and the geographics. Thank you.
So, why microfluidic platform? Just trying to understand. Why not simple other tests like ELISA or something?
Sir, microfluidics, because the micro channel of the microfluidics will allow for the low urine volume to flow through the multi channels, as there are 6 different types of stone.
But there is no problem with the urine collection of 10 ml or 20 ml. It's not like you're actually taking a blood, right? There is a challenge there. Okay, so for urine as a sample, I don't think there's a challenge.
No, sir. Actually, we are going through the closed chamber microfluidics. There will be no risk of the contamination of the urine. Actually, only opening will be for the urine sample collections, and the device will be totally closed chamber. That's what we.
Okay. So currently, how is it diagnosed? Like, I'm sure it's been diagnosed. Yes. What is the current methodology?
Currently, the first line of the diagnosis is the ultrasound.
Really?
Yes, really. And the people then go for the CT scan. Even in ultrasound.
Is that what being practiced, or in India you're talking about?
The stone is detected from the ultrasound and the CT scan.
When patient complains about it, right?
Yes. And the symptoms are overlapping with the conditions like gastritis, GI disorder. First, they visit with the gastrology, then they do ultrasound, and then visit after urology. Then they confirm after CT scan. Even in ultrasound, 41% patient need a CT scan to confirm the stone. And as the treatment totally depends on the stone types, which type of stone is there, what is the composition of the stone. So they have to go for the lab testing, like 24-hour urine analysis and all. So I thought my device will differentiate the stone type there, because there will be 6 channels for 6 different types of stone. So we all know the composition at the time of the testing.
Okay. And you're saying that you're going to do it less than 3 mm, right?
Yes.
So, biochemically, why less than 3 mm? Well, the same technology can actually take care of even if it's 4 mm with the same precision and sensitivity and specificity. Why only 3 mm? Just trying to understand.
Yeah, important because when the stone formation starts, as the chemical deposition concentration increases, the stone size increases according to the concentration of the chemicals. When the stone size is 1 to 3 mm, there is no need of surgical intervention. It can be only treated by hydration and dietary changes. After 4 mm, there is a surgical intervention, like we have to go through lithoscopy and many other surgical intervention according to the stone types. So I'm targeting.
So, is there enough literature available to suggest what you're saying?
Yes, there are.
Globally.
Globally.
So basically what you're going to try to do is to replace this ultrasound imaging with this particular biochemical test, right?
Yes.
It's purely biochemical.
Yes.
Right? And a POC. So essentially screening, right?
Yes, point of care test.
Like any other screening, this also could be added there, right?
So people don't have to go for unnecessary scan.
So by when do you think you will probably go to the market from, let's say you get all the support, like from DBT to BIRAC to, us, like whatever, we give you all the support, what is required. Earliest, when can you introduce this?
Currently, sir, 3 of my chemical assays have been developed. Rest 2 have been going to develop in, and we are ready to test in the real sample and clinical validation. I have put there 5-year roadmap. If we go through this plan in next 5 years, we are planning from lab bench to
5 years. So basically, you are a TRL 1 or 2 right now. Is
yes,
that
sir.
what you're
yes,
saying?
Development, and I have developed alpha prototype.
And you are also currently doing your PhD. You're doing your PhD or you're finished with PhD?
Here is mistaken. I am a fellow at the School of Innovation and Biodesign.
Okay. So your full-time responsibility lies to do research.
Yes.
And is this what your main research is all about, or this is like a sidey thing that you're doing?
This is the main research I am doing. I am developing a product and planning to incorporate my company in next 2 3 months, and we'll go further with support and with all the support.
Great, great. How did this idea happen? I'm sorry I'm bumping into too many questions, but how did this idea happen to you?
Actually, my own brothers one suffer from abdominal pain. We go further visit to the gastroenterology, and they are us like actually.
I understood. It came from your own exposure from your family.
Basically, the stone is very less in size. The people visit multiple clinician visit for ultrasound. As the recurrence rate is very high, the disease comes after every 2 years, 3 years. So people have to visit multiple times for diagnostic. So if they can test the kidney stone at home, and if know by only merely a color change, so this will be great because they can treat it by self.
I am reminded of one startup in IIT Bombay. I'm forgetting the name. I can come back to Manish and Manish can tell you. But there are 2-3 startups out of IIT Bombay who've developed POC tests across whatever you have shown. They are already doing it. They're marketing it, and they have raised close to 12 to 13 crores of money. So please have a look at them. I'm not getting the name. I think the founder name is Nikunj Malpani, but I'm not sure about the company name, but I've had a chance to interact with him. So please do some research about it. So
Talks.
yeah, that's right. Absolutely. Yes, Alka. Great. Thank you. Yeah. Thank you very much. I'm sure you will have seen them many times.
Thank you.
Thank you. Next we have Dr. You have 4 minutes for the presentation.
Myself, Himanshu. I am a Ramanujan Fellow at the Research Center of THSTI. So tuberculosis is basically caused by the pathogen Mycobacterium tuberculosis, which is primarily transmitted in the form of aerosol from an infected person. TB is curable, but despite the fact that it is curable, it still harbors almost 1/4 of the total global TB cases. It's not like that we do vaccine yet. Vaccine, which is Mycobacterium bovis BCG. BCG was discovered almost a century ago and is routinely administered in countries. So administers BCG at childbirth. BCG has its own set of shortcomings. For instance, It is able to provide protection against the symptomatic form of the disease, but it loses its efficacy against. Furthermore, BCG efficacy rules every time, making the adolescent and the adult getting infected. Secondly, there have been instances where people tried BCG, but shown to be that efficacious. Finally, BCG strain can cause BCG masses in immunocompromised and incompetent individuals. So, all these drawbacks together, it's BCG or a booster for BCG in patients like India. Now, this is a list of the different vaccine candidates that are in the clinical trials. For instance, are in the advanced phases of the clinical trials. However, the factor that is missing in this list is that there is no indigenous vaccine candidate in this list. And hence, my today's talk of developing an indigenous mycobacterial lysate-based booster vaccine for adolescent and adult population. Now, coming into the how did we develop this project, or what was the curiosity factor behind this project? So we know that there is this Montreux test, which is basically used to diagnose whether a person has been infected with TB or not. So Montreux test is basically an intradermal injection of purified protein derivative, which is nothing more than Mycobacterium tuberculosis THSTI cell culture filter. So we get that injection if exposed to TB. We get the swelling if swelling. Interestingly, whenever we get this tester, we don't get subsequent TB episodes. However, interestingly, there are some studies where the same cell culture filter preparation, when adjuvanted with some adjuvants like CpG and protection against the TB challenge. Interestingly, there was another study has demonstrated that there are a lot of immune-dominant antigens that are not secreted out into the cell culture filter, but present in the cytosol, and which we call as mycobacterial lysates. And these lysates can contain a lot of immunogenic glycoproteins and violence factors, which makes this lysate an interesting immunogen. Very interestingly, these bacterial lysates are also clinically approved and therapeutics like OM85 or the Vaxum, which is used to treat upper respiratory tract infections in infants. So, with all these background studies, hypothesis that mycobacterial lysate might be a better immunogen and used as a booster for BCG. So, we mechanically distributed the BCG and the Mycobacterium tuberculosis H37RV and got its lysate, and we immunized mice. The mice model that we used here was almost 12 months old. This recapitulates to a human of around 40 to 45 years old. The mice, and we saw that the lysates can activate CD4 T cells in the spleen. That is, we get a nice population of CD44 expressing CD4 T cells. Furthermore, we also found that these lysates can induce antigen-specific central T cell populations in these mice. So, this TCM population is basically necessary to protection against the chronic phase of the infection. That we also looked into the different cytokines which are indicated for protection against TB. For instance, TH1 cytokines like interferon gamma, IL-2, as well as TH17 cytokines like IL-17. And we found that these lysates can stimulate the production of TH1 cytokines as well as TH17 cytokines. In the pipeline, we are also trying to evaluate the protective efficacy of these lysates against aerosol challenge. We are also looking at the safety profile, like looking into the CBC as well as LFT and KFT levels of this immunized mice. Finally, we are also in the process of studying the stimulation capacity of these lysates in human MCs, which are isolated from both healthy as well as latently infected individuals. Finally, we have to study whether boosting BCG with this lysates formulation can increase the protection against TB. So, what are we focusing upon in this project? We're looking to develop a cell-free subunit vaccine against TB, which can be used as a booster for BCG and targeting the adolescent and the adult population with TB. And finally, we're trying to develop an indigenous booster vaccine,
What I'm looking for today is user expertise. Look what are the different parameters we can include apart from what we had thought of to make this product go beyond the publication. Furthermore, do you guys think that this product is really feasible to successfully give the lab? So that's all from my side. Thank you. In terms of like,
yeah. So in terms of like performance against currently available.
As of yet,
okay.
We in India just have the BCG.
Yeah.
The advanced phases we do have the MTB-VAC, which is developed by Spain.
Makes sense.
Yeah, which is developed by Spain, but it is undergoing clinical trials in India also. Yeah.
Okay, makes sense.
What is the efficacy in phase 2 trial of that vaccine that you mentioned? What percentage of efficacy has been reported there?
With the MTB-VAC?
Yeah. Because that's the only one that I know of in the phase 2.
The advanced trial. So what in TB endemic nations, it has shown somewhat like 46 to 50.
Okay. Yeah. And is that a recombinant one? Like, are you what's would be
strain? It is a live attenuated strain. It's
something on the lines of COVAX, right?
COVAX
was also
yeah. So it is 2 gene knockout, the MTB-VAC strain. Yeah.
Okay. And yours is a recombinant? What are you?
Ours is not recombinant. Ours is a lysate based. Okay. So we mechanically disintegrate the bacteria, take out the antigens, and then use it to immunize it.
But that's going to be a very multi-genic approach, right?
Yeah, I know.
So vaccine typically, I don't know, a preventive vaccine, or what will you call?
We are basically calling it as a booster for BCG. We are targeting, say, some attenuated strain, because in immunocompromised strain, there is a possibility of the bacteria getting disseminated and uncontrollable. That's why we are mostly targeting this aspect.
Sure. So, TB as an area is of not much commercial interest from pharma companies and all that. So how do you plan to sort of undertake the clinical development going forward from now?
Clinical development, so far what I see is that we have the proof of concept, which is almost like a TRL3 phase, right? So then we definitely have to look into 2 different animal models, preclinical animal models, the safety for the arrows, like for the protective efficacy study. Like then we definitely have to go for some non-human primates, like with some collaborations.
Okay, thank you.
Yeah, thank you.
Thank you. Next, we have Taruna, a PhD scholar.
Differentiating between sterile inflammation and infection remains one of the biggest dilemmas in acute healthcare setting, which misidentifying the cause linked to delayed treatments, unnecessary antibiotic overuse, and even increased mortality. Hi, I am Tarna, a PhD research scholar working under the supervision of Dr. Bhabatosh Das, Professor THSTI. So my research mainly focuses on identifying the early microbial markers in critical care illnesses like acute pancreatitis, trauma, and sepsis. Patients can develop inflammation, yet in many cases, there is no infection was found. So this raises the question, the immune system is responding to what? So in order to answer this question, we have chosen a disease model, acute pancreatitis. Why? Because early sterile inflammation, which is DAMS and immune activation, can be studied separately from the later infection driven by microbial translocation and sepsis, which is occurs later in the phases of acute pancreatitis, making it ideal for understanding how the immune system responds differentiately to sterile versus infectious triggers. So let me first walk you through what acute pancreatitis is. So pancreas being the vital organ of the body, it secretes certain digestive enzymes and certain hormones acute inflammation of the pancreas, including etiologies such as gallstone, ethanol, idiopathic, and high levels of triglycerides levels, leads to the local injury of the pancreatic tissue. And there are certain, pathways which may induce the systemic inflammation. So the triggers here can be infection, or it could be sterile, such as damage-associated molecular patterns. So in order to understand the trigger of the systemic inflammation is very important because it leads to organ failure and having a mortality of up to 40% in case of acute pancreatitis. So, redefining critical care and why we are choosing acute pancreatitis, why it demands precision research. So, basically, acute pancreatitis, as I said, is no longer seen as a uniform or self-limiting inflammatory disease. It is now recognized as a heterogeneous and dynamic condition with diverse molecular, ecological, and clinical trajectories, ranging from mild, self-resolving inflammation to the life-threatening multiple organ dysfunction. And also it mimics or shares some features with sepsis as well, such as systemic inflammatory response syndrome, multiple organ dysfunction, cytokine storm, and immune dysregulation. So we daily, in the news, we are seeing many headlines saying that we found a drug to treat the infection, we found a drug to combat inflammation, but let me tell you, the reality is we don't have any biomarker or anything to differentiate between the sterile inflammation and the infection. So in order to answer this question, we have done a prospective observational cohort study where we have recruited acute pancreatitis cases with varying severity. Also, the infection confirmed positive cases, healthy controls, as well as the reagent controls. So we have collected the blood, rectal swab, buccal swab, and the skin swabs to do the metagenomics. We have also done the transcriptomics profile of the different cell population of these individuals, where we have chosen neutrophils and PBMC since these are the two cellular in case of infection. And we have also done the culturomics to validate our metagenomics results. So the clinical site was AIMS. We have collected all our samples from AIMS and the sample process done at THSTI. So that's a current status of our project progress. So we have identified certain certain microbes which we have found in the blood, and they are showing similarity with the rectal as well as with the buccal microbiome. We also have identified certain cellular phenotype FACS technique to basically typing. So we have isolated, we have identified certain cellular level of certain cells which are mainly found to be increased in the infection case. We have also validated our results with the culture mix, which is and in transcriptomics also we have found certain genes which were very beautifully differentiating between the two, between the sterile inflammation and the infection. So the idea is to connect the dots and using this multi-omics technique to understand the disease pathophysiology. So using metagenomics, we have identified certain microbial signals which targeted. So just now we have developed, identified certain microbial signatures that were found to be associated with the infection cases and differentially associated with the inflammatory sterile inflammation. We have also screened or identified certain host signals which are present with infection suspected. We also have, staphylococcus automatory subset of cellular population, which mainly is the origin of systemic immune activation. And lastly, we have also used the metabolomics technique to further validate the host versus microbial derived metabolomic signature. So the idea is, our mission is to combine all these omics technique together, that is metagenomics, culturomics, metabolomics, transcriptomics, and then powering innovation in critical care through our precision omics technology. So the analysis platform involves the measuring omics signatures with unparalleled accuracy and coverage that we have in our lab, optimized lab protocol to differentially identify the signals, host signals versus the microbial signals. Discovery platform enable us to the evidence-based personalized testing discovery of diagnostic or prognostic host or microbial biomarkers, and investigating the complex relationship between them. Also, the therapeutic platform then enable us to develop some precision techniques that provide personalized therapeutic strategies. Lastly, I would like to acknowledge my mentor, Dr. Bhavtosh Das, our collaborators, Dr. Pramod Garg, Department of Gastroenterology, AIMS, Dr. Nitya Vadva, Data Management Center, THSTI, Dr. Soumya Jagannath Mahapatra, Department of Gastroenterology, AIMS, Dr. Amit Avasthi, Center for Immune Biology, THSTI, Dr. Deepak Rathod, FACS Facility, THSTI. I would also like to acknowledge DBT for providing us the grant to the in vivo analysis. Thank you. Thank you so much.
Looks like nobody's asking, so I have to do that. Okay, so your approach is to develop a platform which could enable the precision medicine, right? And you're targeting to start with a critical area, which could be infection, right, in the clinical setting. So is my understanding correct?
Yeah. That's what we are doing. So we are mainly, taking or differentiating identifying the biggest problem, which is differentiating between the sterile inflammation and infection. And can we use this because there is no single technique which aid us to identify or to differentiate between the two. So we have to combine all these techniques together in order to get the output
But how will it improve the clinical outcomes? Like, of course, differentiation is one part, which could be a great hypothesis to talk about. But how will it will improve clinical is what I would love to hear from you.
So most of the cases in the ICU are mainly because of infection, right?
Agreed.
And clinicians don't have, they really don't understand whether it's a sterile inflammation or the infection, because the symptoms that the patient is showing is same. The
Symptoms are changing every day.
Yeah.
Every day, right?
There's no single.
One day they'll tell you, the other day they'll tell you MRSA, third day they'll tell you something or the other. So is your solution going to tell them very clearly the set of an infection?
Yes.
This is what the etiology would look like?
So the idea is to tell when to use antibiotic and when to no.
And what antibiotic to use? That's an important aspect, right? When you change the AMR side, because that's a larger problem to solve, right? So
Metagenomics and whole sequencing of the bacterial isolates that we are getting out of the infection cases, and we are definitely the pathogens and checking the genes which could be the most setting, and then we can develop some nucleic acid based testing in order to detect them at a rapid point of care test. Yeah.
Okay. So all this work is done through the human clinical trial, ethics approval, and everything is in place?
No. So recently we got a grant from DBT, and not clinical trial, but a prospective observational study we are doing.
Okay, largely scientific.
Yeah.
And you have a collaboration with AIMS, I understand.
Really, yeah.
Okay, so you get access to the samples from AIMS. Is that what you getting?
Yeah.
That's about it, nothing else?
So the clinical side is AIMS New Delhi. We are getting all the samples and the clinical metadata out of the hospital, and all the lab protocols were designed here in THSTI.
And you also get the data of the patient, like what kind of etiology the patient is suffering?
We do have their clinical metadata as well.
Okay. So how many such samples have you already processed?
So till date, we have around 40 samples in both different severity of acute pancreatitis, like mild 40 cases and severe 40 cases we have involved. And we are collecting the samples in, different week of illness. So in first week of illness, we are collecting, then in second week, then in third week. So acute pancreatitis initially originates as a sterile inflammation, and later it converts to infection. So the idea is to see how the immune activation or how the immune system is differing in the first week of illness with the third week.
Sure.
And then,
have you observed MRSA interactions in this?
Yes.
Okay. Got it. Thank you. Thank you so much.
Thank you so much.
Great work.
Next, we have Tanshi Mehta, PhD scholar.
Very good evening, everyone. I am Tanshi Mehrotra,
I am a fifth year PhD student and currently I am working under the guidance of Dr. Bhabatosh Das in Functional Genomics Laboratory. And today I am here to present rapid point of care diagnostic platform for dual detection of pathogens and antimicrobial resistance both. So my idea is to understand the bacterial genome and then translate it using functional genomic data analysis technique into a field deployable diagnostic assay to detect both AMR genes and pathogens, which is the need of R. So, as we all know, that AMR is a growing threat and it's a silent pandemic which soon will be hitting us by 2050. A study in Lancet Micro has highlighted that almost 8.22 million people can die by 2050 due to AMR, with Southeast Asian region highlighted as carrying one of the highest all-age mortality rate by 2050. Major pathogens contributing to this are gram-negative members of escape pathogens like Acinetobacter baumannii, Escherichia coli, Klebsiella pneumoniae, and in several reports, it has been mentioned that India is termed as AMR capital of the world. So, WHO has also published its priority pathogen list in which critical and high priority pathogens, in which what we are studying is Acinetobacter baumannii, Enterobacterus, in which we have Escherichia coli, Klebsiella pneumoniae, and Pseudomonas aeruginosa. So, till now, many literature have also cited that sepsis, UTI, and respiratory infections are the leading cause of AMR mortality. To understand this, what we have done is to contain AMR, we designed a study in which we have collected total 3 samples from different parts of the country, isolated the MDR bacteria to find the AMR burden at given point of time. And the major samples that we have are from urine, respiratory, and samples which belong to 13 different types of MDR extra pathogens, in which majorly we found are pathogens like Acinetobacter baumannii, Pseudomonas aeruginosa, Klebsiella pneumoniae, Escherichia coli. So, to give a brief overview of the work, what we have done is we have recruited patients at the collaborating centers, collected the samples, then isolated bacteria, did their DNA isolation and the AST profile for phenotype and genome sequencing to identify the molecular profile using NGS platforms and did the functional genomic analysis to identify AMR signatures. This is a study that is published in PNS that where we have found that we have successfully decoded circulating profiles associated with these MDR bugs and try to correlate the profile with molecular profile. And these are some of the pathogens that we found where we are focusing. And this is from all the parts of the country from around 2016 to 2022. And what we have found is that AMR genes are extremely pathogen specific and heterogeneous in nature among gram-negative pathogens. So now what? As we have successfully decoded pathogen-specific AMR gene profiles, we thought to use this data that we have generated to develop a point-of-care diagnostic test. But before that, we need to understand the AMR point-of-care market. So, a business report has reported that the global point-of-care molecular diagnostic market size has been projected to grow at almost $10,121 million by 2030, with North America as one of the lead region and South Asia is the third in row, with infectious disease to be the lead segment and PCR to be the some of the lead companies in the global point-of-care sector, whereas as far as Indian context, India reported as fastest growing regional market in Asia Pacific and is projected to reach $71.4 million by 2030, CAGR of 6.6%, with infectious disease was the largest during 2024. Thus, it shows that it's high time that we need to focus on developing point-of-care diagnostic, which is rapid and cost-effective. These are some of the commercially available gap that we observe focusing on carbon genes, which is relevant for Western countries, but not relevant for the Indian context. Also, conventional diagnostics are slow, costly, and infrastructure heavy, and AMR gene panels are limited and are not specific or relevant. But to fill this market gap, we have thought that there should be some assay which should directly use clinical samples for AMR and pathogen detection. Thus, we are currently validating our diagnostic assay on clinical samples like blood for sepsis, aspirate or sputum for respiratory infections, preparing the cell lines of any thermal cycle, amplifying the specific target using tag primers and visualizing the targets through dipstick. This assay has reduced turnaround time to less than 2 hours and is cost-effective, time-saving, rapid, and can be used at bedside. The panels are intelligently curated with state approach, and thus what we want is, as a science student, kindly help design commercialize diagnostic assay that I have developed my PhD and help AMR-free life. This kit can be used at different sectors like biotech and pharmaceutical industry, public health services, government initiatives, anti-diagnostic programs, breaking the cycle of pandemic through early diagnosis, clinical diagnostics, community health screening, medical camps, military disaster response. Kindly conclude your slides. Some would like to summarize that to understand the circulating pathogen heterogeneity, we have collected different kind of samples throughout different parts of the country. We isolate the bacteria through culturing, did isolation, to identify different type of microbiome. We did antibiotic susceptibility testing. We isolated genome and did sequencing through different NGS platforms. Then did functional genomic analysis to identify unique sequence with functions, and then develop the dipstick. This technology is already different types of samples like sewage and has been used to differentiate between SARS-CoV-2 variants and COVID-19. And now we are developing it on clinical samples. In the last, I would like to acknowledge my supervisor, guidance and support, Dr. Bhabatosh Das, for sample collection, Dr. Dipasri, Dr. Thandavarian Ramamurthy, C.V. Shrikant, and participating sites. For functional analysis, Dr. Raghuram Prakasan, Dr. Shakti Kumar, FJ lab members, THSTI, and my funding agency, UGC. Thank you so much.
Thank you. Comments from investor?
Okay, so my favorite question is TRL stage. So which stage are you in?
We have currently crossed the ideation stage, and right now I am validating my idea on clinical samples.
So which means you would be at TRL 3, 4. Like, is that correct? Manish, help me bridge that gap. I'm still not there. 2 to 3. Okay. But I think it's a wonderful project, and more we do, the less it is, trust me. And please keep up good work. That's all I can say, because I also do not have the clear understanding right now from the science perspective. And I think definitely there is a business, that I'm sure 100%, right? So maybe we need to do a deep dive, and if you can share some data points with us, we'll be happy to evaluate, right? And what is your ask actually, if you can tell me, what's your clear ask for a milestone today?
So currently, since I'm a science student, I am developing this kit. I know the science, I've curated the panels, I know how specific it is, but I don't know the market end, how to approach there, how to reach there, how to commercialize this kit. And so I would like to know that side of the part. And definitely it will be very great if it is actually in market, if it's market.
Of course, before it marks, not many milestones have to be done.
Yes,
but I think it's good to see that you have a clarity, and at least clear your ask. And I'm sure once you share your ask with Manish, then we can sort of check what we can do and what we can't do. Right? Thank you very much. Keep up the good work. Thank you.
Thank you.
Thank you. So next we have Dr. M.R. Kamlapriya, a postdoctoral researcher. This is second last for the day. You have 4 minutes for the presentation.
Alarm. No.
With the latest one.
Yes.
अपना मेरे इसमें लिख के इसमें
Good evening to everyone present. I'm here to present on my patent, which is a biosynthesized nanodrug for anti-HIV activity. Here, my patent is focusing on two aspects. One is nanotechnology and other one is pharmacognosy. And nanotechnology ranges between the size of 1 to 100 nanometer, and materials at this dimension is very effective because of the increased surface area and the workability. So these materials are highly preferred for in vivo applications, for biomedical applications and other therapeutics and targeted deliveries. So I'm choosing a nanoparticle here. Other than that, the plant material, which has ample biomaterials or the phytocompounds that have better activity than the present available chemical drugs, which has so many side effects. So a better lead molecule can be there. Combining this nanotechnology and the pharmacognosy, I want to synthesize a novel gold nanoparticle, which resulted, and first let's check on the problem. Problem is HIV AIDS, which is a global issue affecting more than 40 million people worldwide. And it's been treated with ART, antiretroviral drug, but still there are a lot of impediments like high cost and a list of side effects ranging from mild, moderate to severe. Apart from that, people suffering with HIV have immunocompromised cell structures that leads to co-infecting cancers and other opportunistic infections like tuberculosis. So the trouble is double, and a novel solution that which we come up is combining this nanotechnology and pharmacognosy, which led to the development of a successful gold nanoparticle using chloroauric acid and Camellia sinensis, which is the plant extract used in this patent. Upon characterization, we found that the nanoparticles were spherical in nature, around 15 to 30 nanometer, which is quite a good nano dimension for in vivo applications. And the nanomaterials were distributed evenly without any aggregation, and the structure was crystalline. This led to further in vitro anti-HIV studies in TZMBL cell lines using HIV-1 pseudovirus, and we got some good, consistent, and significant anti-HIV results, for which we further took this for in vivo cytotoxicity studies on animal model like Vista rats. The data from the cytotoxicity studies show that the level of gold nanoparticles are within the permissible ranges. This is quite promising. And feasibility, the synthesis involves just two reagents, the precursor solution chloroauric acid and the plant material Camellia sinensis. Stable, cost of production is less, though there are a number of anti-HIV products available and new medicines are still on the go, but still we are left with DRPs, drug-related problems, even from a recent publication which was 2024. There is a lot of inability such that the available drugs could not reach the HIV reservoir, which further leads to the latent infection to rise up. And the smaller nanoparticles are smarter and efficient, and it can be also used as a targeted delivery in combination with the available drugs or the standalone molecule. In this patent, we have checked it as a standalone molecule. In comparison, we had a market commercial available raltegravir drug, and this patent has proven a better efficacy than the compared commercially available drug. The market size, rapid and simple synthesis of the nano drug, which can be treated for HIV patients across the globe, and the market size is going to be huge and covering public from all walks of life. Value proposition for doctors, a novel drug to treat the chronic illness, improves the strategic medication, healthcare system advancement, and reduces the mortality rate to some extent. For patients, a novel drug to combat the disease, improves health and the lifespan, and reduces the burden of the medical expenses. Yeah, here it ends. Thank you for your attention.
So, I would have liked your scientific data presentation from a gold reference antiretroviral versus your lead formulation, what is nano. So I don't see a comparison. Like, how do you say that it is better bioavailable or it is better pharmacological? So how do you say this at in vivo stage? I'm just trying to understand.
Sir, in vivo, it is not compared towards any drug because we have not developed a HIV model. In vivo, we just check the cytotoxicity of the gold nanoparticle. It was just on the healthy animal model.
No, but cytotoxicity is one part, but then efficacy overweighs the cytotoxicity.
In vitro level, the efficacy has been established in the HIV pseudovirus, but further studies are needed.
Okay, I'm still not able to appreciate why you can't do a comparison of your gold standard drug fed the same animal and then see the overall differentiation in terms of efficacy and toxicity. Like, why can't it be done? I'm just not able to understand and appreciate.
Respect to some commercial nanoparticles. Is that your question?
Yeah, well, that's your IP, right? Your IP is you've developed a nanoparticle based antiretroviral, right?
Yes.
And antiretrovirals are already available in the country or globally, and WHO procures it, and that's the institutional market. All that is very clear, and I don't see much problems with existing drugs. So I'm just thinking that why are you trying to look at nano as one? Second is that why are you not doing a comparison in the preclinical setting itself to see that this is available and ours is slightly better in terms of bioavailability and many more parameters which are important from pharmacological perspective. That data I don't see. What you have is a isolation, which is like silo data. That silo data is not really conveys anything. This is my observation, but I could be wrong.
So with respect to the in vitro studies, we were able to do this anti-HIV screening. And here the focus is the gold particle is not used as a targeted or a delivery molecule, which is the general scenario where people use nanomaterials only or predominantly for therapeutics as a delivery or the carrier molecule. But here, what we emphasize is this gold nanoparticle can be used as a standalone compound or a lead molecule, not as a delivery or a combination molecule. So we were checking the in vitro studies for anti-HIV and we got the significant and the consistent results. And further, we wanted to check the cytotoxicity since some metal is involved, how far it is available and how far it is eliminated from the system, which is a gold standard when we want to propose something in vivo. And so we use this mode to check the cytotoxicity. Thank you, sir.
Thank you.
Thank you.
So next we have the early stage startups, THSTI Life Sciences by Mr. Krishnan. You have 4 minutes for presentation.
So I guess I'm the last person standing here from you and going outside. I'll try to be concise. Yeah, thank you. Good evening, all. My name is Karalmani Krishnan. I'm from THSTI Life Sciences, and we are working on the very interesting area called as cell and gene therapies. What we are trying to do is this. We are simply trying to build a scalable CAR-T IP-based RX manufacturing platform. For those who did not get this title, it simply means this. It means designing a system which can effectively manufacture and deliver technologies, CAR technology, for instance. That is what it means. So the problem statement that we are working on is this. Cancer patients in India have poor accessibility to advanced technologies, affordable advanced technologies like cell and gene therapies. As you can see from the data, only 1% of the CAR-T eligible patients we have treated in last, let's say, one and a half years. So this is a huge unmet medical need. And this is because of 2 reasons. One is affordability, second is scalability of the technology. And this is what as scientists we see. But as investors, they see this as huge demand supply gap. So what we are trying to do is this. First, to address the affordability, it's no-brainer that we have to develop allogenic carts, because in allogenic cart, what happens is this. The cost of developing one batch gets divided amongst 5 or 6 patients as against to autologous therapies. So this is how we bring down the cost. The second is we are doing some science, or we are following science that enables us to distinguish from others. What we are doing here is this. So we are going in allogenic cart, what we are going to do is we are going to use a subset of T cell, which will address the industry problem of having GVHDs. So if we use that subset, we avoid the risk of the GVHDs, number one. Secondly, what we are doing is we have designed a pathway or the system which will allow parallel processing of batches. Means in one go, you can have multiple batches manufactured. So which will allow you to develop 30,000 patients per year. So this essentially means you can go up to 10 times higher what we have been treating so far in the last 1.5 years. So that is what we are trying to do. Once we have developed this, we have this system in place, this can be easily replicated to deploy in the point of care manufacturing, as point of care manufacturing. So this will increase the accessibility to CAR therapy. This is what we are trying to do. So this is a busy slide for, I'll try to explain you. On the left-hand side, we have the regulatory pathway for the development of, and this is the innovation to market plan. So what we are, on the right, we are showing the TRL levels from TRL 1 to TRL 9 or 8 or 9 in market. So what we are showcasing here is that we are at TRL 2 and we have developed initial proof of concept already. So in this journey, what we have done so far is the investors, as a director or the founders of the company, we have invested about 30 lakhs. We got a seed grant of Startup India seed grants of 25 lakhs, and we have also won a grant of 1.3 crores to demonstrate the initial lab scale feasibility here. So this is the result which wherein we have already demonstrated that we are capable of generating antiviruses in our lab, and also we are capable of generating CAR T therapies or CAR cells from used circuit cell lines. So we have also applied for patent. Provisional patent has been filed, and we have published some of the literatures and papers in journals. And we understand that there is a huge regulatory risk associated, so we are collaborating with ICMR, which is supporting us to de-risk the risk that we have. So this is the business model, or what we can say is that, as I told you, there are 2.5 lakh leukemia and lymphoma cases in India. The current cost of therapy is about 30 lakhs. So the TAM becomes 73,500 crores. And we know that 30% of them, these patients are relapsed or refractory, meaning that these patients have already used or exhausted their treatment option and left to die. So these are the CAR eligible patients. So at 13 lakh rupees, which is the current cost of treatment, the SAM becomes about 30,000 crores. So we are targeting for one lymphoma and leukemia cases at 10 lakh rupees per patient. Roughly, we are saying for one product, we will have 500 crores as the SOM. This is the business model we are targeting. It will be obviously a business model wherein hospitals and cancer care centers will have customers, and it will be a perpetual cost structure. And right now we are focusing on India. Once we have succeeded, we can replicate into our markets. So my name is Karumani Krishna. As I told you, this is our team. We have previously developed and delivered products in the market, and we also developed many products throughout our journey and filed several patents. So we know that this is a humongous task and cannot be done by a single company or person. So we have collaborated with clinicians, research institutes, strategic advisors, and labs here. So like AIMS, SIMS, Hetero Healthcare, YDRX, RCV, for example. So right now, our ask is for about 5 crores, which will help us to move from TRL level 2 to TRL level 3. And on the right, I'm showing the fund utilization, how it would look like. I would be really happy to have your feedback, and thank you for your attention.
What's that? Any comments?
There's just so many companies that are coming up in this space. What do you think is going to set them apart?
Very good question. First is science. Develop good quality product, number one, at an affordable cost. These are the two things. And if we make it accessible to patients, then I think any company would survive and succeed.
And what will be special about your science?
Science. So here what we have done so far is that we have plasmids with us. And secondly, we are going to use a subset of T cells, which de-risks the GVHD risk, which is entire industry is facing. So we use that subset of T cells and put the plasmids onto them. This will develop allogenic CARs. Allogenic CAR by themselves, by definition, are affordable, should be affordable at least. So that's our target. Yeah.
Thank you.
Thank you. Thank you.
So this brings us to the end of the energetic pitching session. A big round of applause to the presenter for sharing their work and with so clarity and passion. Three selected pitchers will be felicitated tomorrow on Foundation Day. I request all the investors to submit your evaluation sheet. We are grateful to all the investors for their work and the guidance. We hope this session leads to many successful collaborations. Special thanks once again to IVCA team and Mr. Amit Pandey, Yachna for co-leading these efforts. I now invite all of you to join us for the high tea and feedback outside. Thank you being a wonderful audience. Thank you. Please join us for the photograph on the stage, please, investors. Yachna, Amit.
A gentle reminder for all the dignitaries to join Seminar Hall 1 to give your valuable feedback and have discussion over there. And we render our heartfelt thanks to Dr. Rishi and Ms. Purdy for organizing this event. Thank you.
Make it team, please join us on the stage. Investors, please join us on the stage.
Yes, sir.
My God. 28 हो गया
Thank you all. Wish you a productive interaction ahead and a very pleasant evening. Thank you very much. Jai Hind.